Hi, Yilong. Happy to see you online already.
Yeah.
Okay, this is great. We're still struggling a bit with the audio video in the room. And also for those who tuned in on YouTube already, please be patient a couple of more minutes while we resolve these technical challenges.
Okay, sure, no problem.
Yeah, but the live stream, like the video and the talk by Emma will start shortly. So I guess, yeah, 5 more minutes or so. But it's great to see that you're already in the session, and looking forward to yours as well.
Okay.
Yeah, see you in a bit.
Yeah.
Okay, cool. We're on. Like technology works. Great. Can you unmute my laptop. And the live stream is also working, as just checked. Okay, welcome everybody. This is really exciting to see the room packed with researchers going for all the way to Amsterdam for this GeoMedIA workshop. Really happy with this. Let me welcome you with just some brief slides, and then we move to the exciting first keynote by Emma Robinson. So the purpose of this workshop actually was to gather, to gather indeed all the interested researchers within this discipline to inspire each other, but also to learn from each other's work. And I think we managed quite nicely also when it comes to this in gathering research publications. So we have a proceedings track and an abstract track, which I'll say something a bit more in a bit. But the purpose is certainly bring together experts on geometric deep learning and in medical image analysis, so this focused domain, to hear and learn from invited speakers. So we have Emma Robinson starting off in a couple of minutes, with Michael Bronstein in the afternoon. And we will end today with a panel discussion also to identify challenges and opportunities for further research, and also for us as a community, what should we maybe focus on? And I think now that we're all together, this is a great place to do this. So, yeah, in this workshop, we decided to indeed work with a peer review system, such that indeed you could also submit your work and share it with the field. So we had a full paper track focusing on novel work in this discipline of geometric deep learning for medical image analysis. This is going to be published in PMLR. We already have the publication numbers, and it's on open review, but it will be officially appearing at the Proceedings of Machine Learning Research soon, with a nice team of reviewers across Europe mostly. All submitted works are now today presented as posters, and some of those were selected to give a talk during this session today. So we had the extended abstracts, which are shorter works, where the objective there was such to give you an opportunity to share your newest findings. Yeah. So yeah, this is the program. We're already running behind schedule. It's going to be a tough one. But I think it's nice. So it's sort of concentrated around this room. We have coffee breaks over there. Also the poster session, lunch will take place there, and the drinks afterwards. So I hope you're looking forward to this program as much as I am. As for this attendance, and that's why I'm also this excited, is that we have over 230 registered participants. So at some point we had to cut the registration form, and this is really showing how much interest there is in this discipline. And I think it's like, as a field, we started off without deep learning, and then we had to rely on sensible priors, and geometry is, I think, the way to go. So this field had made huge strides incorporating this into deep learning, and so we're now entering this next phase that we sort of, I guess we, in a short time span, we went over this entire history of computer vision and image analysis development, but then from a deep learning angle. And we're now at this point that we're starting to solve new kind of problems with this. And I think our program nicely illustrates this, which covers all forms of geometry in medical image analysis and deep learning. Yeah, Yama, maybe you can say something about what's going up in the... Okay, so we have submissions. So, I can talk about this actually. So this is just an overview of all the different institutes that submitted. It's a bit a messy word cloud. I mean, because every word is split up, but it's interesting to get an idea, like where all of you come from. Our workshop is also generously sponsored by Alice Unit Amsterdam. So Alice is a network for collaboration in Europe to stimulate and fund AI and machine learning research. And there are several Alice units across Europe, and we're affiliated with Alice Unit Amsterdam. And they generously sponsored this workshop as to allow participants from all over Europe. That's actually what we decided on, to come here via travel grants. And we assigned 30 of those. And I think we covered most of Europe actually. So we have Norway, Sweden, Denmark, all the way down to Italy. And this is, I think, great that we were able to do this. Okay, there's people popping up. Okay, that's where you came from. Scotland, Ireland, Britain, and actually also 2 from the US. This was especially there was one submission by high school students actually, which wrote a brilliant paper. It's in the main proceedings track, and they should also be here today. I'm not sure. Oh, cool! Yeah, they're in the back. That's really nice. Looking forward to meeting you. So yeah, by this funding, we could also have them come over and present their work. So again, this is sponsored by LS. It's a MICA endorsed event, and by the NWO, the Dutch Research Council. Yeah, so since we're running a bit behind schedule already, let's start straight up with the first keynote. It's Emma Robinson. Emma, would you like to introduce her?
Yes. Yeah. Okay. Well, it's also my pleasure to welcome you all here to Amsterdam. It's very exciting to see this finally happen after many months of preparation. Let me just put up your slides here. Yes. Okay. So it's my pleasure to introduce the first keynote speaker of today. That's Emma Robinson. She's a senior lecturer at King's College London, very experienced in brain image analysis, machine learning, and recently also geometric deep learning. The floor is yours. I'm looking forward to your presentation.
Okay, thank you very much. Oh, loud. Am I too loud?
No, it's good.
Okay. Thank you to Helma, Angelica, and Eric for inviting me today, because I am super excited to be kicking off this really stimulating day of talks. So over the next 50 minutes or so, I'm going to talk about some work from my lab, which is looking into how best to leverage geometric deep learning to improve the precision with which we can model the human cerebral cortex in health and disease. So to my mind, the cortex represents a particularly fertile ground for developing geometric deep learning technologies for several reasons. So the first one is, in my informed opinion, the cortex is by far better analyzed as a surface. So the cortex is a thin layer of tissue. There we go. Thin layer of tissue at the outer edge of the brain. Grey matter tissue, shown here on this structural MRI in the darker color. It's highly convoluted surface, home to neuronal cell bodies which are densely interconnected, and those connections underpin complex patterns of dynamic brain activity. So this is a movie of resting state functional MRI on an inflated surface. So we know from non-human primate studies that the cortex is the area of the brain most evolved, and that tells us that it's key to higher order cognitive function. And this is particularly true of the prefrontal cortex. So this is an area of the brain which is highly advanced and at the same time extremely variable across individuals. It's known to be involved in many complex cognitive processes, and it's also implicated in a wide range of neuropsychiatric disorders. So modeling the cortex accurately is extremely important for both biomedical applications and cognitive neuroscience. So for these reasons, over the last decade or so, there's been an increasing number of high-profile papers that analyze cortical imaging data on the surface. Increasingly, surface-based analysis is a cornerstone component of the image processing pipelines for major open data sets. So you've got the Human Connectome Project, the Developing Human Connectome Project in UK Biobank, of which I'm involved in particular. I'll talk about a bit more about these in a minute. And all of these are either directly using or based on the free surfer pipeline, which was developed in the late 90s. So this works by fitting a tessellated mesh to the inner cortical boundary, inflating it to the outer cortical boundary, creating an inflated and smooth surface, mapping this to a sphere, and then learning spatial correspondences between different surfaces using this simplified spherical representation so that we can perform population-based comparisons. So historically, population-level comparisons for the volume, or indeed, so for the surface, or indeed the volume, have always been based on learning dense spatial correspondences based on features of cortical shape, projected to the sphere in the case of surfaces. And so in this space, you assume that all the cortical folding patterns are completely overlapping, and therefore at each location, it corresponds to the exact same cortical region across all individuals, and therefore you can directly compare them. However, it's been known for a really long time now that shape is actually a poor predictor of the underlying cellular organization in, across many areas of the brain. So this is a 2008 study by the Free Surfer team where they had histological data, and they looked at how well you could overlap cytoarchitectural regions based on surface shape-based registration. And it does okay in certain regions in the primary areas of the brain, but when you get to certain areas in the frontal cortex, the overlap is poor. So a cytoarchitectural region is an area that's defined through histology by essentially in a contiguous region with common patterns of neuronal connectivity and therefore assumed to have shared function. On the other hand, there's increasing evidence that if you that the underlying areal organization of the brain is well predicted by the topography of functional activations. So in this work, I developed multimodal surface matching, or MSM, and this allowed us to learn spatial mappings driven by areal features, so features that reflected the underlying areal organization of the brain. So in this case specifically, resting state fMRI, so fMRI when you put people in the scanner and they're not doing anything, and estimates of cortical myelination. And we knew we were on the right track because when you independently validate this on other data, other functional data, in this case task fMRI, you see that functional alignment significantly improves the overlap of key functional activations. So here you've got two tasks, a language task and a working memory task. And when you perform and basically these yellow regions are areas of the brain which are consistently activated across all individuals in the group. And you can see this region particularly here. This is a really important region, highly variable. We're going to talk about it a few times. It's called 55B. And with folding-based alignment, you do a really bad job of learning correspondences across individuals. That means that in downstream analysis, you're doing a really bad job of comparing these across individuals, whereas with functional alignment, you do much better. So using these tools, these powerful tools, we started to be able to build models that are looking to understand the cortex with greater and greater precision. So in this study, MSM was used by colleagues at the HCP to build a model of cortical areal organization. So here they took multimodally aligned data across a large group of subjects, looked at many different features. So you have cortical myelination, cortical thickness, many different functional maps, and generated sharp group averages where they could hand annotate those, looking for borders that were consistent across multiple modalities. And this allowed them to generate the first group average map of cortical organization from in vivo imaging data that closely resembled what had previously only been achievable through post-mortem histology. In more recent work, my PhD student Logan, who's here down there, used MSM image registration to investigate the genetic versus environmental origins of cortical asymmetries. So these are important because on the whole, we know that an individual's left and right hemispheres are generally their shape and organization are highly symmetric, but deviations from symmetry have been implicated in key cognitive processes, and also deviations from normal asymmetry is often considered a common biomarker of neuropsychiatric disease. So in this study, we aligned a large cohort of term-born neonates and used permutation-based statistical inference to look at areas of the cortex which are consistently asymmetric across all individuals, both looking at morphological features and functional maps. And what you find is that many of the key asymmetries that are previously only reported in older children and adults are present at birth. So this suggests that they're genetically primed, and that perhaps, key biomarkers of neuropsychiatric disorders might be detectable at birth. A similar approach is studied here to investigate cortical expansion. So the MSM algorithm has an underlying biomechanical model, so it outputs estimates of cortical expansion in the form of MSM strains. So here we have a small cohort of very preterm babies that are scanned longitudinally. So you have, they're scanned at regular time points, they're registered longitudinally and between each other, and then again you use statistical analysis to investigate those patterns of cortical expansion. And you find that these are not even, rather the primary areas, the areas that are more similar to our primate cousins, are expanding early in the third trimester, but as you get up towards term equivalent age, the higher order areas, particularly in the temporal lobe, are expanding really fast, and this gives you some indication of why babies born preterm often have go on to develop neurodevelopment and cognitive impairments. So likewise, the problem of the impact of preterm birth is considerable, but it's very difficult to know which babies are going to have poorer outcomes. So in this study, again, they use registration of microstructural data to look at regional patterns of cortical to build regional normative models of the microstructural development of the cortex from the Developing Human Connectome project, again looking at healthy term-born neonates. And then you can start to compare preterm babies against it, and you see that the impact of preterm birth is extensive. It's impacting the entire cortex, but from one individual to another, the deviations the deviations from normality are very different. So it's a very heterogeneous and complex phenotype, and it's very difficult, for these reasons, to predict the outcome of these babies. But this is something we really want to do. It could have a huge impact on the long-term outcome of these individuals. So hopefully I'm convincing you that cortical surface based analysis is potentially hugely powerful. There's a lot you can do without it. It potentially has significant long-term impacts for biomedical and cognitive neuroscience applications, but as powerful as image registration-based analysis is, it's inherently limited. So image registration is ill-posed, and in order to be able to learn biologically reasonable solutions, the algorithms are always constrained, always regularized, often by biomechanical elastic penalties, and these enforce diffeomorphism. So they say that the deformation must be smooth and invertible and one-to-one. And that's fine if you're modeling long-term longitudinal development of an individual tissue. But when you're talking about variability of the brain from one individual to another, this assumption clearly doesn't hold. So here you have 3 examples of cortical shape variation. So these maps, the yellow represents the top of the folds, the blue is the depth of the folds, and you can already see that these are highly variable across individuals. If you then go on, these are 3 examples of individual HCP parcellations that were achieved or were obtained from the group average HCP using mean machine learning. So they've registered. These are as good as we can do, getting these brains aligned, and yet there's huge amounts of residual variability. The only way we could get these is by training a classifier on a regional basis, looking for subjects whose brain imaging data closely corresponds to the group average for that reason, train a classifier on those to learn a multimodal fingerprint, so it's an MLP, and then predict those in new subjects. And what you see, for example, in this 55B region in particular, it's here in orange, bordered either side by the frontal and posterior eye fields. Most subjects have this topography. In some subjects, though, 55B is split, and the PEF and the frontal and posterior eye fields are merged. You can't learn a diffeomorphic mapping between this individual and this individual. So, we need new tools in order to be able to interrogate the cortical surface with higher levels of sensitivity and precision, because, signs of complex neuropsychiatric disorders such as autism and schizophrenia and focal epilepsies of the cortex, they're extremely subtle and they're really difficult to detect. We do know already that deep learning in 2D and 3D offers us some level of it offers registration-independent analysis. So we don't need to have pre-aligned all of our natural images in order to be able to detect an object in an image. So my pitch is that surface deep learning has significant potential here to have impact. And the other reason why the time is right now for developing geometric deep learning tools for the cortex is that we're getting an increasing number of large, for imaging in imaging terms, data sets of cortical imaging. So we have the HCP that's got 1,200 very high quality data sets acquired from young adults with an hour's worth of resting state. You have UK Biobank, which we've got 40,000 subjects currently, but the plan is to acquire 100. These are closer to clinical quality. So in their case, they have 6 minutes of resting state. But as I'll tell you in a minute, it's surprising how generalizable these things are between the HCP and the biobank. And the DHCP is a really, it's a smaller data set, but an extremely important one, and it's good because the changes that are occurring during that time are so stark, so it's less subtle things you're looking for. So all of these data sets are multimodal. You have macro and microstructural, so shape, microstructure, and functional imaging data. They are surprisingly comparable across each. Shape is more or less fixed once a baby gets to term equivalent age, so you can compare the shape, allowing for certain image acquisitional noise. You can compare them across all of these populations, and the function is surprisingly comparable between the HCP and the UK Biobank. The full cortical processing for biobank isn't currently freely available yet. We're in the process of developing it, the pipeline. All of these data sets are theoretically open. So, well, they are open. So all you need to do for the DHCP and HCP is sign a data access agreement, and then you can get the data. For the biobank, you need to write an application and pay a relatively small fee. But there's a bit of a lag. And as I said, not all of the cortical imaging data is yet available. So with this in mind, we wanted to set out to benchmark different geometric deep learning frameworks to see which ones were most appropriate for cortical surface analysis. And hopefully I don't have to convince everyone here that this is non-trivial, because there's no one implementation of surface convolution CNNs. And that's because they're non-trivial to implement. One of the key features that makes Euclidean CNN so powerful is this feature of translation equivariance. So when you translate a view across an image, the view of the output of the convolution is also translated, and this allows networks to be translation invariant, and so you can detect objects wherever they are in the scene. And as I've already said, for the cortex, this is potentially really powerful because image registration just isn't giving us what we need. So for a genus zero surface, what we need is rotational equivariance. But if we implement a spatial filtering in the same way as we would do for 2D, we run into problems because there is no global coordinate system. And if you transport a filter over 2 different paths over a sphere, then they end up at different orientations. So one option that has been taken up quite strongly is to instead represent convolutions in the generalized Fourier domain. So this is because the Fourier transform of the convolution of an image and a filter are equal to the product of their Fourier transforms. So as long as we know what the generalized Fourier transform is for the domain of interest, then we can implement it in this way. So for spheres, you have the spherical harmonics. For the rotational group, the 3D rotational group, you have the Wigner-D matrices, which was what was used for S2CNN by Tucker Cohen. And for a general manifold, you have the Laplacian bases. The problem is that if you implement a full spectral implementation, then it's extremely costly, and it's computationally costly. It's going to take a long time to run, And also the filters that you learn aren't spatially fixed. So instead, a number of methods cropped up that proposed learning polynomial approximations for these. So the first one was CheMedIA. The problem with these is that you're learning sort of concentric, symmetric, simple features that are sort of rotationally invariant and but not very expressive. There have also been approximations of 2D spatial filtering approaches that have been proposed. So there's MoNet, which is a mixture of Gaussian's approach, where the Gaussians are aligned with the polar coordinates on the surface. And specific to the cortical imaging domain, a few years back was proposed spherical UNet. So this leverages the regular tessellation of an icosahedron. So most vertices in an icosahedron have 6 neighbors regularly spaced like this, so you can fit a filter like this and then implement a convolution similar, exactly the same as a 2D Euclidean one. So it's a matrix multiplications between these unwrapped filters and the local imaging data, but then your filters are the orientation of your filters is very position dependent, and as we'll see, this is a problem. So we thought really hard about what data to benchmark this on. And in the end, we went with the developing data, even though it was the smallest data set. And there were several reasons for this. So one, the DHCP is released by King. So we have complete control over it and releasing its derivatives. The other thing was that when we thought about what we wanted to test, we wanted a series of regression benchmarks that were achievable. So the DHCP provides two phenotype regression tasks. One is predicting the baby's postmenstrual age at scan, so when they were scanned. And this should be dead easy because the changes occurring over this time are really stark. So it's a baseline test to check whether your model is working at all. Then you have regression of gestation age at birth. So this is seen as a correlate of prematurity. So can you predict how premature a baby was when you take their scan at birth? So we have a smaller cohort of preterm babies, and 94.5 of those were scanned at term equivalent age. And this is obviously a clinically relevant phenotype because we want to know how much these babies' brains deviate from normality at term equivalent age. It's an indicator of how well they're going to do. If we were instead to take the types of phenotypes that we could get out of the HCP, these are they're much more difficult to predict. So trying to predict an individual's IQ from a noisy cognitive test is difficult, and the best correlation that people tend to achieve is 0.4. So it's a difficult thing as a benchmark for developing geometric deep learning tools. And as I said, Biobank is not really freely available. It's not as easily freely available. We also did segmentation because this tends to be the default. This is the benchmark that everyone goes for, but as I will say in a minute, I think it's not really the most discriminative. It's not a difficult task. So we went with data. At the moment, we're just looking at morphological, structurally driven features, so cortical thickness, cortical folding, cortical myelination, no function. We mapped everything to the sphere because this is what we're used to doing. And the thing is that for reasons that I explained before, we don't want our models actually to be too constrained by shape because we know that there is a dissociation between shape and function, and we're interested in the function. We also train the algorithms on data that have been pre-aligned with MSM and the original native non-registered space because we wanted to see how well it would perform if it wasn't aligned. So I'm not going to go through all the results. The paper is here, and Abdullah is here if you want to talk to him about it. But everything was benchmarked by training and testing with rotational augmentations. On template native data, as I said. The main takeaways are that for cortical phenotype regression, expressivity of features is important. So the best performing network overall, on all tasks, is spherical UNet. So it's the most expressive one. And it was the best performing in all tasks. The problem is that it has no rotational equivariance. So when you test it on unseen rotations, the performance is poor. And because the network size is fixed, even when you train with rotational augmentations, it can't learn enough variability in order to predict it. And our data sets aren't big, so we don't really want something where we have to learn through augmentation all the possible orientations of our filters. I should say S2CNN here is slightly handicapped because it's so computationally intensive, and because these are the architectures in terms of the number of features learned are all fixed, it couldn't learn as many filters as all the others, so perhaps there's quite a lot of room for it to improve. But the other worrying thing is that actually these methods aren't really starkly improving over simple baselines. So if you take the data on the sphere, conformally map it to a plane, and then train a ResNet, then actually that's the best performing one on native space data. So we've still got some way to go in developing suitable, optimal techniques for learning on the surface. So reiterating what I said, yes, we did benchmark on segmentation, but these are folding-based parcellations of the brain of regions that are largely consistent across individuals. So with the exception of these Brodmann regions down here, if you just predicted the group average, you would do a good job. So whatever you do, whichever method, pretty much, except for this one, SCNN UG, which is basically hand engineering features by learning the gradients on the mesh and the Laplacian, you do a good job. We did compare against networks which explicitly encode shape. So there was a point net cloud network and a spectral embedding, where here you take the Laplacian embedding of the shape and then you train simple filters in the embedding space. And our feeling from that was, I mean, when it comes to prediction of PMA at scan, babies' brains are growing really fast, so sure, you can do a really good job with shape. All of these are within the range of error of an ultrasound measurement. So they're all performing really well. But when you look at more complex phenotypes, even though these methods are also given the myelination and the thickness, they are not performing as well as methods that where the data has been projected to a sphere. We also started to look at interpretability through occlusion-based saliency maps, and from that, S2CNN starts to come into its own. So here, these are the features that we're interested in. We know that in late gestation, myelination is of the somatomotor strip, and this area MT is happening rapidly, and the folds are all there, but they're getting sharper and deeper. S2CNN, this is when you occlude all features simultaneously. This is when you occlude each one. It's clearly picking up on the myelination and the increase in the sharpness of the cortical folding. So cortical folding is also reflected in the thickness. So the overall conclusion is that these methods are all essentially trading off complexity, expressivity, and rotational equivariance. And for cortical imaging, our feeling is that we need expressive filters, we need rotational equivariance, and we want to build pretty complex networks, so we need the computational complexity reasonable. So looking at all the different methods that we had, we sort of came down with saying that maybe MoNet is the best of most worlds. It's generating predictions that are consistent on native and template spaces and with rotational augmentations. It does a reasonably good job, and it's not very computationally heavy. So these all formed, this work then went into submission for a MICI challenge, SLCN, which we ran for the first time this year. The target was prediction of gestational age. It was set up on Grand Challenge. The data is available on GIN, but you have to sign the DHCP access agreement and prove to us that you have before we can release it to you. So you have to send that to me. And, I think we had some submissions. It was a learning process. It took us a while to get the Docker, an example Docker set up. But we're quite keen to build on this next year, perhaps including more functional imaging data so that we can start to predict more complex phenotypes, like we have information about what the outcome of these babies were when they come back at 18 months. So with all this in mind, we felt that we now had the tools to start developing geometric deep learning applications for ourselves. So I'm going to go through some of these. So the first one is by Logan. All the students who've contributed to this talk are here today, as I said. And here, again, as I said, we felt like we were going to start by using MoNet. So all of these, well, most of the applications you're going to see today are using existing geometric deep surface convolutional methods for the time being. So here, Logan was looking at using MoNet to project the HCP parcellation, the individual parcellations from the HCP to UK Biobank. And this is potentially hugely powerful. The Biobank comes with genotyping data. It's got a huge battery of cognitive test information, and these are healthy aging in adults, and we're interested in predicting which ones of those are going to go into neurodegenerative diseases. There's a lot of information on mental health. So if we can get because there's huge amounts of data as well, so it's a very high-dimensional problem with, so even though you've got like 40,000 data sets, it's still a dimensionality reduction issue. So if you can generate features that closely correspond to the underlying cytoarchitectural organization of the brain, you can see when you average the imaging data within these features, it makes sense. Then you can start to build, you can start to look at the genetic origins of the topographical variation and the links to mental health and neurodegenerative disorders. He used a MoNet training, a MoNet like the HCP, a MoNet for each region, many different features of the cortical shape, myelination, and function are passed into this network, and dropout is used to learn a probabilistic map, which is then thresholded. And the results are really encouraging. So though Biobank has much lower quality resting state imaging data, much lower quality, you're actually, through like using MSM registration, followed by a simple histogram normalization, able to take a network that was trained on the HCP and apply it to the Biobank and get parcellations which on average are highly consistent with the HCP. So this is the overlap of the borders. And better than the HCP, they have this MLP method, which was good, obviously, and powerful. It allowed us to learn the variability of these topographies across individuals, but actually it's quite noisy and it has a bit of a loss rate. So often, in a number of cases, it cannot detect these regions in an individual. Whereas when you use MoNet, it's got a much higher detection rate. So that is really encouraging. But it's not just predicting the group average, it's still able to predict these atypical topographies in subjects where the underlying data supports it. So that's looking really good. So the next work I want to talk about is developing tools for deep discrete image registration done by Mohammed Suleiman, who has a follow-up talk on this at 2:15. So here, this framework takes inspiration from MSM, where again, so MSM is a discrete optimization-based method where rather than allowing this low-resolution control point grid to deform however it likes, it has to learn to predict which of a finite set of labels the point can deform to. And this makes it much more robust to the local minima, which is a problem with functional imaging because it's so noisy. So this component of MSM is quite strong. So this is adopted within DDR, but your label space, unlike MSM where you can only learn a few labels at a time because you don't want to take the label space outside of the resolution of the control point grid to sort of enforce the diffeomorphism, here you're doing it in one shot. So you have a huge number of labels across the surface and you're trying to predict which one, where the image should go, so based on a data similarity term. So you want to move points in the moving mesh to overlap the features as much as possible with the target, but subject to this regularization term that encourages neighboring points to deform together. And this is implemented as a conditional random field RNN, taking inspiration from Matthias's paper from Mikai in 2020. I think there's sometimes some concern or about learning-based registration, that it won't generalize or it won't perform as well as classical-based approaches. But actually, when you benchmark it, you see that DDR is extremely competitive. So this is the equivalent form of MSM, which has only a pairwise regularization term, and it's strongly outperforming that. Here is what's known as MSM strain, so this has a higher order biomechanical penalty, and the green box is spherical demons, which is essentially the best method for aligning cortical folding, but doesn't really work at all on function. So here, these are the results for DDR across subjects. So you're looking at, on average, how similar the folding maps are, how well they overlap, relative to how distorted or how unsmooth the registrations are. And DDR strongly outperformed the S3 Reg, which is a learning-based registration algorithm based on spherical U-Net. And spherical U-Net is struggling because of its lack of rotational equivariance, so it has to sort of hack a solution at the poles. DDR is smooth as the best classical methods we've got, and it's learning really strong matchings. And as I said, a follow-up talk at 2:15. Now, I know I've said that, we don't want image registration as it is, but learning-based registration starts to open up the possibility of trying new things with image registration, starting to think about topology breaking transformation, starting to think about learning the space of variation. So here, Eurong took it, and because it's fast, you can run all the pairwise registrations between all examples in the HCP. And then you get a similarity matrix, which you can generate a hierarchical clustering from, so that you can then register subjects that have similar shapes up through the hierarchy until you start to register branches that are very different cortical folding patterns. And so you're bringing everything into one alignment, where the overall group average space should be sharper, but you're also able to start to pull out variants of cortical folding. So these are the 30 clusters of cortical folding variability just in the frontal lobe alone, and they agree with the variants of cortical folding that have been anecdotally reported in the literature, or only anecdotally reported before, so no one was able to compare them in the same image space. And what we were hypothesizing was if we could do a much better job of aligning cortical folding, the atypical cortical folding patterns, then maybe their functional, these functional variants would cluster out, and the folding may follow function in this instance. But actually what we're finding is no, there really does seem to be a dissociation between function and folding, which is a really important result, because it shows us that we really do need to go further and develop new tools to be able to precisely compare the cortex. The regions for this, I'm not clear. I mean, I guess cortical shape is fixed at term equivalent age, but your brain, your cognition is developing significantly post there. In the first couple of years of life, your neuronal connections overconnect, and then you synaptically prune, and there's a lot of development going on after the point in which your brain shape is fixed. So in this work, Simon, who's also here, basically we said, okay, it doesn't look like surface convolutions are currently giving us the level of performance that we want, so maybe we should take a different approach. So here he translated vision transformers from 2D domains to spherical domains. So in this paper in 2020, what they did was take an image, 2D image, separate it out into patches, and then learn classification as a sequence-to-sequence learning problem. And here we're translating the same thing to surfaces. So we learn patches from low-resolution icosahedrons, flatten them out, pass them through a linear embedding and position embedding, and then through to multi-head self-attention and feed-forward layers. So this is exactly the same network architecture as early vision transformers. So that was published in middle 2021, but since that time, Simon has developed it further. So here's an surface implementation of the master autoencoder, which was shown as an important pre-training algorithm for transformers. So you mask out a lot of patches, you train an encoder on the patches that you haven't masked out, and then a decoder is expected to reconstruct all of the image, including the patches that it hasn't seen. This is work in progress. I'm embarrassing Simon by so these ones are still running. But I think the key takeaway here is these are the results from the middle paper. And since that time, more augmentation, online augmentation. So everything with surfaces is more complicated. So implementing like simulating elastic deformations and things, etc., is not quite so straightforward. But this is all implemented now online. So you've got nonlinear and rotational augmentation and master autoencoder pre-training. And you start to see that the performance on gestational age is really getting down below 1 week's error, which is really good. Yes, spherical UNet seems to still be better, but these are the variants overrun. So sometimes it will give you a really good result, and other times it'll get stuck in local minima, whereas with the transformer, the performance across runs is quite consistent. And clearly, it's the best performing method on native. So I think this shows really strong potential. And the other thing that you can get out of transformers is that you know, the attention maps are inherently interpretable. So here you've got 2 different subjects. So this is where it's used. This is used to predict IQ in adults. And like I say, you know, doing now the performance is actually getting up higher than this, around 0.4 over 0.4 correlation. But that's really as good as you can do. The attention maps are different for individuals, different across heads, and they're picking up key regions of for the cortex that are known to be involved in processes like working memory and decision making. So this is the last one. This is back to Abdullah. So we took everything that you learned from the benchmarking paper and started to say, well, can we go beyond classification and start to build generative models of development? And the answer to that is yes, you can. So in the first paper in MRUA that was published in the summer, he used a cycle GAN. So at this point, the only thing you can do is move a brain from low scan age to high scan age without precision over the exact time point that you're moving to, using a cycle GAN implemented with graph convolutions. But the important thing was that, yes, you can't move it to exactly its follow-up age, but you can preserve its individual brain shape. You're not predicting the population average. You're not making its brain change shape. And that was actually really, really difficult to do. More recently, and he's got a poster later on, and I really encourage you to talk to him, he's extended that to a 3 cycle. So if you make it move multiple steps, it encourages it even more to preserve the individual brain topography and variable shape. And so when you then go on and test it, so if you train an independent regressor, a MoNet regressor of brain age, and see whether it's doing the simulated brains are doing a good job of fooling it, like it really does an excellent job, except for in the sort of areas where we've got very limited data. And these are the visualizations. So here you've got the ground truth scans, and these are the simulated scans. And you can see that it's clearly smoothly aging the baby's brain to the point where you've got much finer detail at 44 weeks as the folds are getting sharper and more detailed. But it's preserving these functional individual folding shape. And similarly for cortical myelination here, we expect that at this late gestation, the myelination of somatomotor strip should be happening rapidly, and the prediction of myelin is again closely resembling what we should expect in the individual. So that's it for me. Thank you for listening. If anybody is interested in these types of applications, we have a large number of PhD programs, projects available. So we're thinking about extending the surface transformer techniques to cardiac applications. This is a project led by Daniel Rocket from Imperial. Shape analysis of deep brain structures for early diagnosis of Parkinson's disease. Propagating the Allen Atlas genetic transcription model to imaging data and trying to better understand Alzheimer's disease with physics-informed networks, and looking into early biomarkers of a neurodevelopmental impairment from babies that have been born with congenital heart disease. The applications are open now, and I think they close early December, so it's important to get on it. And these schemes are always looking for industry collaborators. If anyone wants a nice, challenging, but long-term really impactful potential application for geometric deep learning, this is one. So that's it from me. Thank you so much for having me. And thanks to all my team that have contributed to it. They're here in the front row. Please come and talk to them.
Thank you. Thank you very much, Emma, for this very amazing presentation. Very nice overview, and even some cutting-edge work with numbers that are still running. So thanks for sharing that. Are there any questions from the audience for Emma? Yes, there's a question here in the front.
Really great talk. I was just wondering, when you talk about native space, is like nothing preprocessing or normalization, also no SCPC and nothing about normalization?
The native space is the space in which the data was acquired prior to spatial normalization. The image features are intensity normalized. Is that what you were asking?
Yeah, thank you.
Could I ask a follow-up question on this? So the native space means that we do not do the registration, is that correct?
You don't do the registration,
Yeah,
so
okay.
all of the image data sets vary with both a global transformation and a nonlinear elastic deformation.
Right. So do you think this is the way to go, to directly, indeed, I guess the answer is yes, but so does this sort of bypasses the need for, well, accurate segmentation or registration tools? Yeah.
So I think there's several things. So one is that, I mean, you could probably reduce the variability. I mean, you haven't got huge data sets, so maybe you want to reduce the variability that you need to learn by doing image registration. But if you do, you need methods that are robust to, that are not dependent on that image registration, because there will be residual variability that cannot be aligned. And when it comes to many neuropsychiatric disorders, things like focal epilepsy, you have these tiny subtle lesions that are really difficult to pick up. They're really difficult to see by eye. It takes like many years of training for clinicians to be able to spot them. And so you need methods that, and often when you do in registration, what you're doing is you're comparing the bottom of a fold with the top of the fold, and you just can't, there's so much noise from the image registration that you can't see anything at all. So you need methods that are robust to that variability and that are able to pick up very subtle differences across the population, I think.
Right, thank you. Yeah, brilliant. So there's more questions here.
Hi, thanks for the great talk. You made a good point of that diffeomorphisms are sometimes wrong even for longitudinal data from the same subject. Because you might end up having one-to-many mappings or something like this. Did you consider this also for the supervision of the registration algorithms? I guess then it's also difficult to get like any ground truth for
I think it's
supervising.
difficult. I think where you'd want to go is have some level of constrained allowance of topology breaking. So if the evidence from the data is really strong that you should break topology, then you should do it. But obviously, it's the only way you can. There is no ground truth. We can't take their brains out and dissect them. So it's the only way you can really get a feeling of whether that's right or not is through qualitative assessment by sort of neuroanatomical experts.
Okay, thanks.
We have a question here in the front.
Hi, first of all, thank you very much for the great presentation. I'm wondering, because now that you talk about the registration, so some of the unnecessary and sufficient conditions for registration is diffeomorphism, and sometimes it's difficult to achieve it. So majority of deep learning networks, they don't achieve, from the mathematical point of view, strictly the diffeomorphic transformation, because they lack of actually a very explicit constraints to regularize that. So I'm wondering, what is your message on that? Is it like the need for more mathematical tools in that regard? Because deep learning can take on that, but that not necessarily, or what we see so far, not fulfill this.
No, so if you look at like diffeomorphic voxel morph, it's not fully diffeomorphic,
yes, exactly,
on the testing data.
Exactly.
So DDR, the results are, there's no negative Jacobians, and that's the data. So I think we don't put any explicit constraints in there because we don't want to enforce them. We want the method, the result to be as smooth as possible, but to open some possibility for diffeomorphic constraints. So I would not be trying to put them in, I think.
And what would be the reason not to enforce the property?
Because you can see from the data between individuals, areas such as 55B, that the only way to map from one individual to another would be to break, you would have to basically fold the mesh, break topology, or split it.
And there will be like a not necessarily like a diffeomorphic, but some constraint like a homomorphism that actually constrain the topology. Might be good on mainly on the message on need of more explicit constraints to
Yeah, I
guarantee
think that you need some some constraints to
Yeah.
to generate the results that you're looking for sure. But so I think that's where we want to look next. But it's a combination of saying, can you develop more advanced image registration methods, or could you, or can you use generative modeling to learn a space of variations that would allow you to pick out the residual, the interesting points, you know, from the latent space.
Okay, thank you.
So there was a question up here.
Hi, thank you for the nice presentation and the big summary. I have a naive question regarding to the approach of the non-Euclidean case, what you have with the local charts. Did you show one picture where you have one chart, but did you also try to extend this classification problem by adding also one more parameter, the atlas parameter? So just asking the question, I have one atlas and my sphere, and another atlas and another sphere, and doing convolution, getting down one more, reduce the dimension, and ask yourself what could be the group acting on to somehow identify the.
Are we talking about like group equivariant networks and looking for like the right groups to reflect the kinds of variation we're seeing?
Yeah. Just extend the question by adding the atlas also to the sphere.
I'm not entirely sure. I think we're definitely interested in these sort of group equivariant methods that are equivariant to more types and groups of variation, but I think it's quite difficult. It's not, you can't say that it's just, I don't think there's some restrictive group of rotations or something that will give you all the variability that you need. Does that answer your question?
Yeah.
Not quite sure what you mean by atlas. Atlas to me, it'd be like a population average image of a brain. So I think we're talking about different things.
I mean an atlas in the sense of manifolds.
Yeah.
So you have your sphere and you have atlas of your sphere, and then you show that the convolutions somehow can don't capture all the information because you have just one atlas, one chart.
Yes.
And the idea was somehow extend, take more atlas, and
I think we're definitely thinking about it. Yeah, I think we need something else.
Right.
We have
Thanks.
time for one final question here in the front.
Hi, thanks for the talk. So on that last paper that you presented using CycleGAN for subject-specific age regression, how did you quantitatively measure the that subject-specific features were preserved? Like, did you have longitudinal data for that, or?
Abdullah's here, so it's PSNR. What was it?
Yeah, so a couple of them. PSNR for that paper, but later on I've used a lot more. I've used structural similarity and stuff.
So you're trying to see how consistent they are with their follow-up scans through various measures of similarity measures. Also seeing whether you can fool a regression network, whether they are of the right age. And then obviously with everything you do with brain imaging, you have to qualitatively, you have to QC it visually.
Thanks.
Thank you once again for the very nice presentation. I think we'll see much more of your group also today in the poster sessions and the oral sessions. So that's great. Let's thank the speaker one more time.
Thank you.
Thank you, Emma.
Yeah, and now.
Yeah, may I say something? Now, so this is the kind of interactions that I really hope for in this workshop. Like, there's a lot of, you see that it immediately sparks new ideas for future research, and it's great that we have this poster session later on, such that you can go into further depth of this. So sorry for hijacking this.
No,
go ahead. Yeah.
No worries. I should take the mic from you. No, we actually continue now with the first oral session. So it's an in-person event because it's exciting that many of you are here, but there are circumstances that someone cannot be here personally. So the first presenter is actually here via Zoom. So I'm looking at the AV team in the back if that will work. Yes. So the first presenter is Yilong Yang from University of Southampton, and he will present his work. If you can, maybe we can see your screen as well.
Yeah.
the work will be on a scale equivariant UNet for histopathology analysis.
Sorry, wait a moment.
And good to know, maybe for the presenters of an oral presentation, if you are presenting, it would be good if you can go to the guys in the back during the previous talk to be mic'd up, etc., so that we can continue smoothly. You can connect your laptop up here. And for the pitch session, we have very different instructions, but we will tell those later. Are you able to share your screen, Yilong?
Yeah, I'm trying to share.
Okay, there we go.
Okay, so can you see my screen now?
We can, but maybe you can, yeah, so you have to still start playing, I think.
Yeah.
And then maybe swap displays.
Yes.
Now we're good to go, okay.
Okay, so morning, my name is Yilong Yang, and I'm presenting scale equivariant UNet for histopathology image segmentation. And this work is done with my supervisors, Sasan and Sri. Okay, so a function is equivariant under some transformations if the output always transforms in a certain way when the input is transformed. And in this research, we focus on scale transformations and study functions which are equivariant under them. And in digital pathology, scale variation occurs when specimen slides are scanned at different magnifications to be digitalized. And the CNN trained on images at a specific scale generally cannot generalize to other scales, which greatly restrict the capability of computer-aided diagnosis models. For image segmentation tasks, the prediction should scale in proportion to the input, making scale equivariance a necessary property. And we aim to encode the scale equivariant property into the 2D convolution domain, in a 2D domain convolution. And also we hope that the scale convolution, the scale parameters can be adjusted during training to adapt to data sets and free us from hyperparameter searching. In order to process images on multiple scales at the same time, we utilize the Gaussian scale space theory to construct filters. Firstly, we randomly initialize some sigma values with each of them drawn from disjoint ranges and then calculate the filter basis. Then the convolutional filter is constructed as a trainable linear combination of filter bases. And here, alpha is the trainable coefficient, which is shared among multiple scales to guarantee that filters are the same in shape and only differ in size. And now here, the filter basis can be adjusted as we allow sigma to be trained. Here is a two-layer example to show how the pipeline of training a scale equivariant network. So the input is convolved with multiple scale filters, and the result in features that correspond to different scales. Then all subsequent layers process these features simultaneously and independently. In the last layer, each pass or each branch generates a prediction map y hat, which is used to calculate the cross entropy loss. we learn some over these loss functions and use back propagation to train a network. Then we just simply replace the standard convolution layers of the UNet with the proposed scale equivalent layer, and then calculate the model's segmentation performance on two histopathology data sets. One is for breast cancer semantic segmentation, and the other one is for nuclear segmentation. Before going to compare the performance with other state-of-the-art scale equivalent models, we first check the performance of each scale branch or each scale path. So this table demonstrates that the best prediction shifts to the branch with a larger sigma value as the scale of image increase. And this is consistent with our intuition that to capture the same information from enlarged images, filter size should also increase. And then all right, so suppose we have gamma scales in the model. Although in previous slides I showed for parallel paths, but pretty generally, suppose we have gamma scales in the model. So given an image with unknown scale information, the model will produce gamma probability maps. And here we explore three strategies to generate the final segmentation map. The first one is just take the average of a softmax output, and the second one is PDIS. PDIS denotes per pixel prediction selection based on prediction confidence. So for each probability map, we consider the difference between the largest and the second largest class probability as a measurement of the predictive confidence, and choose the most confident prediction as the final label. And right, so lastly, to mitigate against the concern of an incorrect prediction made with high confidence, we propose prediction ensemble strategies to weight multiple predictions based on their confidence. These two figures show the per scale test performance of different approaches on two histopathology data sets. The performance of all models is similar when the test scale is close to training scale. However, as the test scale moves away from the training scale, the performance of the standard CNN drops significantly. Although the performance of season disco and our proposed scale equivalent network drops as the test scale changes, these models look more robust to scale variation than convolutional neural network. And as observed from the table, the proposed method with prediction selection and the prediction ensemble strategies outperform all other compared methods, and it also achieves the lowest equivariance error. As we mentioned before, we allow the scale parameters sigma to be trained in disjoint ranges, and here we run another two evaluation experiments to show the effectiveness of imposing range constraints. So as shown in the table, model trained with range constraint outperform models with sigma being fixed or being trained completely freely on both data sets. And then the model trained with fixed sigma values outperforms performs slightly better than the freely trained one. Here we also show the learned scale parameters of layers after training is finished. So the left two figures, figure A and figure C, show the sigma values of models trained without range constraint. So as can be seen, allowing sigma to be trained freely result in the case that multiple sigma converges to the same value. And this is detrimental to the feature representation of a model, since the same sigma means that the same scale of the generated filter, therefore features are redundant and are not scale equivariant. And this is the reason why the model trained without range constraint demonstrate the worst performance. And in figure E, we freeze sigma values without overlapping. Therefore, multiple scale filters can be constructed to extract features from different scaled images. And this can be the reason that the fixed sigma, the fixed model outperform the freely trained one. However, the manually selected sigma may not be the optimal choice that fits the dataset best. Moreover, the optimal state of sigma values may vary from dataset to dataset. And these observations motivate us to train sigmas in disjoint ranges. So overall, our experiment demonstrated that the proposed scale equivariant layer can achieve a state-of-the-art performance on images presented in unseen scale, and also allowing sigmas to be trained empowers the model capability of finding the dataset adapted scale parameters. However, since we learn the sigma training, the Gaussian derivative filter basis should be updated after each node of backpropagation, which is more computationally expensive than using a pre-calculated filter basis. And also for now, the scale ranges are manually chosen. Okay, so thank you for listening. Any questions?
Yes. Okay. So we have volunteers. Can one of our volunteers volunteer? Yes, Chase. So maybe you can give the mic to. Sorry, there was someone there who was just slightly before you. So.
Hi, thanks a lot for the presentation. It was really interesting. So I have a question with regards to like how this scale equivariant network would work on the problems that are not just detecting nuclei, but for example, when you need to classify like cancerous tissue versus non-cancerous, because in some diseases, the nuclei appear larger at the same scale. And so I wonder how this equivariance was influenced this difference between classes, which is actually a sign of disease in particular cases. Yep, thank you.
Yeah, so if my understanding is right, you are asking that if these models are used for segment tissues from segment different type of tissues, and the different type of tissue may have different scales. So will this method show better performance?
So for example, some cancerous tissue, like sometimes it can have enlarged nuclei. And if you want to, for example, distinguish between classes of normal tissue and like cancerous tissue, how would this scale equivariance influence this difference between these classes?
We haven't I have an exact example to show the strengths of this method, but maybe we can try later to see if this demonstrates better.
Okay, thanks.
One more question here.
Yes, thank you for the great and interesting talk. You were talking about the distribution of the sigma parameters, and I was wondering, did you, when you trained them freely, did you also consider some kind of regularization, or was it completely freely trained?
It's just completely free trained without any regularization, without any normalization, just completely freely.
Thank you.
Yeah, thank you.
We'll take one more question here on the left.
Hello, yeah. Did you try benchmarking against, or just comparing to cropping and then rescaling? Like, is there an advantage to having I presume it would, but an advantage to having a scale equivariant network compared to augmentation? Also wondering if you can kind of reverse this question and then say, well, maybe it's, because if you rescale without, if you try and do what I'm suggesting, right, you end up with poorer resolution. So could you rephrase this whole thing as not just scale equivariant, but actually resolution equivariant? Because there's a lot of issues in image processing when you have different resolutions, and then some networks just stop working. So yeah, that's kind of two things I thought would be quite interesting. Did you try that?
Yes, thank you for your suggestion. We haven't tried that, but we noticed that yes, just rescale image will lose some information, but we haven't tried the experiments you are saying.
Give it a go, yeah.
Some good advice there. Thanks a lot, Yulong, for joining us remotely. Let's thank Yulong one more time, and then we...
Thank you.
Then we proceed to the second speaker of the session, who I know is here because I've seen her. So it's Renfai Yu, who's over there. And she will talk about group equivariant networks for diffusion-weighted image analysis. So something completely different, but very much in the realm of geometric deep learning.
Hello. Hello. Yeah, great. Huh.
I think it's not you, it's us, probably. 1 second.
Okay. Oh, okay. Is it shown there? No. I think we're putting us even more behind schedule.
It's fine, we have a magical 15 minutes in the afternoon so that we discovered yesterday night, so it's okay. Misschien kun je hem ja, denk ik wel. Kan je haar microfoon doen? Nee. Can you airdrop the presentation to his laptop?
AirDrop, yeah. Rent failing. Oh, okay.
No, this. You're okay like this? Okay, we're all set. Thanks for your patience. I think it's just a slider, maybe.
Hello, hello.
Yeah. Okay.
Yeah,
So,
sorry for the inconvenience. It's we're really behind schedule now. So thank you for coming to this presentation. And my name is Renfei Liu. I'm from University of Copenhagen. Today I'll be presenting our work, Group Convolutional Neural Networks for Database Segmentation. Today's talk will be outlined as follows. First, I'll talk about the motivation of this work. Then I'll introduce the data we used in this, all the experiments, diffusion-weighted imaging data. And then I'll walk you through the theory behind all the frameworks. And then finally, I'll propose all our models. And then I'll introduce the tasks that we performed using all our models and the experiments we designed for all the testing. And finally, I'll present the results from all the models, and I will then conclude and briefly talk about future work, and then there are references. So the motivation of this work is very simple. To define, as we can know, to define convolution or correlation, the underlying space must have a group structure or be a homogeneous space of a group. And as we can see that in the case that convolution is mostly used in 2D image analysis, what we do is that we slide a kernel function, which is the window that we slide from from the bottom to the from the up to the bottom, left to right, and then it covers the whole space. And no matter which path we take from point A to point B, it's always the same result. But for curved spaces, this may not always be satisfied, as you probably have seen this graph before, that because of the curves of parallel transport, for example, if we want to move the black arrow from the front to the back of the sphere, depending on which path we take, like the blue path or the red path, we end up with different results. And additionally, convolutions can be equivalent to other actions than just translation, like in the 2D image analysis. And so here comes our data that we're using that has this problem. So diffusion-weighted imaging data, they measure the diffusions of molecules in tissues. And we have obtained this type of data from the Human Connectome Project, HCP dataset. And here's a showcase of the size of the scan. And these signals, we interpret these signals as a function mapping from R3 times S2, like a product space, to R. Because in our experiments, we always just use single shell data setup. So so it's not like a multi-channel situation. So in this case, if we interpret the data in this way, each voxel is a spherical function instead of being just intensity values like in normal volumetric data sets. So here's the showcase of a voxel. It's not just a vector intensity values, but more of a spherical function with orientation information. And here we walk into the theory part. So in the classical CNN setups, which is usually used in 2D image analysis, in here the image is usually interpreted as a function mapping from the 2D Euclidean space to Rn. And here, like usually the Rn is R3 because of RGB images. And what we do in convolution is that we convolve this image function with a kernel function. And the kernel is translated using a translation action, like the T2 group, a translation group. And in here, the R2, like the 2D Euclidean group, is diffeomorphic to the translation group itself. So we can actually view them as the same thing with a change of origin. So in that way, we can actually generalize this kind of convolution on a Euclidean space into a more general setup, which means we view the image as not a function mapping from one Euclidean space to another, but we view the image as a function mapping from a group to a vector space. And then we can rewrite the convolution formulation in this way. Instead of like integrating over the R2 space, we integrate the functions over the group. And in here, we can notice that we just replaced the 2D Euclidean group with a group structure. But this is not always the case that we have this nice structure that the underlying space of an image is diffeomorphic to a group structure. What if we have just images mapping from a general manifold to a vector space? So in that case, what is usually done is that we lift the functions from this manifold to a group structure, to a group, and then we can do the group convolutions that were introduced in the previous slide. But in order to do this convolutional lifting, there is a restriction, which is the manifold itself, it must be a homogeneous space of the group it is lifted to. So to summarize, how we do group convolution on like more general manifolds is that we first lift the manifold onto a group, and then we do the group convolutions in that group after the lifting. And then in the end, we project the functions from the group back to the original space. Usually it's done by pulling from the fibers, and then we just collapse the function from the group back to the original space it was from. And the perk about all these operations is that they're all equivariant operations. So after all the dull theoretical introduction, how can we apply all those theories onto DWI signals? So we thought a lot about this, and then in order to do a lot of comparisons, like which space, like the R3 part, the S2 part, which part of this type of data is more important, and are they like interactive within each other? So we designed two types of discretization of the DWI data. Type 1 is we completely ignore the spherical part, the S2 part of the signal, and then see the images instead of as a mapping from R3 times S2 to R. We see it as just, we collapse the S2 part and flatten it into a vector. So we see it as just a general 3D image, volumetric image. And in that case, the group where that would be in the action would be the T3 translation group. And then the type 2 discretization is how it's supposed to be. We see the signals as a function mapping from R3 times S2 to R as introduced before. But then we can also like make two cases out of this type 2 discretization. The first one is we discard the R3 part and see each scan as just individual independent voxels, and they're just a bunch of spherical functions independently. They have no neighboring information, and we're just like trying to model voxels independently. And then in that case, we lift the functions from the S2 to the SO3 group independently. And then finally, finally, if we take into account, the second case, if we take into account the R3 and S2 part together, so there is spatial and orientational interaction in the signal, we view it that way. Here's our final darling here, the SE3 group, and we just lift the signals from the product space to the SE3 group. And in order to increase weight sharing and also save memory and speed up the training, we separate the convolutions from this product space into two parts. One is the S2 part, one is the R3 part. And in the R3 part, the actions are rototranslations. And then in S2 part, of course, it's just rotations that translates the whole signals. And here's a chart showing the actions in using different group actions. And as we can see, like in the SE3 network, the rotations that acts in R3 and in S2, they're the same rotations. So like even though they're separate convolutions, they interact in a way that the rotations that are used to rotate the signals are aligned, completely aligned. So here is a visualization of the T3 network, the classical CNN applied to a 3D image. And I believe all of you have seen like this kind of graph like 1,000 times, so I won't dwell on it. So the second network, the SO3 GCNN, as we talked about, is that we view each scan as just a bunch of independent voxels, and each voxel is a spherical function. And how we model SO3 group is that we use the regular representation of SO3. So we discretize this group structure using icosahedral symmetries. And it's just discretized by 60 rotation matrices. And then finally, here's our darling. The visualization would be like in this video. As we can see that like in the R3 part, there are rotations and translations acting on the kernel. And the rotations in the R3 part are exactly the same rotations aligned with the kernel that the actions that translate the S2 kernels. And now we walk into the tasks and the experiment part. The task would be we segment the brain into 4 regions: the CSF, the subcortical region, white matter, and gray matter. And we, in order to save memory, like we extract the meaningful voxels and also their surrounding neighborhood patches, either 7x7x7 patches for the SE3 network and the classical CNN, or the 1x1x1 patches, which are just independent voxels for the voxel CNN, the SO3 CNN. And the experiments, as we talked about, would be the classical CNN, the SO3 group CNN, and SE3 group CNN. What we did with the 52 scans that we obtained from the HCP dataset is we trained with 1 scan, all networks with 1 scan, validated with 1 scan, and tested with 50 scans. Here's a showcase of the data and how we processed it. On the left-hand side is just a showcase of just a slice in a scan. In the middle is the label image given in the dataset, and on the right-hand side is how we interpreted the labels into a label image that we're going to use in our networks. And here are some results that we obtained from all the networks. For each network, we designed 2 capacities, a smaller capacity and a bigger capacity for each network. So in order to exploit like how network capacity can influence the results. And like it's not surprising that like the SE3 CNNs, they perform actually the best in almost all cases. Even though with a smaller setup, like with only 2,500 parameters, we achieve slightly better result than the classical CNN with almost 1 million parameters. But then we came into this question that, so if we have enough parameters, we do not need the geometry in the data, do we? So we did an equivariance test in addition, which means that we rotated the test set randomly using 90-degree angles, and so that we don't have to do interpolation, and then fed into the trained models as tested before, and then tested with all these rotated unseen data, we can see the performance drop. Like we only tested these with the bigger capacity models we have, and also we can see that the SC3 CNN dropped. Accuracy dropped from 88% to 84%, while the classical CNN dropped from 85% to like 47%. And the SO3 CNN dropped even less than the SC3 CNN because of like there is no interpolation at all in that case. So from this, we can kind of like infer that the equivariant networks, they're very robust against variations in data. So even if the data is unseen, it can capture the incorporated variations. So here are some results from the predictions of like the bigger capacity models from each experiment. And the baseline is the SO3 CNN, and then ours is the SC3 CNN. And so we can see that like especially from the blue area, the SC3 CNN generates a much cleaner prediction comparing to the ground truth, and other methods, they're kind of like noisy for the blue area, and which I believe is the subcortical region, which is always a challenging region to predict. So we have discovered that exploiting the spatial and orientational interactions in the data is very crucial for efficient learning of the features, and incorporating complex group actions of the 3D rigid motions, the SC3 group, might not be essential for like very highly aligned and pre-processed data like the HCP dataset, but it shows significantly higher resistance to the variations in data. And in the future, we can use real-world variations that is recorded by scanners, but that's a challenge too. And we can also apply these methods to other data modalities with specific group actions for the tasks. For example, like the scale equivariant CNNs, like that's another kind of group actions or like a feature we can have. And thank you.
Thank you very much. Are there questions in the audience? Yeah, there's one here.
Thank you for the very interesting talk. I was wondering if there is evidence in using diffusion-weighted imaging to segmenting this kind of brain instead of the classic T1-weighted anatomical brain that's most frequent using for this kind of task.
Yeah, so I get asked that question a lot, actually, and we did use the T1 labels and like reprojected into the resolution that the diffusion-weighted images they have, and what we believe is like our end goal is not to do segmentation, but instead of that, we're trying to model this type of data using a different method. And there are other methods that are trying to model diffusion MRI data, and then they seem to like work well. And then what we are trying to do is like using a different method to model this type of data, and then performing some tasks to see whether it works or not. And the task can actually be other tasks as well. It doesn't have to be a segmentation. We're not really trying to do a segmentation marathon here, actually.
Okay, thank you.
One question here in the front.
Actually, just a follow-up question from the first one. So did you actually compare your accuracy with the T1 datasets as well?
Other datasets?
Yeah, other datasets, especially the T1 datasets, like in terms of extracting, segmenting the regions, how accurate is it?
No, because you mean with other, so we only obtained this data set, and then we try to compare with other methods that are applying like the 60 or like the equivariant networks to DWI data sets, but then they use a different data set as well. And so like it is really a challenge for us to obtain different data sets in that way. And this is the only data set we got like so far.
Do you plan to do some clinical test where you can acquire the same subject's data in different modalities and do a better comparison check?
Yes, so we have talked to a collaborator that did like very similar things, but they do the equivariant networks in a spectral way, like the Fourier type of method, and but then we don't have their data set. But we compared our methods using the HCP data set, but then in the future we do plan to collaborate on in terms of like using their data set to test our model.
Okay, let's save for further questions for the discussion during the coffee break, which will be about now. Let's thank Renfei one more time. So, as you may have noticed, we're a little bit behind on schedule, so we want to reconvene at 11:05 exactly. So there's coffee over there, but there's also coffee machines over there. So distribute a bit, and then be back at 11:05. Thank you.
presentation. So everyone that is a speaker for this one, please make sure to put your slides in the back, please. We are going to use a single computer for this. So it's my great pleasure to introduce our first speaker, Chinmay, talking about structured knowledge graphs for classifying unseen patterns in radiographs. So the floor is yours.
Thank you. Can you hear me? Yeah, perfect. And can you bring up the slides, please? Okay. Before we get started, just a quick word. This is my first time speaking in front of such a huge audience. So if I make a series of mistakes, please, I beg your pardon beforehand. So let's get started. I'm here to present our paper titled Structured Knowledge Graphs for Classifying Unseen Patterns in Radiographs. What do we mean by unseen patterns? So the idea is we start training our model with instances belonging to certain classes that it can see during the training. We refer to these as the seen classes, while when the model is supposed to be executed during inference, it's supposed to work well on already seen classes, as well as instances of the classes that it has not seen during training. This paradigm is generally referred to as generalized zero-shot learning. It's a well-established paradigm, and it becomes particularly interesting in case of medical images, because we often have to deal with rare or novel diseases for which we oftentimes do not have enough samples or even a single sample. Now, the common framework for handling this problem involves having a module for learning the visual features, getting some auxiliary information, and finding a way of aligning these two sources of information together. In our case, we would use chest radiographs or chest X-rays in order to extract the visual features. We would use some auxiliary knowledge, which would basically be about the different diseases we are interested in. And finally, we would try to align the two in order to ensure that things work better during inference. Let's get started. Let's first take a look at the visual features. We ended up choosing chest radiographs in order to extract the visual features because, one, they are very easy to obtain, and it's not very expensive compared to something like MRIs. And perhaps more importantly, they can be used to diagnose multiple diseases such as pneumonia, cardiomegaly, hernia, etc. However, there is a flip side that a single chest radiograph can also indicate presence of multiple diseases. And as such, during inference, we are very likely to encounter a radiograph which can have a combination of seen as well as unseen diseases. Thus, we fall into the realm of multi-level generalized zero-shot learning task, which is a bit more complicated compared to a single-level task. Let's move forward and take a look how are we going to incorporate this auxiliary information. So a well-established way is to use a lot of medical reports which are associated with these obtained radiographs. And we can do things such as obtaining their BioBERT or BioBERT2vec embeddings. These embeddings are usually based on the distributional hypothesis, which states if you have two words which have similar meaning, then in the embedding space, they would have similar embeddings. Essentially, they would be close to each other. And this is a visualization that we created for looking at these different diseases. As one can imagine, brain tumor is closer to the brain, and atelectasis is closer to lung and pneumonia. However, all these relations learned are not explicit. They are implicitly learned as a side effect of the distributional hypothesis. We would be referring something called concepts quite frequently, and in our case, concept basically means diseases, organs, or even chemicals that are used in order to diagnose these diseases. Another source of information which can be used are knowledge graphs. And knowledge graphs differ from these word embedding models in the sense that they explicitly encode the relationship between different concepts. So how does this look like? We would have a tuple of two concepts and the relationship between them. For instance, pneumonia is going to be related to lung structure by the relationship type has finding site. And here is our overall idea. Basically, we propose that word embedding model tend to learn these implicit dependencies, whereas knowledge graphs tend to learn these dependencies explicitly. And these two sources of information are essentially complementary to each other. Thus, if we end up combining these two, we expect, in theory, that we would have a better representation learned. Now, this is the overall picture that we started with. We have looked at the different components. Let's see how we use them in our setup. So this is the schematic diagram of our method. Looks a bit complicated, but we can take a look at the individual components and perhaps it makes more sense. Starting with the image classification network, which is basically a simple classification network based on DenseNet. But this is trained only using samples of the scene classes. Thus, if we use the final layer classifier weights as representation for these different diseases, these representations are meaningful only for the scene labels, whereas it's almost random for the unseen ones. To bridge this gap, we require some way to obtain this auxiliary information. And as we said, we want something based on this knowledge graphs. Now, which knowledge graph do we use is again a big question, but there are lots of options, but we went ahead with UMLS, or Unified Medical Language System, because it's a huge knowledge graph with medical ontologies which are pretty fine-grained, and it has hierarchical as well as associational information. Basically, it's always fine to be on the side where you have enough information rather than a lack of information. The flip side is such graphs tend to be really huge. So in order to use them, we use this idea of parsing, wherein we start with what we call the seed labels. Seed labels are basically both seen and unseen labels, essentially the information that we wanted. And when we ended up parsing it, in green you see the labels of our interest, and in red you see all the extra labels that get parsed, these auxiliary labels. And this is a diagram. I do not want all of you to stress your eyes, it's not very clear. But maybe the major takeaway is you hardly have anything green, and this is already too noisy to work. So we use a simple idea of shortest path-based pruning strategy in order to remove a chunk of these nodes so that it becomes tractable. So on the left, you have the original parsed graph, and on the right, you have a much smaller version where the number of nodes are drastically reduced. This is something that we can work with. This is computationally feasible to work with. And that's what we try to do. So we parse this graph, we go ahead and initialize it with their BioBert embeddings, all these individual nodes, and we process them using a series of graph attention layers. And finally, we want to perform the semantic alignment between the two sources of knowledge. Because only the scene class visual features are semantically rich, we use them in order to align with the corresponding elements. And we finally replace the classifier weights of the original vision backbone with the classifier weights from this graph processing module, so that during inference, we do not have the graph processing module in picture at all. The weights are already copied, the semantic knowledge is already transferred. We are good to go with a simple classifier. And how does all of this look in numbers? So we use the area under receiver operating characteristic. I hope I pronounced it correctly. And we test our method on two different data sets, NIH chest X-ray and Indiana University chest X-ray data set. In the middle, the second paper is the baseline and currently the state of the art that we try to compete against. We are more interested in the performance on the unseen classes as well as the harmonic mean, so that we do not lose a lot on the performance of the seen and improve a bit on the performance of the unseen. As we can see, there is a marginal improvement by using our method. And this improvement is consistent across the two data sets. We also do a series of evaluation to ensure that what we are thinking that we are learning is actually what we are learning. So for this, we first begin with removing the knowledge graph completely and just using the word embedding model. You can see in the first line, there is a huge deterioration in performance, indicating that only the word embeddings obtained by BioBert are not good enough. The second question we want to answer was whether we can use any random graph and it does the same thing. I mean, it would be nice because we spent a lot of time in parsing the UMLS. If we can do it with any graph, why bother? Thankfully, that does not work. So it was not a whole effort in vain. We observed that using any random graph tends to lead to deterioration in performance. And finally, we wanted to see how does the performance improve if we use better embeddings. So we used the BioBert 2-vec embeddings and compared it against the UMLS. We see that as we increase the strength of the embeddings, we also observe a performance improvement. Finally, a good thing about using graph structure, at least for us, is that it also gives some sort of explainability to us, which is very important in medical application. In our case, we use the GNN explainer in order to make sense of the final predictions by the model. I'm sure people in the audience perhaps know GNN explainer better than I do, so there's no point in trying to explain it to you guys. But basically, you remove a particular node and you try to see if there was a performance decrease because of this removal. If so, then the node was important, otherwise it was a redundant node. This is one of the visualizations. We have more in our paper. But essentially, the major takeaway would be that the graph structure is mostly retained in our GNN explainer experiments. And we see that there is an extensive pruning of different nodes which are not very relevant in terms of the overall evaluation or overall prediction requirements. So in conclusion, we show that we can use a knowledge graph and it kind of works. We also propose a method based on our pruning and processing, which also helps us in improving the performance. And the method is also extensible to multiple data sets. But there are a few things that we have not done yet. First is, maybe I was not very explicit about it, but we ended up treating the graph as a homogeneous one and not a heterogeneous graph. But we have another paper in the extended abstract session, and I would use the stage to pitch it up quickly. We tried the heterogeneous thing as well, and we have few results in case you are interested. And second thing is we do an extensive pruning, and we still have to evaluate whether this was a smart thing to do or not, or whether there can be better methods of pruning. Thank you so much for your attention, and if you have any questions.
Thank you for the great talk. So now we have some time for questions. Yes.
Hi, I was wondering, the pruning, did you check the performance based on the known classes or the unknown classes? Because I can imagine that they could be important for some unknown classes that you maybe haven't checked yet.
That's a very good point. So when we prune, the pruning is based on finding the shortest path between the labels of our interest, so both known and unknown. But it's a valid point that if we prune things away, we lose a lot of this explicit relation that was encoded by these extra connections. And perhaps that is where the BioBut embeddings tend to help us, because BioBut embeddings are already rich with this sense of local information. Imagine you are pruning things away, and if most of the prune nodes are the nearby ones, perhaps this BioBut embedding already has this information. So with these explicit node embeddings, we kind of escape the side effects of completely pruning a lot of nodes. But to answer your question, in our case, we stuck with all the nodes of our interest. So both seen and unseen, and we keep track of the shortest path between them.
Thanks.
Yes. Yeah, we have.
Yes, a short question. So you showed the results just based on the features, and then based on your method with the graph structure, and then with the random graphs, you actually saw that it deteriorates. So why is that? Wouldn't the network just ignore the graph if it is?
Yeah, that's a fair point. So we, in our limited understanding, if you have, okay, so graphs tend to work on this idea of homophily, right? If you start connecting random stuff together, intuitively it feels like there should be a side effect of connecting things that should not be connected. So if you connect two concepts which were not technically or which are not adding too much information in this case, then either you learn with something which is not very useful, or you learn something which is completely wrong. And that intuitively for me is the reason why it seems it does not work. It may very well be an effect of the number of parameters. So you have these redundant connections, you're not adding anything, but you still have the same number of parameters and the model is overfitting completely, and it's not generalizing in this case. That would be something I can imagine.
Thanks.
Thank you.
Hi, hello. Many thanks for the interesting talk. My question is about which software you use to build the graph from the UMLs? Like which, did you use just SQL queries, or which kind of software you use to build the graphs?
Yeah, thank you so much for the question. Maybe this gives us an opportunity to pat ourselves on the back because we did not use any extra softwares to parse it. We got hold of the original RRF files, which are basically these relations which are very noisy, and we did the parsing ourselves. So we have a simple Python script which takes around 2 hours, so it's not optimized, but it goes through the stuff and it parses things. And yeah, so it's true, we could have gone through the route of SQL queries, because when we reached out to people at UMLS, they were suggesting us queries. But in our case, we wanted to have more flexibility. So we downloaded these original raw files, and we do the parsing ourselves. We decide what to parse and how to parse them.
Okay, thank you. And there are, I think, also more graph dedicated databases that can be used, like, I don't remember the name, but type.
There is SNOMED, which is very famous, and SNOMED is usually cleaner compared to UMLS. So if I have to do the thing again, I would perhaps go for SNOMED.
Okay, thank you. Thank you very much.
Thank you. So one quick question. So when you talk about explainer, you mentioned, okay, we can remove a graph node,
Yeah.
and we expect that the performance is not decreasing. So are you removing just, what is the strategy? One node or actually a ratio? And actually that will be more insightful to see under a certain ratio, you know that actually the performance should somehow, like a, let's say as a delta perturbation should stay under the sink. So what is the strategy for doing that? It's just like a one node removal, a ratio, a set of nodes, or actually a ratio of edges as well?
Yep, so in our case, we essentially allow these, okay, so suppose this graph. So in our case, what we did, we just made this this edge connection as something trainable, which the network can decide if it needs or not. So we optimize this graph and we ask, try to remove certain edges and tell me the edges which are not useful at all. And if it ends up with some isolated node, so in this case, for instance, it removed both these edges. So we would just remove this node completely. This is still on the graph side. This is not directly associated with the chest data graph. So this is not done in end-to-end fashion. We just wanted to explore the graph part. I hope it answers.
Yes. Yeah. So it's like based on a ratio. If you're removing 2 edges, like that means that you explore a ratio assets. So to make sure that you are not removing something that is semantically useful.
Yes, exactly.
Thank you.
Thank you so much.
So because of the time, we need to move to the next talk. Let's thank again the speaker.
Thank you so much.
Just to double check, for the next speaker, Jad, is not here, right? So Nairus is in here, a comparative study of graph neural networks. Sorry. Other ones that actually they have the slides there, so we can, because yeah. So for the sake of time, the paper XHComp, who is the speaker, you have the slides in the back, right? So you're ready to go. Can we go with you first? Yeah, thank you. Yeah, in the back, please. Yeah, thank you. Please be reminded that all the speakers for this session, please put the slides in the back with the team, with the video team, please. So what? Very nice talk, very nice. So what happened with Jad? He's not here, he's not presenting? Yes, okay, he's the one that sent the email about it. So no one of the co-authors are presenting.
Arus, and then Christian, and then Oscar.
Okay.
So Oscar's presentation I have. I think, I have that. Christian just went to the back, and Arus was
there, and Christian is there. So we have.
Is coming now, yeah.
Okay, perfect. There wouldn't be an event without a short delay. Yeah. Yes. I will be surprised if we are on time, actually. Yeah, that happens. Yeah. The slides are there ready? Okay. Yeah, then please, yes. Who is that? Yeah, we have. Your micro is active? Can you check? Hello? Yeah. Sorry. Can you say
Sorry?
hello? Hello?
Yeah, okay.
Okay.
All right. Hello, I'm Nairuz, and I'll be presenting a comparative study for graph neural networks for shape classification in neuroimaging. So since automated segmentation has matured a lot, and we can now extract accurate shapes from medical images, as shown here in the diagram, and these shapes can be in the form of meshes, GNNs have raised as a promising approach to analyze these extracted shapes. And clinical endpoints may be computer-aided diagnosis or disease detection. So to extract a mesh from a stack of segmented images, one can use algorithms such as marching cubes, and then you have a mesh as shown here that's represented by a set of nodes and edges connecting these nodes, both of which can carry additional information. So for example, nodes features can be anything like simple spatial information or more complex geometric feature descriptors, or even carry information related to the pixel intensities, like going back to the image. Now that we have automated segmentation, GNNs have expanded, and there are a lot of options, and it's hard for practitioners to decide the best architectural setup to analyze these medical images. So these choices are the number of convolutional submodels, which I'll expand on the next slide, the type of convolutional layer to use, and the type of geometric features to encode at the node level. So for one subject and one brain scan, we can extract 15 brain substructures, as shown here. And I listed them at the bottom so that these are the parts of a single brain. And to utilize that, we have two options. Either we input each subgraph into a separate sub-GNN, and in that case, it learns structure-specific embedding. Or we have a single shared GNN that learns from all the subgraphs, and in that case, it generalizes learnings across all structures. So we tried both methods and we found performance to be similar. Therefore, we decided to move forward with having a single shared GNN, because then we have a reduced number of parameters, reducing training time and cost. So that's an overview of our approach. So the input to the shared submodel is the extracted meshes. And the shared submodel includes 3 convolutional layers, ReLU activations, and a global mean pooling layer as a readout layer to aggregate node representations into a single graph embedding. And then the embeddings shown in blue are stacked or concatenated and pass through a fully connected layer for the final classification. So we evaluate this pipeline on 2 tasks. The first is sex classification, which is the proof of concept task. And we take advantage of having data sets from different sources, so we have differences in acquisition, which allowed us to perform extensive testing and to compare our method choices. The biggest data set comes from the UK Biobank, and therefore we used it for model development, whereas CamCan, ICSI, and OASIS were used as test sets. The second task, which is the clinically relevant diagnostic task, is Alzheimer's disease classification. We also use it to confirm our findings from sex classification. So our experiments are designed to evaluate and compare three main aspects. The choice of node features, so we can have like no node features at all by setting a constant value, or positional node features, which are the XYZ coordinates of the mesh nodes, or FPFH, which is a pose invariant feature descriptor. The second point is the effect of the strength of data augmentation on robustness and generalization. To do that, we wiggle the mesh nodes randomly by a maximum offset of 0.1 millimeters, 0.5, and 1, or nothing at all. And the last set of choices is for the convolutional layers. So we compare GCN-conv, GraphConv, and Spline-CNN. And please note that edge attributes are only processed with Spline-CNN. And that's why the option of not having node features at all is only possible with Spline-CNN, because you would still have information in the edge attributes. So here we're showing the ROC curves for sex classification, and we're changing the node features, but the data augmentation is fixed to 0.1, and Spline-CNN in the shared submodel. So you can see FPFH outperforms across all four data sets, and that's because it's pose invariant, and the data sets come from different sources. So positional node features, they rely on positions, so they didn't do as well as FPFH. And then we demonstrate the benefit of data augmentation. So you can see the blue line, again, outperforms, which is of strength 0.1. So it doesn't really help with UK Biobank, because that's the in-distribution test set, but across all out-of-distribution test sets, you can see that data augmentation did help boost the performance. Then we evaluate the three different convolutional layers using the best settings, so FPFH for node features and 0.1 for data augmentation. And we find that GCN-conv and Spline-CNN perform similarly, closely followed by GraphConv. And then, because our training set comes from UK Biobank and a limited age range, so we don't have subjects younger than 44-year-old, we wanted to see if our performance would degrade for younger subjects. But if you look at the blue bars for CamCan and ICSI, you can see that the model actually generalizes well across all age range, because the blue bars are for people less than 40-year-old. And then again, we look at classification performance in healthy subjects and subjects with Alzheimer's disease in blue and orange. Again, there's no much difference in accuracy, meaning the model generalizes well. And then our second task, Alzheimer's disease classification, we look at the effect of the node features. We use Spline-CNN to be consistent with the sex classification experiments just presented, and data augmentation of 0.5, because here we have a smaller training set. And again, we find FPFH to outperform other node features. Then we evaluate the convolutional layers. But here we find GCN-conv to perform slightly better than Spline-CNN, with a substantial decrease in performance for GraphConv. So we presented a proof of concept task, sex classification, a clinically relevant task, Alzheimer's disease classification. We evaluated and compared using different node features, different convolutional layers, different strengths of data augmentation, and we found FPFH to improve classification performance on all out-of-distribution test sets, spline CNN and GCN-conv as both viable options for convolutional layers, and data augmentation being essential in GNNs, especially when we have a small training set, like with Alzheimer's disease classification. We had less than 800 samples for training. In the future, we intend to investigate other applications for the proposed multi-graph architecture. Thank you.
Thank you for the great talk. So we have time for questions. Yeah.
Hi, thank you for the great technical overview. I was wondering whether you corrected for total brain volume, as like sex is associated with total brain volume and Alzheimer's disease with atrophy.
You mean like normalizing to the volume?
Yes, whether you normalize the total brain volume, because otherwise your results might be simply explained by just the total brain volume differences and not the very specific mesh structures.
Yes, that's right. No, we haven't done any normalization. We used the meshes as is, with their true dimensions.
All right, thank you.
Thank you.
Hi, thanks for the interesting talk. I was curious if you also compare to graph-agnostic methods, like if you try to use a 3D CNN, how well would it perform? Because we know probably it will do also very well. So I was curious if there is actually a point in using graph neural networks in these scenarios. Thanks.
Yes, thank you. That's a good point. So yeah, we wanted to have this as a baseline, but we haven't done that yet. And if anything, we were even thinking of using the image information to be incorporated with the graphs, to have a combination of both methods. But yeah, we haven't done that yet either. Thank you.
Hi, first of all, great talk. So I have two questions. First is the choice of using the ROC metric for evaluation. So did you also consider other metrics like FN score or accuracy to compare with other models? So my question is, what is the reason to choose ROC as the main metric for evaluation? And second is, did you also encounter any data imbalance like class imbalance or anything while working on the problem?
Okay, thank you. So yeah, we can use other metrics. We just went for rock curves. The data sets are not imbalanced, so yeah, what we got, all 4 data sets, there is the imbalance problem. But yeah, we could look at other data sets and see how that works. Thank you.
I have a maybe naive question on how you did the combination of different structures. You mentioned that you could either have separate GNNs or you could have weight-shared GNNs, but did you not also consider having just kind of one graph that has all the structures in one?
Yeah, so you can, but it's hard to know where to add the edges between the 15 substructures and what information to put on the edges. Like to design it, it's quite difficult. So we haven't done that. Yeah, thank you.
Thank you. Quick questions. First of all, thank you very much for the great talk. So I'm wondering, because one of the key message towards the end of your talk is about, okay, the augmentation has a good influence. So I'm wondering, because in graph GNNs, one of the key highlights, for instance, in self-supervised is this graph augmentation, where you have different kind, like future level, topological level. So I'm wondering if I understood correctly, you have only one that is not. You are altering only the not for augmenting. So have you tried to see actually what is the real influence of the graph augmentation? That means that removing certain nodes and edges without modifying the semantics or the features itself in the graph.
Yeah, so we picked the most simple data augmentation method, which is wiggling the nodes. But yeah, there are other augmentation methods, like you mentioned, to remove edges or nodes. But we haven't tried that, but yeah, it would be interesting to try that out and see. Yeah, thank you.
Thank you. So let's thank again the speaker. So the next speaker for the XHCOM, Christian. Yes. Okay. Thank you.
And yeah? Okay. Okay. So, does that work? Yes? Okay, hi. Hi everyone, my name is Christian, I'm from the University of Lübeck, and today I'm going to present the work of my colleagues Lasse and Matthias. Matthias, my supervisor, is also sitting in the audience. And it's entitled XHConf, Leveraging Graph Convolutions for Efficient Permutation and Rotation Invariant Dense 3D Medical Image Segmentation. Okay. So, first of all, the motivation, of course. As we heard today, orientation is and orientation independence is a big thing here. And as I put it, MRI volumes can be oriented differently in 48 canonical orientations. Like the voxels could be oriented along the different axes and flipped or rotated. And that's what you have, for example, in a public MMWHS data set. So there the data is differently acquired. So, and the problem what we have then is when you train a network on the segmentation task for this cardiac MRI segmentation, and you input another orientation of data, then the model would not very much succeed in doing a prediction, which is why we need some kind of rotational invariance. So what you might be asking is, couldn't we just use more augmentation, which is true. But for that, we would increase the data variability, and we would need more parameters to capture all the variety, I would suppose. So the problem with this is that you maybe not have the computing power in the clinics. So there might only be CPUs available, not GPUs, what we have in the lab. So that's a problem. And our question was, is there a way to remove the parameters and at the same time improve the quality of the segmentation? So we would opt for a more efficient network kernel design. And this is the question I just raised. Can orientation dependency be removed and computational performance be increased at the same time? This is the question. Okay, so yesterday in the train I realized all the methods, basic methods I'm presenting here, there's an expert of you sitting here in the audience, an author, and that's a bit dangerous for me as an early career researcher to explain your methods to you, but I hope I can do it well, and I try, and if it's wrong, then please correct me afterwards. There's a CNN rotation invariance approach, which is very much related to the talk of Renfei Liu we heard in the last session. And as I got it, they're not doing some kind of an augmentation of the data, but augmentation of the kernel, rotation of the kernel, then having several activation maps, and then later on aggregating them via max aggregation to have a single reduced activation map. So this is, so to say, an augmentation of the kernel, as I would put it. Yeah, and then the question, I'm also I'm talking about voxel data and CNNs, and you're maybe asking yourself why I'm here. So we looked at the concept of edge-confs, and those edge-confs were developed for irregular graph data, but they kept in mind this CNN locality property, what you have in a normal kernel. So the idea of the edge-conf is you have a surrounding neighborhood with k and n neighbors, and you learn a nonlinear edge function E, and you do a symmetric aggregation, like a sum or max aggregation, on the node connected to all of these neighbors, and then you learn a dynamic, or you dynamically update the k and n graph through that. So we took that concept and we thought, okay, why couldn't we take it back, what you took from us, from the CNN world, and get it back to the CNN world again? So if we now look at a classical kernel, and we would probably not use all the filter elements, but we would connect our center voxel to the surrounding voxels and have that concept of edge confs on a regular grid. So that's what we're trying to do, use edge convolutions for gridded voxel data. And this is the basic idea. With that, we realize a message passing between the center voxel and its neighbors. We do a max aggregation of the features, which gives us symmetric invariance and permutation invariance. And we also add instance normalization and relevant nonlinearity. And with that, we remove the 3 by 3 by 3 spatial parameters and only have 2 parameters per channel left. So a significant reduction of the parameter size. So this is our experimental setup. We, first of all, on the top here, we have a very light baseline standard CNN, or a mobile device optimized CNN with very few parameters. And then we have at the upper bound, so to say, our heavy baseline, which has a lot of parameters, classical CNN. And from that on, we are trying to integrate our kernels. First of all, we have an experiment with a normal kernel, of course, to see what's happening. We do normal prediction. Then we integrated a method which we call the Simpermoot, where you have the parameters equidistant from the center voxel with the same shared parameter. And thanks to the reviewers, which gave us a really great review, we also incorporated the 6 neighborhood Simpermoot kernel as an ablation experiment. And of course, we have our edge-conf method. So these are the results. I think I wanted to show only one first, but okay, now we have them all. So for the normal kernel, we can see that we get a very high score, above 80%, which is due to the fact that there are very many parameters in the model. But if we input permuted input here at test time, you see that the score drops by about 39.3% of dice score, which is not that good. For the light baseline model, we have a bit of a less score, which is also explainable, and also here the score drops of about 42% point of dice. With our Simpermoot kernel, the full variant, we get not as good results and a drop of 14.7% of dice. And with the Simpermoot kernel with the 6 neighborhood, the non-permuted input result is also a bit less than with the full neighborhood because we have less parameters. Interestingly, the aggregation and edge-conf-like convolution resulted in quite a good score, not as good as the baseline full model, but nearly, and also above 80% for normal input. And when we input permuted data, the score only drops by about 2.8%, which is really convincing. So these are some qualitative results. Here you have the training orientation. It's the prediction of the full NN unit framework. And there you have the edge-conf prediction for the training orientation. And if you flip that around, you see that the heavy kernel NN unit does not produce so convincing results, whereas our method can keep the labels in place. Okay, so we tested that on the cardiac data set as well, because the abdominal data set from last slide was an artificially generated one. And here is the data which is differently aligned in the training set and for the testing set, so as found in the data set originally. And here you can see that if I train on the AIL-oriented data and predict on the RIA-oriented data, that our method actually performs quite well. And yeah, of course, the light baseline does not perform as well. And the SIM permutation approaches can also keep the scores quite okay. So what about parameter reduction? I said this is our main, or one of our main motivations. So in the model, compared to the heavy baseline, we have 93% fewer parameters, which is quite good. We have 95% fewer flops, and this is also visualized in the graph you see on the right. And for the CPU, we have a speed up of 3.2, which does not hold true for the GPU, because GPUs are highly optimized for those kernel-based operations, so we don't get a speed up in the training. Yeah. Okay, as a recap, this was our question: can orientation dependency be removed and computational performance be increased at the same time? Yeah, if we use edge convolutions, it can. It showed it can. Yeah, I think further research could go in the direction that we more examine the nonlinearity influence and try other neighborhood schemes or gathering schemes for the gridded voxel data. So, and with this, I would like to conclude my talk. Thank you very much for listening, and I'm waiting your questions.
Thank you for the great talk.
Thank you so much. Did I understand correctly that you allow me this moment to bash on your explanation of group convolutions?
Yeah, please bash in.
No, just kidding. It was really well done. Great talk. I think it's really inspiring that you managed to have a very efficient method for maybe what you could consider non-linear convolutions, right? Based on what's happening in the graph learning community with message passing. And I was wondering, like, now you use X-edge-conv. If you have any other ideas, or like as a core computational layer in a network design, that maybe could also to push that idea of non-linear layers a bit further, because convolutions are linear, right? And but now the convolution layer itself with X-edge-conv is non-linear. And I was wondering a bit, like, if you further inspiration from that graph community or plans to work on this end.
So first of all, the plans I just laid out, so like try different gathering layouts. Yeah, we can only cover like full permutations and full rotations of 90 degrees right now with the approach. Good. So a more dense neighborhood would also increase the number of parameters, but could cope with slighter orientation changes. This is the things we thought about. But currently, I'm very open for ideas here. It would be great to hear your intuition on maybe later on what kind of layers we could integrate as well. Yeah.
Thanks. Yeah, we'll have a chance to chat later. But it's really nice to see also a focus on efficiency actually in the talk. So thanks for that. Yeah.
Thank you.
Yes.
Yeah, thank you very much for the very interesting presentation. I was actually wondering, in your first result slide, you showed that when you rotate your images, you still have some performance drop. And I would expect, it looks like your method is entirely, like, fully equivalent in both directions. Do you have an intuition where the performance drop comes from?
Could you put back on the slides? Oh, that's the first, no, last. Okay. So you mean?
I mean the 2.8% that you lose there.
Yeah, the 2.8%. It could be, we discussed it as well. So we're using this NN unit framework. I don't know if you know it, but it's a standard unit. It's, it does some kind of pre-processing for the data, automated, which is really good. It does post-processing, and I suppose it could be that if you turn around the volume, you could have another kind of millimeter spacing, and then the pre-processing would probably change a little bit, and this is why we could get a performance drop. We would need to test with more folds, and we didn't do that in the current work, but yeah, we could extend that to show if it's significant for all the experiments to try. Yeah.
Yep. All right, that makes sense. Thanks.
Okay, thank you.
Thank you. Because of the sake of time, let's move discussions afterwards. Let's move to the next speaker, the last one. Thank you.
Thank you very much.
Now? Yeah, okay, perfect. So, hello, I'm Oscar. I'm from the Technical University of Catalonia in Barcelona, and I'm a PhD student supervised by Verónica Vilaplana. And I will present you our recent work, self-supervised graph representations of hollow slides. So this is the outline we will follow during the presentation. And in the introduction, I will tell you about the field of computational pathology, how graph representation learning is being applied to this area, and the contributions of our work. So digital pathology emerged from the digitalization of pathology slides into multi-resolution high whole slide images, which looks like this one, and are giga-size images. So in order to be processed with a convolutional neural network, for example, they are usually split into patches, which are independently processed by the model. Since it is in general difficult to obtain fine-grained annotations or patch-level annotations, usually a multiple instance learning framework is adopted in which patch embeddings are aggregated to output the final vector representation. And now let's see how graphs are being applied to this field. On the one hand, we have the cell graph approach, which basically consists of taking smaller images, detecting the cells, creating a graph of cells by connecting those that are nearby, and processing these graphs with graph neural networks. But this approach is, the motivation of this approach is that this space of cells is more interpretable for pathologists than the raw pixel space of the images. But this approach is constrained to smaller images, since a whole slide can contain millions of cells. So to work with whole slides, we have this patch graph approach, which basically attempts to extend the multiple instance learning framework by leveraging the position of the patches on the image and contextualizing them with their neighborhood patches. So the problem with this approach is that it relies on a convolutional neural network to encode the patches that ignores this entity space of the cells. So with that all, jointly with the fact that it is generally difficult to obtain these patch level or fine-grained annotations, we propose a framework for the representations of whole slides with cell-level information. Concretely, we create graphs whose nodes encode the cell-level information of any shape regions, and we attempt to apply this approach to downstream tasks, and concretely we focus on region of interest detection. So now let's see an overview of our method. Sorry. These are the steps of the method. We require a preprocessing step, which basically consists of detecting the cells of the whole slides we have. But we're skipping this step here. So the first step basically consists of dividing the cells of the whole slides into different clusters. So we end up having, we cluster the cells based on their position on the image. So we can see every cluster as any shape region of the image. We do this in an overlapping way. So we end up having overlapped any shape regions of the image. And for every one of these regions, we create a graph topology. We obtain a graph topology with the Delaunay triangulation algorithm. So we end up, we split the whole slides into overlapped cell graphs. Okay. So, however, this slide contains millions of cells, so we need some intermediate steps, which basically are we subsample the cells, we cluster the subsampled cells, and we then obtain all cluster assignments by taking the 3 nearest neighbors. So we end up having these overlapped assignments, since some cells may be assigned to more than one cluster. And finally, we obtain the topology with the Delaunay triangulation algorithm independently for every cluster. So the next step consists of basically encoding these cell graphs to a vector representation. Since we have no annotations, we employ a self-supervised learning algorithm, concretely the infograph algorithm, which attempts to maximize the mutual information between local representations, which are the cell-level representations, and global representations. So we employ graph neural networks to do so. This is the architecture of our model. We employ a 5-layer graph isomorphism network followed by an LSTM jumping knowledge layer that outputs the cell-level representations. This is followed by a global pooling implemented as the concatenation of the mean, variance, maximum, and minimum pooling, and a fully connected neural network that projects this concatenation to the final vector representation. And this encoder is jointly trained with a discriminator that attempts to distinguish between positive pairs of local and global representations and negative pairs. So the hypothesis is that the discriminator easily distinguishes between them, means that the mutual information between the representations is high. So at this point, we end up having the whole slides split into different overlapping regions, and we have a vector representation for every one of these regions that depends on the cell information of the region. So the next step consists of representing the whole slides as graphs by leveraging these encodings we have obtained. So we end up having this graph representation of the whole slides, whose nodes are the encoding the nodes are the regions, the nodes features are the embeddings we have obtained, and the adjacency matrix is once again obtained with the Delaunay triangulation algorithm. So this is analogous to the patch graph approach, but rather than employing squared image patches, we are using these any-shape regions, and we leverage this cell graph information rather than working on the pixel space of the images. So the idea is to leverage this graph for downstream tasks. And in this work, we also focus on region of interest detection, which we address as a clustering task because we had a lack of annotations. So we didn't have enough annotations to train in a supervised manner. So we addressed it as a clustering task, also with graph neural networks, because if we were employing, for example, k-means to cluster the embeddings, we would be ignoring this graph topology. And on the other hand, if using a classical graph clustering algorithm, we would fail at incorporating these embeddings. So that's why we opted for the graph neural networks. Concretely, we employed deep modularity networks with this architecture, 1-layer graph convolutional network. And this is followed by a 2-layer neural network and a softmax activation function that outputs the soft cluster assignments for every node of the input graph. And the overall method is trained to optimize spectral relaxation of the modularity problem, which is NP-hard without this relaxation. So at this final step, we have these soft cluster assignments for the nodes of the graphs. So now let's see the evaluation. As it is common within the self-supervised learning community, we have followed the linear evaluation protocol, which basically consists of you train an encoder, you freeze the weights, you get the representations, and you feed a linear classifier to a downstream task. So we have done this at both cell level and region level. This is the data we had. We had 2 sets of whole slides, one of breast cancer tissue, for which some of these slides we had no annotations at all, and for another subset of whole slides we had some region level annotations labeling the slides as labeling the the regions as tumor or normal. These are the number of cell graphs we have obtained from every set of Holy slides. And then we had, on the other set, we had lung Holy slides. For the lung, we had no annotations at all, but instead we had a set of 20 patches with cell-level annotations, labeling the cells as either tumoral or normal. So for the for the cell-level evaluation, we trained an encoder with the lung Holy slides, and then we evaluated on the annotated patches. So these are the patches. We had the cell-level annotations, labeling the cells as either tumoral or normal. Yellow ones are the tumoral, and then we created this cell graph representing the patch this cell graph of the patch, and we forward the graph through the trained encoder and picked the cell-level representations just before the global pooling layer. And these are the representations we employed to fit the linear classifier to a downstream task. So for region-level evaluation, the approach is quite similar. We trained on the unannotated Holy slides, and we evaluated on the annotated ones. So to do so, we had this kind of Holy slides with the region-level annotations here. And we created a ground truth by extracting the cell graphs and labeling them according to the annotations. And we finally forward these cell graphs through the trained encoder to obtain the vector representations, which are the ones employed to fit the linear classifier. So these tables show the results. First one for cell-level evaluation, which is compared with the raw input data. That I missed that, but the nodes features, when we extract the cells, are the basic cell morphological features. So we compare the performance of employing the cell morphological features of a cell to predict if it is a tumor or not, which we obtain these results, and compare with our embeddings that clearly outperform this baseline. And for region-level evaluation, we also compare the method with the aggregation of the cell raw morphological features and the number of cells, which is also relevant in this kind of tissue. And we also outperform the baselines with the embeddings. So I don't have time to go through the clustering results, but as a conclusion, we presented this framework that leverages this cell-level information of the Holy slide. We end up having these representations, these hierarchical representations of the slide that are analogous to the patch graph approach, and we have required no annotations, and we have seen that the representations learned are useful at multiple levels of application. For region detection, which we employed graphs, graph neural networks as well with clustering, we saw that it worked well with many of the images, but in others it collapsed and dropped somehow the performance. But we need more data for running validating these experiments. So that's all. Thank you for listening, and if you have any question.
Thank you very much for the great talk. So now we have time for questions.
I think it's lunchtime.
Okay, I will start then. So first of all, again, thanks for the talk. So I'm curious because one of the frequent keywords in your talk was clustering. So deep clustering, I have two questions actually. One of that is that even if it's the classic perspective from classic Liaud's principles to more recent ones, that is deep learning clustering, they fall under the same disadvantage that you have to deal sometimes with trivial solutions. So because sometimes can happen that you can get some clusters where are empty. So yeah. How do you solve that? Do you encounter that? That is quite often.
Yeah, we follow the method which are deep modularity networks. Yeah. And the main loss is the spectral relaxation of the modularity problem, and then it has a regularizer that attempts to, it's like the a norm of the count of the cluster assignments. So there's a regularizer to avoid this collapse, this trivial solution.
Okay, So it's included there.
Yeah,
so
exactly.
that never happens in your algorithm approach.
It doesn't happen that we saw that all clusters were assigned, some samples were assigned to all the clusters, but we saw that with annotations, we had some of the annotations that almost the entire image was labeled as tumor. Yeah. So that's what dropped the performance of our clustering. Okay. You know, because it was the opposite. I mean.
Yeah. Yeah, so. I see. Yeah, thanks. Yes.
Thank you for your interesting work and your very nice talk. My question was about when you are constructing, for instance, the cell graph. So the first graph you are using then to construct the WSY graph. Have you investigated, in fact, other methods to connect the different cells together? Because it seems to me that the Delaunay triangulation might not maybe be the best approach to modelize the structure of the tissue. And that, for instance, when you are constructing the supergraph, your work was about creating more interesting embeddings to then construct the supergraph on top of it. But have you maybe tried to also explore some methods to see how then you can find the best way to yes, create edges between these supernodes instead of just taking the Delaunay?
Yeah, we did it at the beginning, but then we moved to this approach because basically because we read a really interesting paper published in a journal. I don't remember the name now, but we followed this approach due to their results. But yeah, I think the most common one is to create a KNN graph and then restricting to a radius graph. I mean, deleting or removing the long distances.
Okay, thank you.
It's something to be explored.
Yeah. So I saw that in the slide where you were using the GIN network, GIN, that it was a 5-layered one. Yeah. So did you encounter any oversmoothing, because the more deep you go in the graph neural networks, it is more susceptible to oversmoothing of the data. So did you even try with 2, 3, 4 layers of GIN networks?
No, we didn't. Actually, the oversmoothing is like, I think, more common with node level, but here we were encoding graphs. So that's why we went deeper, because we were encoding the graphs. And we experimented with some other number of layers, but hyperparameter tuning with self-supervised learning is a bit tricky, because on what I base my tuning, I mean, if I base this in the results I get during the linear evaluation, that's not entirely fully self-supervised.
Thank you. So one final question. I'm curious about, because one of your motivations when you open your talk is, I want to reduce computational time cost if you instead create a graph that is completely dense. So one standard way for medical image domain is that you do, or other data sets, over segmentation, like superpixels. And from superpixels, you can create like a more sparser graph. So, and that looks quite similar that you do patch, but actually the superpixels are irregular shapes. So have you also as well compared about how is patches versus your approach and superpixels? So
you mean like defining each region with the superpixels?
Yes, usually what is happening is that a superpixel encodes like a set of cells, for instance, that you have, and then you can do it as a node, and then you can create a more sparser graph there.
Well, that's a good question. Yeah. We wanted to base the experiments on the understanding the whole slide as a point cloud, and so this superpixel approach, like, would require the image. In this work, we were focusing on the cells, so that's why we employed this clustering.
Okay, thank you. Okay, lunchtime. Okay, one question.
Thank you, Oscar. I have one announcement. We decided to start the second keynote, or the after break, at 1:10. So, okay, we gained some minutes there back. This will be a keynote by Michael Bronstein. Looking very much forward to this. Michael unfortunately couldn't be here in person, but he did manage to, we arranged the Zoom meeting, so it will be a virtual meeting, and still, of course, he's open for questions during the session. But therefore, I also find it important that we're here on time, right? Because, I mean, a in-person talk, we can easily shift a bit, but this is scheduled. So please be sure to be back on time. And the final thing is, maybe you have forgotten about it, but we had indeed an excellent program, and we had the Best Paper Award for this. So we actually, by the end of the day, at the closing ceremony, we will announce which contribution we considered most impactful and interesting to our field. So please stick around to the rest of the session, as well as to the closing ceremony and the upcoming panel discussion, which will be a lot of fun, I think. And for now, enjoy lunch, which is over there. Yeah. Hi, Michael. Good to see you online.
Hi. Do you hear?
That's really great that you can manage to be here in your online presence form. So that's excellent.
Thank you.
We're
Sorry,
setting up.
yeah. Let me share the slides, maybe.
Yeah, yeah. So we will start in about yeah, in a couple of minutes. People are still trickling in. So in the program we said 1:15 now in the updated program. So and thank you also for your flexibility in this. Yeah. So we allow them to take a seat, and then I'll make an announcement. Yeah. I hope you're coping well with all the turbulent times at Twitter.
You know how it goes. Somebody wrote, "I cannot complain."
No, yeah. Okay.
In both senses.
Yeah. No, but it's great. A great opening slide, by the way. So far it's been actually a big success the workshop. Like we have a packed room. We have some online presence. Things are running smoothly. So and we had a keynote this morning by Emma Robinson, which was really amazing. I think on cortical surface processing. And I think your talk will be a great addition to this to really have all aspects of geometric deep learning and medical image analysis collected. So
thank you. Thank you. Eric, so it's an hour, right? In total, including questions, right?
Yeah, you have an hour. This is Jelmer Woltering, the other organizer.
Hi, nice to meet you. Thank
No,
you.
you have an hour. Yeah, let's try to aim for that. I mean, we've been quite flexible throughout the days. If it's a bit shorter or longer, and it would be nice indeed if we have some chance still to have discussion and questions.
Yeah, of course. Yeah.
I'm quickly checking with tech if you can get a feed also from our room. Michael, this is Angelica.
Hi, Michael.
Hi.
Okay.
And Gemma, yes.
Yeah, and Jelmer, you just, yeah. Okay. I'll announce you in a minute. I'll leave the call, Michael, and then I'll address you via the microphone here in the hall.
Sounds good. Yep.
All right. Welcome back. So we continue our program with the next keynote by Michael Bronstein. Okay, yeah, so the slide is already up. Would it be possible to see a video of Michael as well? He is here online, so it's not just a slide. So it's my pleasure to introduce Michael Bronstein, who is, I think, the face and one of the main driving factors behind this whole community of geometric deep learning. I think this term actually came from one of his proposals, one of his many ERC grants awarded to Michael. So Michael is currently professor at Oxford and has been consistently delivering high quality work and innovative work when it comes to leveraging geometry in image processing in general, but especially in deep learning more generally lately. And it's my absolute pleasure to have you here, Michael. So looking forward to your talk. Yeah, please go ahead. The screen is yours.
Thank you very much. Thank you, Eric, and for this flattering introduction and for the invitation. And I'm sorry that I couldn't attend in person, but I'm very happy that modern technology allows me to be present without being present. So I would like to give in the next hour an overview of what geometric deep learning is, right, and more from the pedagogical perspective, and then show some applications on which we've been working at Oxford and also with my collaborators, especially in Switzerland, in Lausanne, on protein modeling and potential applications to drug design. So I like to open with this quote. So it comes from Erwin Weil. He was one of the most important mathematicians in physics in the 20th century. And he wrote thus poetically about symmetry that he described as wide or as narrow as you may define its meaning. One idea by which humans through the ages have tried to comprehend and create order, beauty, and perfection. And indeed, symmetry plays a very important role in mathematics, in physics, in other sciences, in art, in music, whatever, more or less, whatever area you touch, there is something that can be described as symmetry. And the term itself is geometric, so it comes actually from ancient Greece, where, as you know, the foundations of modern geometry were laid in the seminal work of Euclid, the Elements, which was the first axiomatic construction of geometry that even today we study at school as the geometry, and we know as Euclidean geometry. And it's been around for more than 2,000 years, somehow unchallenged, the only geometry that existed, even though many illustrious mathematicians for hundreds of years tried to do something with the so-called fifth postulate that talked about the parallel lines, and unsuccessfully, and until the 19th century, where this monopoly of Euclid and his geometry was broken with the first introduction of first successful constructions of non-Euclidean geometries that maybe initially were derided and rejected. They rejected, but towards the end of the century, it became clear that there exists other geometries that do not necessarily satisfy the assumptions of Euclidean geometry. And it created a situation where there was a zoo of different types of geometries, but no clear order or clear hierarchy, how they are related and what actually defines a geometry. So here comes Felix Klein. He was only 23 years old when he was appointed as a professor at the University of Erlangen in Germany. And it was customary then, I think still customary in some German universities, to deliver an inaugural address where he would describe his future research at this university. That actually he didn't stay there for more than 3 years, and he ended up in Göttingen eventually, which was the center of mathematics in Germany at that time. And this entered the history of mathematics as the Erlangen program. So I think the University of Erlangen obviously is very proud of this association. And it was a radical change in perspective on how you would approach geometry. You can say that comparable to Descartes and his method of coordinates, which was the first algebraization of geometry. That was the second one, modeling geometry using group theory, basically as a space with some symmetry group attached to it. And you can define geometry by looking at the properties of geometric objects that remain unchanged or invariant under the action of this group. And this idea was really of profound impact in geometry. So it took quite some time to incorporate Riemannian geometry. That was done mainly by the School of Cartan about 50 years later. It also had a bigger cultural impact in mathematics. So some follow-up works such as category theory were abstractions and generalizations of these methods. So a lot of new fields actually, you could say, that stem from this work of Felix Klein. But probably the biggest impact that spilled outside of mathematics was in physics. And in physics came the realization, so first by a colleague of Klein, Amy Noether, that also worked in Göttingen, that you can derive from first principles conservation laws in physics. You can derive them from considerations of symmetry, and came the realization, also thanks to Weil, whom I mentioned in the beginning, that you can build all physics from these first principles. So in physics, as some of you probably know better than me, you can describe what is called external symmetries that model the structure of the space-time, right? This is what is called the Poincare group. That is associated with special relativity and gives rise to a special geometry that is called Minkowski geometry, and also internal symmetries of the fields that describe different interactions. And this is what we nowadays call the standard model. So maybe with the exception of gravity, this is all physics that we know. And I can quote another important physicist, Nobel laureate Philip Anderson, that said maybe very laconically that it's only slightly overstating to say that physics is the study of symmetry. Now, of course, you can ask what's the relation to deep learning. And in deep learning, we also have a kind of situation that happened in geometry. In the past, we have a lot of different neural network architectures that historically were derived for different types of data or for different use cases, such as convolutional neural networks, for example, that came from motivations in neuroscience and were derived for image analysis, or, for example, recurrent neural networks that were developed to deal with acoustic signals or sequences like text, or maybe graph neural networks that originated in chemistry, where you describe a molecular structure as a system of relations or interactions between atoms and their chemical bonds. So what is the common denominator of these different architectures, and can we derive them from the first principles, same way as you can derive geometry from some common framework or some common blueprint? And this is maybe a little bit arrogantly what we call the elegant program of machine learning or geometric deep learning. So this set of foundational first principles that allow us to derive not as a kind of hack or some historical legacy, but from first principles of symmetry and invariance, some of the most popular architectures that are nowadays used in deep learning. Now, if you look at the problem of machine learning, right, maybe in a very simplified and slightly naive setting, so think of supervised learning. Let's say you have a set of images of cats and dogs and you want to classify them, right? So you can think of it as a kind of glorified function approximation or curve fitting problem. So, and some cynicists say that this is what deep learning is, right? So it's some glorified function approximation. The problem is that we can, of course, approach this problem from the perspective of approximation theory, and we can show that even very simple neural network architectures, such as the perceptron that dates back to the 50s, the seminal work of Frank Rosenblatt, if you just use 2 layers of such neural network, you get universal approximation properties. So these are results that were shown in the late 80s, in the beginning of the 90s, and go back to the Arnold-Kolmogorov theorem, and even earlier to the Hilbert problem. Basically, you can approximate any continuous function as this simple neural network to any desired accuracy. But these results usually don't tell you how to do it and what would be the efficiency of this approximation, or in other words, how many samples do you need to take of these functions. And in fact, what you can see is that very well-behaved functions like Lipschitz continuous functions require a huge number of samples in order to approximate them with desired accuracy. And basically the bounds are very pessimistic, so it scales exponentially with dimension. And because most machine learning problems deal with very high dimensional data, such as images might be thousands or even million dimensional, sometimes you will just need more samples than the number of particles in the universe. And this is especially evident in computer vision or image analysis applications, where if you take an image, right, and you treat it as a vector, as a point in some high dimensional space, and you pass it into such simple neural network architecture as a vector, then you can see that if I shift the image by just 1 pixel, I will get a very different input. And as a result, the network will be, you need to show a lot of examples to train the network to recognize these shifted versions of the digit 3 as the same class. And this is, of course, this is what we do often with, this is called data augmentation in deep learning. But this is obviously not ideal because we know somehow that it's still the same object, it just underwent some geometric transformation. And indeed, this kind of intuition was captured in a new type of architectures that followed on the work of Rosenblatt, first the neocognitron of Fukushima, and then, of course, the classical convolutional neural networks by Jan LeCun and co-authors. And the idea there was to break this problem by sharing weights and having locally connected neural network architectures that could be expressed as convolutional filters and also efficiently implemented on first on DSPs and then on GPUs. And that was really one of the driving forces of the deep learning revolution, the availability of hardware and its compatibility with these algorithms. Now, in other applications, we can encounter different types of data, so that do not necessarily have this grid structure. And one example is a molecule. So this is a molecule of caffeine, and I know that people laugh at me when I show a cup of coffee with caffeine with my daily dose of this compound. But basically, if we want to deal with problems such as drug discovery or virtual screening of molecular compounds, we need to be able to predict some chemical or physical or biological properties of these graphs in some way, right? That will have relevance and allow us to distinguish these compounds from, let's say, something that would be potentially successful as a drug. And here again, we can pass the node features representing the atoms of these chemical compounds to a simple perceptron network. The problem is now we don't have any canonical ordering like we have in images, so we can reorder the nodes of this graph in any arbitrary way. And molecules are just one example of maybe small graphs. We have gigantic graphs such as social networks or different interaction networks or interactomes in biology, or meshes in computer graphics, or functional networks in biomedical applications such as brain imaging. And recently there has been, in the past decade, really an explosion of graph structured or network structured data with especially proliferation of internet technologies and social networks and so on. So the idea of geometric deep learning is to, if we go back to this problem of the curse of dimensionality that we've seen, comes with the realization that our signals are actually not just high dimensional vectors. So they have some underlying geometric structure. So in particular, the images that we've seen, they live on some two-dimensional domain, a grid. And the structure of this grid can be described through a symmetry group that I denote by G here. And then the action of the group on points on the domain is manifested through some group representation. So that is a shift operator that acts on the image. And I can incorporate this property into the function that acts on these images, right? Like my image classifier, basically in the form of some inductive bias. And the three main components of geometric deep learning are the domain and its symmetry group that can be actually selected separately from each other, dependent on the problem, the family of signals that live on this domain, and how the group acts on these signals. So again, depending on the type of data, it might act differently. And then the functions that incorporate some form of invariance or equivariance. And the example of cats, right? This was really one of the main motivations behind neocognitrons and convolutional neural networks, that if I want to classify an object in an image, I don't care where exactly it is located, right? So I don't care where the cat is located in the image if I want to say that this is a cat. So this is an example of invariance with respect to translation or shift invariance. Or if I want to segment out the cat from the background, in this case, I get what is called translational equivariance, in the sense that the output is transformed in exactly the same way as the input. And geometric deep learning blueprint really is some combination of invariant and equivariant layers, maybe interleaved with some pooling operations that implement another important physical principle that is called scale separation. But bottom line is that you can recognize this kind of structure in most of the neural networks that are now used, whether it's graph neural networks or convolutional neural networks, or even some forms of recurrent neural networks implement this type of structure. So they can really be derived from the same blueprint by the appropriate selection of the domain of the symmetry group, and then baking it into the architecture. And I will mainly talk about graph neural networks today, and some slightly more exotic versions that can be applied to meshes or discrete manifolds. So talking about graphs, graphs are really ubiquitous. They are a very convenient mathematical abstraction of systems of relations and interactions. You can model more or less anything, starting from molecules to entire communities or social networks, right? So from nano to macro scale. And in the very basic form, you can think of a graph as a collection of nodes that are connected by edges, so ordered or unordered pairs of nodes. And let's assume for simplicity that we associate some d-dimensional vectors with every node. So these are node attributes or features. And we can represent this data in the form of two matrices. So the node features, a matrix of size n by d, n is the number of nodes, d is the dimensionality, and the adjacency matrix that captures the structure of the graph is a matrix of size n by n. Now, in doing this, I'm cheating because I don't really have a canonical ordering of the nodes of the graph, so I assume some ordering. And if I change this ordering, I can basically I need to change the way that the data is represented, right? So I need to apply permutation to the rows of or the rows and the columns of these matrices. So this is a representation of the permutation group that is a symmetry that is associated with the graph. Now, the types of tasks that we can associate and we can talk about in the problem of graph machine learning is predicting graph-wise properties, like I give you a molecule and you need to predict some physical property like water solubility, or you want to solve node-wise problems, for example, detecting spammers in a social network. And there is another third type of problems that I don't want to discuss today, which is link prediction. You try to predict whether there is an edge between two nodes in the graph. So in the first case, we don't care how the inputs are ordered, right? So how the nodes in the input of my function are ordered. I want the output to be the same. So we're talking about permutation invariant functions. In the second type of problems where we want a node-wise output, I want the output to be permuted in the same way as the input. So we're talking about permutation equivariance, right? So here, up to the choice of the group, we get exactly an instance of the blueprint that we've seen before. And graph neural networks are really just a concatenation of several permutation equivariant layers or equivariant functions that concatenated together give you overall permutation equivariant representation. And if the task is graph-wise, then you can also do some pooling that is permutation invariant. And you can see it as an instance of geometric deep learning blueprint, again, by the right choice of the group, which is permutation, the right choice of representation, right, which is permutation metrics, and then the functions that are that respect this symmetry can be implemented through message passing. So what is message passing is some form of neighborhood aggregation. So for every node in my graph, I'm looking at my neighbors. And basically what a node sees is a soup of nearby nodes, right? And maybe edges if we also have some information on the edges. So I remind you, I don't have a canonical ordering of the neighbor nodes, right? Or nodes in the graph in general. So the way that I aggregate them, and I denote this aggregation by a function phi, must be insensitive or invariant to the order of the neighbors. In other words, it must be a permutation invariant function. And I can apply this function at every node of the graph, right? And this way I get an output for every node of the graph, which is easy to see is permutation equivariant. And the choice of this function really defines the distinction between different types of architectures of graph neural networks that we encounter in the literature. And there is a broad zoo of these, but fortunately we can classify most of them in the following three types or flavors, as we call them in our book. So the first flavor is what we call convolutional graph neural networks. And in this case, I have an aggregation that depends only on the structure of the graph. So in the simplest case, it can be just the adjacency matrix, right? Coefficients of the adjacency matrix. And I collect my neighbors and I weight them by the adjacency. So, and here, this is some node-wise transformation. Can be, in the simplest case, just linear transformation. And it is typical also to use a nonlinear activation function. So in a more general flavor, we might have aggregation coefficients that depend not only on the structure of the graph, but also on the features themselves. And in this case, this is implemented as attention. So the typical representative of these architectures are GATS or graph attention networks by Petr Veličković. And in the most general case, we have a nonlinear biparametric function that can be interpreted as a message that is sent from node J to node I. So these are the message passing graph neural networks. And it's interesting that message passing graph neural networks, if we choose the aggregation operation to be injective, are formally equivalent to a classical algorithm in graph theory that is called the Weiss-Lemon graph isomorphism test. And it's a method that was devised in the 60s, and the motivation actually came from chemistry. And the method is named after Andrei Lemon and Boris Weisfer, two Soviet mathematicians that were challenged by a chemist called George Vladuts to devise a method that would allow him to search molecules in large data sets. And molecular search that he wanted to do, it was structure-based. So in molecular compounds, typically similar structures correspond to similar chemical properties. So he wanted to be able to determine whether two molecules are structurally similar. And at that time, not much was existed in computational graph theory, so they actually believed this method to be a polynomial solution to the graph isomorphism problem. It happened to be not the case, as was shown by counterexample, but it created an entire family of algorithms. It's not a single algorithm, so it is often referred to as the Weisfer-Lemon hierarchy. And in the simplest case, the test goes like this. So I give you a graph, and assume that all the nodes are colored in the same way, right? So they have the same label, a discrete label. So what the Weisfer-Lemon test does, it's an iterative color refinement procedure. So it examines the neighborhood of every node, right? And in this case, for example, I'm looking at a node with 3 neighbors, and it applies an injective function. You can think of it as some form of hashing. So these, for example, this neighborhood structure will become a new label. And this neighborhood structure has a different one, right? So we have here a blue node with 2 neighbors. So this will become another color, and as a result, I will have a graph with 2 different colors, right? We have, let's call them green and yellow. I can repeat the same procedure again. So in this case, I have 3 different types of neighbors. I have a green node with a green and a yellow neighbor. I have a green node with 2 yellow neighbors, and I have a yellow neighbor node with 2 green and 1 yellow neighbor. And these become 3 different distinct colors. But if now I repeat the procedure, the colors will not change, at which point I will stop and will produce a histogram or a distribution of these colors. And this will be some kind of graph descriptor. So if I give you another graph and I see that the distribution of colors is different, then I can tell for sure that the graphs are not isomorphic. So there is no edge-preserving bijection between their nodes. But if the colors are the same, then I don't know. So it's a necessary but insufficient condition. And in fact, we can find simple examples of non-isomorphic graphs that the Weisfer-Lemon algorithm cannot distinguish, like the example shown here. And this is really a problem in chemistry. So I remind you that this algorithm originated, or at least was motivated by structural similarity of molecules, but even as simple molecular compounds as I show here cannot be distinguished by the Weisfer-Lemon test. So fortunately, as also I mentioned, it's not a single algorithm. There is an entire hierarchy of them. So what Weisfer-Lemon described in their paper actually is what is now nowadays called the 2WL test, that looks at not at refinement of nodes, but at pairs of nodes, and they can do higher dimensional tests. So these can be also confused by special families of graphs that manifest strong regularity. So a family of graphs that is called Kiefer and Immermann graphs. And as we said, the message passing graph neural networks are equivalent to the 1WL, to the iterative color refinement procedure that I described. There are also constructions that are equivalent to KWL, mainly devised by Hagai Maron, KGNNs, and some more exotic constructions like our work, also with Hagai, like subgraph union networks that sit somewhere in between. So they're upper bounded in their expressive power by 3WL, but they look like standard message passing. Now, another trick that is also commonly used in the literature is what is called positional or structural encoding. So if we know that, for example, WL cannot detect certain structures, we can just pre-compute them by counting, for example, triangles or clicks in the graph, and then attaching this information as additional features to the nodes or to the edges of the graph. And you can also think of it as a kind of problem-specific inductive bias. So if I know, for example, that in organic chemistry applications, very prominent and important structures are cycles or rings, like in the molecule of caffeine, we have a cycle of length 6 and 5. I can count cycles of this size, and you can see that by using this information, we get some problem-specific inductive bias that improves our ability to predict chemical properties. In this case, it's water solubility. You see that the error is decreased dramatically when we count cycles of size 5 and more. Now, there are some more exotic architectures that are used in GraphML literature. I will not go into much details of this, but roughly speaking, if we think of classical GNNs as the following concept, that I give you a graph with some information on the graph, and the graph is used in a dual way. So it's part of the data, but it's also a computational device. So I use the same input graph to propagate information on the graph. And then by propagating, I can also discover some structures within the limitations that are dictated by the Weiss-Frey-Lehmann test. So on the other hand, we can say that we completely ignore the graph and we consider just the set of nodes and the corresponding features. And in this case, there is no interaction whatsoever between the nodes. So here we are talking about type of architectures that are called deep sets or point nets that are actually very common nowadays in computer graphics, especially for the analysis of three-dimensional point clouds. The other extreme of this, we can assume that we also ignore the input graph and we assume that any node can interact with any other node. So the graph is complete or fully connected. And then we can learn it for the given task in the form of attention, right? So we learn some soft adjacency metrics. So this is how transformers operate. And these are ubiquitous nowadays in natural language processing. So somewhere in between lie methods for graph rewiring. So where you can say that your graph maybe is not necessarily the best to propagate information. So I can change it, for example, to facilitate message passing. And then I use this graph to propagate information instead of the input one. Or in transformers, especially, one sometimes wants to implement functions that are not necessarily permutation invariant in NLP problems or in text analysis. It is important to be dependent on the position. So you can attach positional encoding to the nodes, or the same methods can be applied to graphs as well. For example, the structural encoding that I mentioned. And then you can do also more interesting things. So you can take a graph and lift it into higher dimensional topological space, such as simplicial or cellular complex, and do more interesting message passing. You can, instead of considering the graph, consider it as a collection of subgraphs that are extracted by some policy, and then consider a more interesting symmetry group that is a product of permutations of the nodes of the graph and the graphs in this multiset and this collection. You can also account for the symmetry of the data itself, and this is the type of equivariant neural networks that actually Eric has been working on, and one of the pioneers, and these are used in chemical applications and famously used in AlphaFold 2 by DeepMind for predicting protein folding. And finally, if you have a graph that has special structure like grid, then you get classical convolutional neural networks. So let me just briefly mention topological message passing. So graphs are examples of topological spaces that are called cellular complexes. So nodes are zero-dimensional complexes, edges are one-dimensional complexes, but you can also go to higher dimensions, and you can invent a way, or what we call a lifting operation. So we can say that, for example, all cycles of certain size will correspond to cells, and now instead of passing information only between adjacent nodes, you can also pass information up and down in this hierarchy. So you can pass information between nodes belonging to the same cell through this cell. And we can show that this is strictly more expressive than the standard Weiss-Frey-Lehmann algorithm. So I mentioned already the equivariant graph neural networks. So here, by appealing to analogy in physics, again, we can make the distinction between internal and external symmetries. So the external symmetry will be the symmetry of the domain, which is the permutation of the nodes, but the internal symmetry will be symmetry of the data. And in chemistry, we are specifically talking about the graphs that have also geometric realization. So these are molecules that live in three-dimensional space. So for example, the coordinates of the atoms, you can rotate your molecule and you don't care about how the molecule is rotated or translated in space. So you want to be equivariant with respect to geometric transformations of these coordinates, like rotations. And this can be captured in the form of equivariant message passing. And as I said, this has been used very successfully in chemical applications such as drug discovery and in structural biology such as protein folding. Now, even more exotic models, you can derive graph neural networks as discretization of differential equations. Basically, there is some underlying physical motivation that allows you, for example, to think of graph neural networks as some dynamics of particles that live in some space using, for example, diffusion equations or wave type equations. You can also incorporate positional encoding and graph rewiring. So this is an example of how features are diffused on the graph, where the graph itself is changing throughout the evolution. And you can also describe the expressive power of graph neural networks by looking at the limit of such diffusion equations. So this is also a diffusion equation on exotic topological object that is called the cellular sheaf. By looking at the limit of this diffusion, we can, for example, say how many classes of nodes we can distinguish under some restriction of this sheaf structure. So, this is an alternative, in my opinion, that is more interesting and more flexible than the standard Weiss-Farrell-Lehmann hierarchy. So in the remaining time, let me talk about some interesting special cases of graphs which are grids. And as you know, a grid is just a graph, and if you assume a grid with periodic boundary conditions, so this is what is called a ring graph. So every node here is connected to a pair of nodes, right? So it's one-dimensional grid. And what is striking here and different from usual generic graphs is that we have fixed neighborhood structure and we have fixed neighborhood order, right? I can always identify a neighbor that is, let's say, to my left and to my right. And therefore, the most general aggregation functions that had to be permutation invariant in the past now can be more specific. So I can have aggregation functions of this form. And if this aggregation function is done in a linear way, then we get the familiar convolution that can be described as a circulant matrix, right? So I can look at convolution as multiplication of my feature matrix X by a matrix that has the same elements on the diagonals. So this is called the circulant matrix, right? And this is a form of implementing convolution with periodic boundary conditions, what is called circular convolution. Now, the special property of convolution or circulant matrices is that, unlike general matrix multiplication, it is commutative. So the product of two circulant matrices is commutative. And this is just a special case of this is commutativity with a special circulant matrix that shifts the elements of a vector by one position. So this is what we call the shift operator. And this is just another way of saying that convolution commutes with shift, right? So I can first apply convolution, then shift, or vice versa, the result will be the same. And this is essentially a manifestation of our symmetry principle, right? The translation equivariance. It appears actually that this works in the opposite direction as well. So if I have a matrix that commutes with shift, it necessarily must be circulant. So it necessarily must be convolution. And as you see here, convolution emerges automatically from translational symmetry. Now, I don't know how about you, but when I was taught convolution in signal processing first, it came like a formula out of the blue, well, saying that you kind of match local filter, right? And all this kind of motivation, but nobody really explained the geometric motivation for it, that actually it can be derived from first principles. And here, I hope you can see that it's derived from first principles. Now, one thing that if you studied signal processing or harmonic analysis that also comes together with convolution is the Fourier transform, right? And the Fourier transform is also something that comes out of the blue. So it's some kind of orthogonal basis that has the complex exponentials or the sines and cosines. So here is how to derive the Fourier transform again from first principles. And again, we can look at circulant matrices because they commute. We know from linear algebra that commuting matrices are jointly diagonalizable. So they have the same eigenspace, or in this case, you can say that there is the same eigenbasis that diagonalizes them. So we can pick up one matrix, for example, we can pick up the shift operator, right? And say that all the circulant matrices are diagonalized by the eigenvectors of the shift operator. So now it boils down to taking this matrix, which is very simple. It just has one shifted diagonal of ones, and by hand computing its eigenvectors. And surprise, surprise, these actually appear to be these complex exponentials, which is exactly our familiar Fourier basis. So another missing piece is that the eigenvalues that will be different for all convolutions happen to be the Fourier transform of the coefficients that form the circumvent matrix, and we get this duality between performing convolution in the spatial domain or performing it as element-wise product of the Fourier transform, which is the bread and butter of modern signal processing, because the computation of the Fourier transform can be done efficiently by exploiting the redundancy in the Fourier basis, which is the basis of fast Fourier transforms in n log n complexity. Now, we've talked about graphs, we've talked about grids. Let's talk about continuous geometric objects, which are manifolds. So for people working in machine learning, maybe one would wonder why at all to consider manifolds. So these are somehow exotic objects. So I will give you two motivations. So one is representations of geometric objects, of three-dimensional objects, like the Stanford bunny here. So of course I can represent it as rasterized volume, but if I'm interested only in what happens on the surface of this object, there is huge waste of information, right? So I need to represent also what's inside the bunny, which I don't even see, right, in many applications, and I don't care. Now, second thing is that we can use tools from differential geometry to naturally model the deformations of these objects. For example, when we deal with articulated or deformable shapes like the human body, and in problems in biology, to which I hope to get in a few minutes, we can actually abstract this way a lot of information that is unnecessary and maybe too complex to model. And as an example, when proteins interact, what the molecule when a molecule sees a protein, it doesn't care about the intricacies of the fold of the protein that you see here in different colors. The only thing that matters is how the overall molecular surface that appears from the outside. So this is what really guides the interaction of the molecules and determines whether two proteins will bind or not. So it allows to abstract out some internal structure and simplify otherwise very complex problems to something that is geometrically easily tractable. So the two types of invariants that we encounter when dealing with manifolds are, first of all, we need to deal with manifolds as local spaces because they don't have typically global coordinate systems. So it will be local choice of system of coordinate, or what is called a gauge in physics. And another type of invariance is global deformation. So I can deform the manifold, and as a result, some of its properties might change. So just to give you a crash course on differential geometry, which is not really the purpose of this talk, but just to give you a very intuitive concepts, you can think of manifolds as locally Euclidean spaces. So it means that around every point, I can map a small neighborhood of a point into Euclidean space. That is called the tangent space or the tangent plane in case of two-dimensional manifolds that I'm showing here. The Riemannian metric is just a way to locally measure length and direction. So it's an inner product defined on this tangent space. And then every property that I can express in terms of this metric entirely is called intrinsic. And isometries are metric preserving deformations. So if I change this manifold without affecting the Riemannian metric, then basically it will be imperceivable from these internal structures. Now I can try to implement something that looks like convolutional neural networks on manifolds by just taking a function that lives on the manifold, mapping it to the tangent space. This is done by an exponential map. So it's an intrinsic map that allows to express the signal locally in the tangent space around every point, and then just multiplying it by filter, same way as I do with a sliding window in an image. The problem, of course, here is that written in this way, I cannot really compute this expression because here we are, what we write are abstract geometric vectors, right? And when you study vectors and vector spaces at school, the crime against humanity that is committed is that vectors are often described as arrows or collections of coordinates. So vectors are not arrows or not collections of coordinates. These are objects that you can add and scale. So in order to think of them as arrows, you need an extra structure that is called an inner product that allows you to define directions. And in order to think of them as arrays of coordinates, you need a system of coordinates, right? But this is really what you can deal with on the computer. So you need to define a local system of coordinates, so a local reference frame. This is what we call a gauge that is defined at every point of the manifold and is smooth. And now with respect to this gauge, you can define a filter, but of course you want to make sure that no matter how you change the gauge, because this choice is arbitrary, the result will be the same, right? Or more correctly, it will be transformed in the same way with the transformation of the gauge locally. And on Riemannian manifolds, this ambiguity is rotation. So I can rotate my local system of coordinates, preserving the orientation. And the work of Taco Cohen and co-authors showed that you can design filters that are equivariant with respect to this change of local coordinates. Now, here maybe comes the disparity between the theory and the practice. So in theory, you cannot really select a gauge that is unique on sphere-like objects. So this is a result in differential geometry that is known as the Poincaré-Kopf theorem or Hairy-Ball theorem. And it says that if you have a hairy sphere, you cannot comb it without having vortices. So there will be some points where this tangent vector field vanishes. But in practice, you can construct these local reference frames that maybe with the exception of a few singularities are actually quite stable. So this is what we did in some first works. Maybe it was not mathematically beautiful, but it worked actually quite nicely. Bottom line, what all these exotic constructions allow you to do is something that looks like convolutional neural network, but defined intrinsically on the manifold. As a result, if you bend your object, the filter will be deformed in the same way. So you bake a lot of invariants that you want to deal with into the architecture itself. And some of the applications that I would like to talk about, they actually come from the problem of structural biology and in particular dealing with proteins. And as you probably know, proteins are really the most important molecules that we know. I think maybe slightly poetically, but completely correctly, you can call proteins the molecules of life, because we don't know any life form that is not based on proteins, at least currently. And proteins are encoded in our DNA. They form everything from viruses to whales. And proteins are in our body are responsible for a lot of functions, from defense against pathogens. Antibodies are big proteins, delivering oxygen to our cells, transporting stuff through cellular membranes, right? Mechanisms such as the calcium pump, different communications. So many hormones are protein molecules. The enzymes are also proteins that catalyze chemical reactions, and otherwise would not happen, and so on and so forth. And proteins are biopolymers. So in other words, they are molecules consisting of small building blocks called amino acids that chain together under the influence of electrostatic forces, fold into complicated three-dimensional structures. So this is a problem that is called protein folding. And if we want to design a protein that has certain structure and as a result endows the protein with a certain function, we want to solve a kind of inverse problem, right? We want to come up with a sequence that folds in a certain way. And it's very tempting to think of these kind of three levels of complexity. So we have a one-dimensional sequence of amino acids, and first bioinformatics algorithms were invented to treat them as strings using simple computer science methods. Then the sequence produces certain structure, right, by means of folding. And this also that was postulated to be a problem that is fully determined from the 70s by Anfinsen, and as recently shown by DeepMind and other works. Indeed, we can predict structure very accurately from sequences in some situations. And finally, the structure endows the protein with function, for example, whether it will bind chemically certain molecules, right? Whether, for example, two proteins will interact, or a protein and a small molecule will interact. And this is how drugs work. So it's the good analogy here is a lock and key metaphor, that you have same way as you have a key that fits only one lock uniquely, then you have a protein whose surface has these kind of pocket-like structures that have both geometric and chemical properties. So these are charged surfaces that will accept only one specific molecule that is shown here. And this is actually how drugs work, because these pockets are responsible for activating or disrupting chemical reactions. And if I inject a molecule into my body, for example, by drinking from a cup of coffee, right? So this is a small molecule that is shown here. It's actually not caffeine. It's a similar compound from the family of xanthines. So it will get into my brain and bind to this protein that is called adenosine receptor. And it will activate some chemical reaction, and it will cause certain, maybe even psychophysical changes in my brain. And this is how most drugs work. So you can very roughly and very maybe primitively think of them as a kind of molecule that has holes or pockets, and you stick something into this pocket and it blocks certain chemical reaction. Now, in many cases, there are interesting, there are other mechanisms, for example, protein-to-protein interaction, where you have two proteins that bind together, like the program death proteins. So this is a mechanism that allows the immune system not to destroy our own cells. And these are more complicated cases because the interfaces where these proteins interact are flat. So they don't have pocket-like structures, so they're considered undruggable by small molecules. And one of the breakthroughs in the past decade was the successful design of biological drugs that are proteins themselves. So it's a protein, typically an antibody, that is injected into our body, and then it binds to one of these proteins, PD-1 or PD-L1, and basically gives back control to the immune system. In case of some cancers, actually some cancers express these proteins, and basically if the cancer cell would be normally destroyed by the normal functioning of the immune system, this mechanism is prevented, and as a result the tumor grows and we have a problem. So these kind of biological drugs allow to give control to the immune system to destroy cancer. Of course, the question is how to design these kind of proteins, and with my collaborators in Switzerland, we described a method that we called MASSIVE, that is a geometric deep learning architecture that predicted molecular interactions between proteins, and we could use it to solve important problems such as predicting interaction sites, or predicting which ligands will bind this site, or doing fast protein-to-protein interaction search. And in particular, we can take some target, like for example, the PD-1 or PD-L1 protein. We can screen large collection of small peptides or protein fragments, and then build a protein drug around them. Right, so that will be the active part that binds the target, and this way we can design de novo from scratch new proteins with desired function, like binding our target. So that was a long collaboration. So you can see here are some examples that were actually confirmed experimentally. So this is a new binder for the program death ligand. So that's the typical oncological target for immunotherapies. You can see here the designed protein and its crystal structure, and these plots show that it actually binds the target and it's specific. And this is its structure that was confirmed by X-ray crystallography very accurately. So it indeed it is what we designed and not something else. So that's the level of details that you need to convince biologists that this protein is actually works. And here's another example. So this is probably a protein that is the most studied in the recent history. So that's the spike protein of the SARS-CoV-2 virus that causes the COVID disease. That was the plague that pestered us for the past almost 3 years. And here you can see that this is where it binds to the ACE2 receptor. So that's where the virus enters into the host, into our body. So here again, we designed proteins that will bind to this spike at a slightly different site. And by doing so, it will prevent its interaction with the host. So here we show that, first of all, that it indeed binds. And then we also had experiments on different variants of this spike protein from different variants of the virus. And you can see that we are still able to bind to many different versions of the virus that existed throughout the pandemic, as well as a comparison in pseudovirus neutralization comparison to an AstraZeneca drug that was clinically approved. It's an antibody that was designed traditionally by traditional methods, by evolution. So I think it's pretty impressive, or at least to me as a computer scientist, that something that we did on a computer actually works in the lab. And well, I'm not saying that it's necessarily immediately a drug that will be available, but I think it's pretty impressive that it is designed end-to-end in a data-driven way. Now, the dream, of course, would be to have something that looks like a DALI 2 or the Mid-Journey or image, and some diffusion model that will generate a molecule with some properties. And in this case, the property is conditioning on the target protein pocket, right? So we want the molecule that fits into this pocket. And these are example of recent works with Max Welling and Bruno Correa and his team, where we showed two examples. So design of a molecule by a diffusion model that is conditioned on the target protein 3D structure, as well as a diffusion-based linker that starts with molecular fragments that are docked to the target protein and then links them together. And this is one of the standard ways of structure-based drug design, where you start with small fragments of molecules called pharmacophores that then are linked together into a single compound. Now, a cheaper alternative that is being explored by the pharma companies, instead of designing a new molecule that usually will take a lot of time and money to approve, to go through all the clinical trials, you start with a drug that is already approved and considered to be safe, and you use it either against different target or in combination with other drugs. And graph neural networks have been used here as well. So the pioneering work was done by Marinka Zitnik, who is now a professor at Harvard Medical School. So she in particular predicted the side effects of pairwise combinations of drugs using graph neural networks. And a similar method, I was involved in a big collaboration that was funded by the Gates Foundation called Recover, in collaboration with Mila, the group of Joshua Benjo, where we tried to find synergies between different oncological drugs. And actually that gave also interesting results, some of which were consistent with clinical trials. And finally, this is not necessarily need to be limited to synthetic compounds. We can actually look at the compounds that naturally occur in plants and in plant-based foods, what is called phytochemicals, so drug-like molecules that, for example, give broccoli its color or blueberries its special flavor. And this is really the dark matter of nutrition, because if the regulators and consumers track the content of, let's say, proteins or carbohydrates or fats or vitamins that are contained in food, nobody tracks, for example, the content of indoles or polyphenols. And these are really molecules that interact with other molecules in our body. They have biochemical effects, both cumulative and immediate. And we wanted to understand what they actually do and whether we can find foods, for example, that might have a compound similar to oncological drugs. So the way we did it was also using graph neural networks. So we tried to study how drugs interact with proteins in our body, and using graph neural networks or similar methods to predict drug-like molecules from their interactions with target proteins. And once this kind of network was trained on known FDA-approved anti-cancer drugs, we were able then to apply this classifier to other compounds that come from the plant kingdom, basically molecules contained in food, and this way create a map of potential anti-cancer compound containing foods. And you can see that there are many quite usual suspects, such as cabbages or tea or grapes, that contain compounds with known and actually biologically confirmed functions that can prevent oncological diseases. Now, I'm not saying that you can cure cancer by eating the right stuff. It is probably the doses are significantly lower than, and the specificity of these compounds is not as good as what is used in chemotherapy, for example. But there are actually some anecdotal evidences that show that consumption of certain foods significantly increases the outcomes of cancer treatment. So it's interesting actually to see how these can be integrated with traditional therapies as a co-therapy, and maybe at least knowing that you're eating foods that are not interacting adversarially with the chemotherapeutic, the traditional therapies would be very important. And to my knowledge, there is no current nutritional guidelines that are widely accepted for cancer patients. So probably the coolest part of this collaboration, and I would like to finish on this tasty note, was a collaboration that was sponsored by Vodafone Foundation with stellar chefs. And here you can see Bruno Barbieri. So those of you watching MasterChef Italy would immediately recognize him. So he's a Michelin star chef that used the ingredients that we identified to prepare simple and efficient recipes that might have therapeutic or preventative effect. So I will stop here, and I started with a quote, and I would like to end with a quote. So this one is from Helvetius, that I think captures very nicely the idea and the spirit of geometric deep learning. And he said that the knowledge of certain principles compensates for the lack of knowledge of certain facts. And indeed, I think understanding the principles from which you can derive different deep learning architectures is probably the right way of teaching and approaching deep learning problems and applying them to new types of data, new types of problems, and building future better architectures. So I will stop here. Thank you very much for your attention, and we'll be happy to answer any questions.
Thank you very much, Michael. This has been a very inspiring talk, especially the last quote. Resonates very well with what we're trying to achieve here today, indeed, to see how can we leverage geometry in medical image analysis as an inductive bias, let's say. Can I ask you, Michael, to stop screen sharing such that we can see you? Because it's, yeah, okay, perfect. Are there any questions from the audience? Yes.
Hello, thank you for the talk. One of the things I always think about is that we're always talking about the equivariance and symmetry of the structure of the data as in the physical structural graphs. But actually, if you think about different graphs, different symmetries have different meanings. So for example, if it's like a social network, if you add an extra edge, it tends not to mean very much, but in molecules, if you add an extra edge, it becomes like an explosive molecule and stuff. So one thing is that I've always trying to understand beyond, permutation and variance. Is there ways of learning invariance or equivariance to, functional symmetries, for want of a better word,
Yeah, it's a good question. So it's probably in this case, well, in the case of molecules, the symmetry comes from the very representation of your data, right? So if you think of a molecule as a graph, then you have the problem of how to order the atoms. Now, learning the let's say specific chemical group that you add and you make something, maybe some innocuous compound into an explosive, and indeed you can just change a few atoms and I don't know, something good will become very extremely toxic. This is something that the neural network potentially can pick up, right? But the underlying symmetry is actually what makes this learning possible at all, because otherwise you would need to show the network all the possible ordering of your atoms in your graph. Now, there are some generalizations of these symmetries because graphs themselves, they also have internal symmetries, right? So you might have parts of the graph that looks the same. This is actually one of the complications, and this is what also confuses the graph isomorphism methods or message passing. This is actually a big problem in link prediction, for example. So there's some more general methods that build on category theory, and this was done actually here. It was Pim de Haan and his collaborators, Taco Coyne and Max Welling, and I hope I'm not forgetting anyone. So they a work that is called Natural Graph Networks that uses more general notions of symmetry that go beyond groups.
All right, thank you very much.
Okay, thanks. Are there any other questions in the audience? Maybe I have one question in the meantime, I think we reach a new phase in our research field that we now start to target new type of problems and be creative with all the tools that we have. And so it's very clear that if you have understanding of the geometric structure, that we should leverage it. And maybe this is a bit like an open question, of course. I guess I know the answer, but do you feel that maybe this is the way to go with research, or is there still a lot of work to be done when it comes to leveraging geometry? Because looking at your talk, I see a lot of components that have been nicely figured out. But is it true that, we're on that component, we're almost done, let's say?
Well, so I don't know. I think it's very, in a sense, it's a very ungrateful job to make predictions in machine learning, because I will probably say something and then a few weeks after a paper will come out that will contradict it. But I think deep learning, geometric deep learning has been criticized for, or I can say already for years, people were saying, okay, you're explaining the past. Can you do something new with this framework? And I think, for example, equivariant message passing shows convincingly that you can do new architectures, and at least this is now widely used architecture. My opinion is that we are not leveraging enough physical intuition, and physics not necessarily in the form of physically informed neural networks where you make something that is physically plausible, right? Like modeling molecules, but taking a physical system as a metaphor. Because we can say a lot when we have a physical system that has certain conservation laws or certain invariants. So we can produce neural network architectures by discretizing differential equations that govern such systems. So we can better understand what graph neural networks do, for example. And I think this is really powerful. And there are deep connections to differential geometry that we are trying to explore. So we had a paper at ICLEAR that looked, for example, at the problem of graph rewiring and bottlenecks using Ricci flows, which is a construction that is used in differential geometry and topology. For example, it was famously used to prove the Poincare conjecture. So yeah, something like this. So I think there is a lot to leverage still from these fields.
Yeah, that's also my impression, indeed. And so are there any other questions at this point? Yes, here's one.
Hi, thank you for the talk. So in conversations, deep nets have been criticized for not having the property of causality, which is said to also be the way that humans have higher order thinking. So do you think with graph neural nets, we can use the same structure, but just a different sort of operation, and eventually come closer to this reasoning capability of understanding images so that your networks are not weak to adversarial attacks and they don't confuse the data itself?
So I think, well, probably, first of all, I will probably give a very high level answer because I don't have a concrete answer, but there are several things that you mentioned. So robustness to adversarial attacks has actually been studied quite extensively in graphs, in particular the works of Stefan Günnemann and his group. So you can actually prove provide some basically provable robustness guarantees in certain cases. In terms of causality, so well, in this case, you will probably be trying to build a graph from your data based on the downstream task. And this has been done as well, especially in biological problems. So Caroline Uller, for example. So you try to basically to describe by a graph some mechanism that, for example, explains how a cell works. And there's, as a result, for example, the mechanics of disease and therapy. Regarding reasoning, so there is also this field of neural algorithmic reasoning, and this is what Petr Veličković, for example, has been working on in the last couple of years at DeepMind. So roughly speaking, if you have, let's say, graph algorithms, right, like Dijkstra, so algorithms of this type, they are totally explainable, right? So you know exactly what it does. They generalize across different types of data, right? So I can run it on any graph and it will produce the same thing, right? The same answer. But they are not generalizable across tasks, right? So I cannot use Dijkstra algorithm to, for example, do graph partitioning, right? Neural networks, on the other hand, they are, you don't know exactly what they do, right? So they're not very explainable. They do kind of generalize across different data, not always, but they do generalize across different tasks. So, and probably evidence to it is that you can train a language model and use it for anything from generating text to spam detection, right? So neural algorithmic reasoning, in a way, tries to combine the advantages of these two worlds. And this is probably a way of approaching reasoning in some way, right? So algorithmic reasoning, of course, not necessarily human reasoning, but yeah, I think that could be an interesting direction that also involves maybe more abstract geometric or geometry-like constructions like category theory.
Thank you. That was a great question and a great answer, actually, because we've been talking mostly about geometry and leveraging in our architectural designs and computation tools that we have available. But it's maybe also time to think beyond what we currently have and think about these computational tools. Maybe one last question, and then we move on to the next session.
Yeah, I wanted to also ask, do you think there might start hitting a limit to graph learning due to the sort of complexity I'm talking like complexity theory, complexity of some of the sort of problems that we're trying to solve, like graph matching. Wait, let me be specific, like graph edit distance sort of calculate. I think that one is NP hard, and my complexity theory is years behind me. I can't remember if that's solvable with classical computers, but I think, do you think we're hitting that sort of limit to what we can actually, what's possible to solve?
Well, I agree. So some problems have too high complexity, but there are always relaxations, right? And well, the best example is probably the graph isomorphism test. So it's actually unknown complexity. It's usually put in a separate class. So we know that it's not NP hard, but we, on the other hand, do not have a polynomial time algorithm. So there was a claim, I think, of Laszlo Babai about quasi-polynomial time, but I think this paper has never really been peer-reviewed, so I don't know whether this is true. So basically, the Weisfeld-Lehmann type of algorithms, they do solve in polynomial time a subset of the problem. So probably finding the right formulation that allows you to have efficient algorithms is very important.
Thank you.
Thank you very much, Michael, and also the audience for their questions. I think we have to move on to the next part. So I would like to thank you again, Michael, for this wonderful talk, and hope to see you in real life sometime soon.
Thank you. Thank you.
Okay, so next up in the program is Mohammed Suleiman. With a talk on geometric deep learning for cortical surface registration, which was already somewhat announced in Emma Robinson's talk. We're looking forward to it. Jelme is checking if it's all ready to go. Yes, and the screen is up. Okay, cool. Yeah. The floor is yours, Mohammed.
Thanks. Okay. Yeah, I think it's really very hard to give a good presentation after a very magnificent keynote lecture. But anyhow, I'll try to do it quick and in a good way. So hi everyone. I'll be talking today about our work on geometric deep learning for cortical surface registration. My name is Mohammed, and this is a joint work with Logan Abdullah and under supervision of Emma Robinson. So I've thought a lot about how to start this one, and then I say, okay, I'm going to put this slide, but it might be a very simple one for a lot of people. But anyhow, so image registration it's a very simple task, or you can formulate it as a very simple task in which you have like two pair of images. That's the case of pairwise image registration, and you would like to find a spatial correspondence between these two images. You might have a point of interest in one of these images, and then you would like to find what's the spatial correspondence on the other image, or like another anatomical point on the other image. I think I don't need to talk a lot to convince you about the importance of cortical surface registration, but it's really very essential task and step in neuroimaging research, and that is actually because if you have multiple features in different brains, you probably would like to project them all into same average template, and then where these features will overlap over each other, and then you will have the opportunity to do comparison between them. So classically, because the importance of this cortical surface registration, people have been doing that by starting by projecting the cortical anatomy to the sphere, which then simplifies the registration problem, and then the registration is performed by optimizing a similarity measure between the features on a target sphere and a feature on another moving or deformed source sphere. While doing this operation, it's very important always to keep track of the smoothness, because otherwise you might end up with peak distortions, and those peak distortions might result in unplausible anatomical structures. That's why really smoothness is also very important while we're doing cortical surface registration. So the contribution of our work is, firstly, we are developing the first deep discrete learning framework for cortical surface registration. that has the ability first to learn large deformations, more like large deformation than what deep regression frameworks are able to do. And also, we found that by a lot of experiments that it has the ability to improve performance in brains with atypical topographies. We also impose regularization as a part of the network. We have a trainable regularization network in a form of conditional random field implemented using RNN, which we found provide us with smoother warps. So our registration at the end is very smooth when we compare it with traditional classical and learning-based registration methods. So as a general overview, so GeoMorph is an unsupervised learning-based framework for cortical surface registration, in which we do registration or we solve the registration problem as a multi-label classification problem. So I'll come back to this point later, but as a very quick overview, we consider that we have a low resolution control grid, and then we have a fixed finite number of labels around each one of these control points. And then our registration problem becomes a multi-label classification problem in which we would like to have a model that will tell us, okay, you have this control point, try to find what's the optimal label assigned for this control point. So our learning is based on using spherical geometric deep learning architecture. Just to recap, and this is like have been covered in the first keynote by Emma, if you have our cortical surface, we normally like represent it in infilated sphere where you have the yellow lines are the top of the folding, and then the greens are the bottom of the folding. And this get projected into sphere. And this is where we kind of start doing our geometric deep learning. So that means the brain is topologically equivalent to sphere. And we have a sphere, then we have a surface. And we know that one of the good graph convolutions, or one of the good surface convolutions is coming from Monad. So we use Monad for doing surface convolutions. But one of the tricky things here is that while Monad will let us to learn features, there are other things that also we need to take care of while we're developing our model. And that is why we do not use any sphere, but we use an icosphere, which is like has some triangular meshes and vertices as well. And the reason behind that is icosphere has this very unique architecture that will let us to redefine some of the basic operations that we take it for granted in CNNs. So like if you have a set of points, so in a certain resolution icosphere, it's very easy to go for the upper or a higher resolution icosphere by just inserting a point that is in the mid between two adjacent points. And you keep doing that until you get whatever resolution that you would like to get from an icosphere. And you can do vice versa as well, if you would like to go from a higher order icosphere to a lower order icosphere. And by that, we can easily define the upsampling operation as like using interpolation of existing points, and then the downsampling operation becomes very easily the operation of extracting lower resolution icospheres. The third thing is also if you have a network, sometimes we need to do a pooling, and then the pooling becomes also very easy because then if you have any, so like the new point will actually be it's either mean or max depending on the type of the pooling that you're using of the direct neighbors in the icosphere. So with that in mind, so what are we hoping to develop while we're doing this registration idea? So we are hoping that I can have certain model, and that model will take my moving image, project it into sphere, and then I have a reference or what sometimes we call a fixed image. That model will take these two together, and then if you look at the the white dotted here, you can clearly see that there is a misalignment between like the main features on the fixed image and those on the moving image, and then we are hoping that our model will be able to align these main features to get our registered image. So how are we developing this idea? So we start by, as I've said previously, so we define a set of control points, and those control points on our moving mesh, and those control points are generated from a lower resolution icosphere. And then around each one of these control points, we define a set of labels. And those labels normally are from a higher order icosphere. So each one of these control points, if I go back, will be surrounded by a certain number of labels. And then we do hope that our model will be able to learn the optimal label assignment for each one of these control points. So for example, here, our model might say, okay, for this control point, it has to move here, so that the features on the moving mesh will be better aligned with those on the fixed mesh. And how do we do that? We do that by solving a multi-label classification problem. So once a model tells you what's the optimal label for this control point, it's very simple. Then you move, interpolate, and resample. So with that in mind, I'm very sure this is very scarce slide, but this is the general architecture of GeoMorph. So we start by, so the network takes both the fixed and the moving image. And then the first part of the network is what we call a feature extraction network that extracts features from each one of these two images or two spheres, and then it gets concatenated, and then we have the second part of the network, which is the classifier network. And this is the network that will tell us what's the optimal label assignment for each one of these control points. I just would like to raise out that all of these operations are redefined on the surface. So we have, it's either like surface upsampling, downsampling, or even pooling are redefined as I've explained in the previous slides. Then we have a spatial transformer, and that spatial transformer, the output that you get here is initially your deformed data grid. So probably this is what you are hoping for. But the problem here is that you are not imposing any kind of constraint. So the control points can move freely wherever they would like to do. And that is why we have the part of this conditional on the field RNN network, which tries to force neighboring control points to move in a similar manner. You would assume that if two adjacent control points are close to, like you would assume that two adjacent control points will have similar trend. If one moves to the left, then the other one most probably will be moving also to the left. So the condition around the field network takes the original deformed control grid, which learned without any kind of constraint, and then gives you an updated version of that that's taking into account the dependency relation or the gen-centered relation between control points. And then once that happens, you resample your features again, and you get your new registered features. So very quick, so we do coarse-affine image registration. So we start with a lower resolution control grid and lower resolution labels. We do that at the first stage of registration, and then we use a larger or like a higher resolution control point and then a higher resolution label grid. And we train our network in an unsupervised way using a sum of three losses. The first loss is a similarity loss, and the second one is a smooth loss, and the last loss is what we call as rigid as possible loss. And this loss is actually trying to enforce the updates that happens in terms of like the updates that happen in the deformation to be as closely as possible to each other. So we assume that the updates, like the update, for example, that happens from the first step to the second step won't be that much difference. So now what's actually happening? You take your reference image, which is the fixed image, and you take your input. You can clearly see that it has nothing to do with the reference. It has like approximately zero cross correlation. And then the first part is we do an affine alignment, where now we're getting like a better cross correlation, and then we feed that into our network, where we get our deformed image, which has like a better now cross correlation similarity. So quickly, in terms of the results, so we have evaluated our model using the HCP data. And actually, I just would like to emphasize that it was very tricky to do the evaluation, because if you rely only on the cross correlation similarity, then some models, as I will be showing in the next slide, might give you a very good cross correlation similarity, but they might end up having a very bad distortion. So you cannot only evaluate in terms of the similarity between the two images. So what we thought is more fair. We have done like a good hyperparameter tuning for all these for all these different methods, and then we decided to fix the cross correlation, the similarity, and look at other measures. And those other measures is mainly about the distortion. The areal distortion is how much the area of the triangles in the icosphere are distorted such that your method try to force this cross-correlation similarity, and then also the shape distortion. As you can see, geomorph is, in terms of like similarity plus distortion, it provides the best performance in terms of all measures of kind of areal distortions. Some methods, for example, like S3Reg, which is a learning-based method, has a very high peak distortions. The second thing is also like we have tried to do a histogram evaluation of the distortion, like looking at all the distortions that happens at all test subjects. Just to recap that, the more distortion that you have around 0, then the better your algorithm is, because that means you are trying to force similarity, but at the same time, you are not losing the shape of your brain. So you can clear that most of the distortions of geomorph is around 0, like comparing to other methods. What's better than geomorph is spherical demons, which is not like extendable to functional data. This is the same for areal distortions and shape distortion as well. So finally, like a quick alignment quality. So yeah, again, so we are getting a very good alignment quality when we compare with other methods. And also when we plot the shape and the areal distortions, you can clearly say that we do not have these like kind of peak distortions that appears, for example, in MSM pair and S3Reg here. So finally, the problem is there are a lot of parts in the network, and then to test the effect of each one of them is really very hard, or like at least to put them all here, it was like very difficult. So I've decided just to pick 2 to show you like what each part how each part is affecting the final performance of the model. And I'm happy later to discuss like if someone is interested on other parts. There are a lot of stuff, so I'll just pick 2 of them to show you. So the first one is the conditional random field, and as I have said, this is the network that imposes smoothness by encouraging neighboring control points to move in a similar manner. So again, just at the recap, it takes the control grid, the deformed grid that's learned without any kind of constraint, and the probabilities that will tell you like this label is highly probable to be like the final destination of this control point. So just as a quick comparison, so this is our performance with the final model that I have shown. But once we drop the conditional random field network, I mean, as you would expect, we will be getting a better cross-correlation similarity. But then if you visualize the results, you can clearly see that we are having now a very high distortion, in which now we are like losing some of the main like regions in the brain. The other thing is the part of the feature extraction plus as rigid as possible loss is, and this is actually built on the top of our previous work, which is DDR. So what this part is actually adding is it's just providing better results in terms of distortion, and this is kind of expected because now we are having like an extra loss that will somehow regulate the distortion that's happening here. So what's next? So what we are currently doing is, though this is all evaluated on Silk depth data, so we are now like rerunning everything on the functional data, which is actually which was the main driven of this work when we first started doing it. And like results will be soon out. With that in mind, yeah, thank you very much for listening.
Thank you for a wonderful talk. I will take questions from the audience. Maybe I would like to start off with a question of my own. I have a question regarding how well this method, which is really intriguing, generalizes to non-spherical objects, or to meshes in general, because now you still project on the sphere. And I think you predict these control points, right, per location. And does this work by the premise that based on this icosahedron subdivision scheme, you have a similar or the same set of control points actually at every node? Or is it also generalizable to the case where there's different stencils, if you will, or control points per location?
So the main concept is there, but you need to redefine. So in terms of learning features, the monoconvolutions will be easily adaptable to different structures.
Sure.
But then you need to go back and redefine some of these other operations that we are defining based on the sphere structure. I think this is the only thing that you will need to redo again.
But don't the control points that you want to predict at each node need to be semantically or geometrically meaningful in the same way everywhere you go? Or am I allowed to freely choose a set of control points that I want to predict at each location on my manifold?
So in terms of defining the control points, we are defining them equally spaced, so that you will somehow make sure that you are covering like the whole domain of the shape that you are having. I think as long as you can replicate that, I don't see an issue of like generalizing this to different domains. That the concept is that you would like to have an equally spaced control points that will cover the whole domain of the shape, and then once you can redefine that on the different shapes, then I think it's easily doable.
Okay, nice. Yeah, there's a question by Matthias.
Yeah, thanks a lot for the nice talk. It's maybe a follow-up. So, is this the possible displacements that you can assign to one control point? Is that layout rotationally invariant, or it isn't?
That is fixed. So we fix the number of labels and the locations of labels at each step. So that is just defined at the beginning. And
but
then
that also means that if your brain and the projection to the sphere is different.
Sorry?
If the projection of the brain to the sphere is different, if your brain was kind of rotated 180 degrees, does it still work the same way, or is that?
It still works the same way. So because it's relevant to the control point. So wherever you define your control point, those labels will always be located around this control point in hexagonal shape. So even if you rotate it, I think it's still the same.
Okay, thanks.
Okay, I think it's indeed a matter of gauge invariance. That's kind of what the question refers to. Also, my question touched on these stencils or these control points are maybe defined relative to, well, I can order them in any particular way I want, right? So this implies that the method that you use, the convolutional or the graph network, has to be equivariant on the mesh?
Yes.
Yeah, okay. Are there any other questions? Here up front.
Hi, first of all, thank you very much for the great talk. So I'm wondering about, in your loss, where you're optimizing, actually you mentioned you have a smooth term and actually something for topology preservation somehow for plausible transformation on your grid. So, and then in the table, you have DDR, that if I understood correctly, is your previous work, right, and the current one. So taking a look about performance-wise, I'm wondering if actually this additional regularizer that you have there for enforcing the smoothness actually has the effect reflected or is not quite reflected on the performance in the table. Because, can you go to the table, please? Yeah, DDR and geomorph are quite close at each other, which might be actually, from a statistical point of view, quite the same performance-wise, right? So I'm wondering if actually the impact, where is coming from the geomorph, or what is the advantage of the DDR with respect of, for instance, the loss that you're optimizing?
Yeah, I think so. The first thing that you can clearly see, maybe the highest effect of geomorph is on the max distortion. So it's like this is the highest effect of what's happening from the as little as possible loss. But actually, when we started extending this work to work with functional MRI data, we dropped that term. So we found that we no longer need it. So for the current setting, it's there and it's providing a better performance in terms of distortion in general. The difference is not that much. The higher difference is coming from the max distortion, so it's like penalizing these max distortions. But when we applied that for functional MRI data, we found that we no longer need it, and we have dropped that term. Yeah.
Okay. Yeah. Thank you very much. I think. Let's thank you again one more for this great talk, and then move on to the posters.
Yes, the last part of the session. So we have seen 8 great orals already, but we actually had 25 accepted papers. And every paper has a poster over there, so we'll have the poster session immediately after this. But before we go there, we will have 16 presentations still, at a very high pace. So I'd like to ask all of the poster pitch presenters to come forward. Each one will have 1 minute. And unfortunately, there was one... luckily, they're all on one laptop and they're all connected. There was one video that I first want to show of one of the posters that unfortunately couldn't be here physically. Er was een geluidskabel, toch? Oh, wait, okay, hij zit er al in. Okay, so this is the first pitch, which is exactly 1 minute.
Although MRI is the gold standard imaging modality for soft tissue analysis, in cardiac MRI, the images are acquired in a collection of sparse 2D planes. We are proposing a multi-step 3D reconstruction pipeline which, starting from sparse misaligned cinema images, automatically generates a mesh with corrected misalignment. The main contribution is the mesh deformation unit, which addresses both sparsity and misalignment issues in a single model. Here, the unit-inspired encoder-decoder structure is capable of efficiently performing multi-scale feature learning directly on anatomical surface meshes. Application on synthetic dataset derived from 3D statistical shape model shows good alignment between reconstructed and gold standard meshes and quantitative performance of average surface distances below underlying image resolution. Analysis over 1,000 reconstructions of the UK Biobank study demonstrates clinical matrices in line with other population studies. Thank you very much for listening. Please feel free to contact me for any discussions.
Guys, take note, that's 1 minute. So, you are very nicely ordered, but I think our pitches may be in a different order. So, if you see your name, please come forward and take the stand. Yes, so we start with Patrick.
Hi, so I would like to introduce our work called Eigenvector Grouping for Point Cloud Vessel Labeling. This is a joint work between Hemolens Diagnostics, Tuplux, and Wrocław University of Science and Technology from Wrocław, Poland. Our task is vessel labeling, which is a refinement task after voxel-based vessel segmentation. And in our task, we want to solve the two issues which are common for vessel segmentation. These are false positive vessels being detected and disconnected vessel branches. So previous approaches utilized point cloud based networks. We utilized the same network and they use different grouping strategies. They were mostly agnostic towards underlying point cloud structure. We propose a novel solution which is based on grouping the points with the top eigenvector of local point neighborhood. This grouping is efficient because it is points are being grouped along the direction of the vessel. Thank you very much.
So, hi. So in the disease model. In the disease progression model field, it's fairly common to assume that the data lies on a manifold and that the progression follows a geodesic over time. So if you manage to somehow learn a Riemannian metric of the observation space, you can define all the trajectories of the patients for biomarkers, for instance. However, it doesn't scale to high-dimensional data, such as neuroimaging data. So we proposed an architecture that blends a variational autoencoder, so for dimensionality reduction purposes, with a very simple, straightforward linear mixed effect model in the latent space. And our main point here is that we can mathematically derive the fact that it's just an extension, actually, of the well-validated Riemannian progression model framework. And the neural network actually does nothing more than just learn a fancy metric for a very high-dimensional space. So it should not be considered as a black box because it's just a parametric way to learn the metric. Thank you.
We'll all the way at the back of the queue.
Okay, hello again. Hopefully you remember something that we talked about initially. If you remember, while I was concluding my previous presentation, I said we did not use heterogeneous graphs and we used a lot of pruning. And I am here to assure you that we did not, we did follow up on that. So it's not that I just opened a question and then went home and slept. We are working on it. The reason why this is not a full-blown paper is that we still have a master student working very hard back home. So hopefully by next time, this is a full-blown paper. But we still try to see what happens if we move to a heterogeneous graph. How does it work if we try to combine both these sources of information still, and if it works better or not. So if I have intrigued you even a bit, please stop by my poster and we can discuss things in detail. Thank you.
Yes, it's me. Okay, good. So, yeah. So a correct measurement of blood pressure is very important to monitor the health status of a patient and more in general of a human being. Nowadays, we have light portable devices that can obtain in a non-invasive way some biological signals from an individual, like the photoplethysmogram, PPG, or the electrocardiogram, ECG. As the blood pressure is known to be linked with both the PPG and the ECG, can we develop an algorithm that predicts the former from the latter? Many non-geometric algorithms have been proposed for this task, but we tried to give a geometric deep learning answer to the problem. So we proceeded by giving to the PPG and the ECG signals a particular graph structure that could be fed to purpose-built graph neural network to predict the average blood pressure over a desired period of time. We found that the resulting models actually do not underperform the standard techniques when it comes in terms of both accuracy and also interpretability. Thanks for your attention. I look forward to speaking with you.
Hi all, my name is Florian and I'm a PhD student at the AI and Medicine Lab at TUM. And among others, we work on privacy-preserving deep learning, especially on differential privacy. And differential privacy is interesting for us because we want to prevent reconstruction attacks from happening on medical models and protect patient data. However, there's a big trade-off, especially in the prediction performance and also on the computational overhead. So we look at developing more efficient models. And for that use case, we looked at equivariant convolutions and how they perform under a differential private setting, mostly because their characteristics are actually a good combination with differential privacy, maybe even more so as a non-private training. So if you're interested in the results and want to talk more about model efficiency and learning theory in general, come by at my poster later and we can talk.
Okay, my name is Ramya Reddy.
And my name is Ram Reddy, and we're students at Thomas Jefferson High School in the United States. And over the past few years, we interned at Dartmouth under the guidance of Dr. Joshua Levy and Dr. Louis Vicus. So our motivation for this project was the limitations of colorectal cancer, or CRC screening. And in the past, in the United States alone, there are like 50,000 deaths caused by CRC every year. And to solve this, we need to improve CRC screening and prognostication.
So one important factor in prognosis is tumor infiltrating lymphocytes, or TILs, and this is because they can tell whether a tumor can evade the immune system or not. In addition, when localizing TILs, pathologists usually need to have an extensive curation of annotations, but we were able to substitute this with an automated staining procedure. We also innovatively were able to filter out whole slide images based on quality of co-registration and applied graph neural networks to classify immune cells. The graph neural network model outperformed comparison approaches, and we're also planning on using our using our research to further explore the tumor immune microenvironment.
And thanks for listening, and we hope to see you at the poster.
Okay, you can hear me, right? Perfect. All right. So shape reconstruction using deep learning has many applications in computer vision and in the medical domain. But recently, implicit neural representations achieve state-of-the-art performance in the shape reconstruction task. And these implicit neural representations, they try to represent the shape by by a function, by the C-level set of a neural network at theta. However, such implicit neural representations have some key challenges. For instance, assume we want to combine these implicit neural representations with a latent space, then the current literature does not properly address questions like, can we adequately learn a template shape, or does this linear interpolation of latent codes actually correspond to a plausible intermediate shape? And these questions are actually important as they relate to generalizability. And in this work, titled Structure Preserving Implicit Shape Encoding via Flow Regularization, we present an approach that jointly learns a template shape and tries to improve the shape interplay such that we actually have physically plausible intermediate shapes. So if you're interested, please come visit the poster and I can tell you more. Thank you.
Hello, I'm Abdullah. I worked with Dr. Robinson, who gave a talk earlier this morning, modeling cortical neural development on the surface. Cortical neural development, very important. Lots of myelination changes, changes in cortical folding, and two continuous variables interplaying post-menstrual age at scan, J at birth, inter-subject variation as well. It's quite a complex task. But you'll have to come to the poster to find out how. We get these very nice age progression, regression results. Much more excitingly, if you take a preterm and you predict its progression and then take a second scan and try and find the difference between what we predicted, you get correlation cognitive outcome. So, woo! Thank you.
Yeah, hello everybody. My name is Fabian Sinsinger, coming from KTH in Stockholm. And the project that I present today is on tractography, so diffusion-based tractography with rotational equivariant agents. So diffusion image, as we heard before, is you have an image from an MRI acquisition consisting of a field of spherical function at each point in the image. And what tractography is trying to do is you try to track a lot of local paths that represent the neural fiber bundles. And recently there has been reinforcement learning added to this family of methods, but the problem is that while previous older methods have rotational equivariant between the inputs and the outputs, the reinforcement learning methods, they don't preserve that. So our suggestion is basically to add the spherical equivariance back into the model by using spherical agents and spherical critics for the model. And we run some very preliminary results in order to show that we're indeed like more rotational equivariant. But this is ongoing work, so if you have ideas or if you have anything, we're still in the method development phase, so we can talk and probably I might change things afterwards and might adapt. Thanks for your attention.
Hey, can you hear me? All right, so I'm Andrew Lizarraga, and I would like to pitch to you StreamNet, Wasserstein autoencoder for white matter streamline analysis. Here's just a bit of a preview of what the architecture looks like, but we could, of course, discuss that in further detail during the poster session. So what's the problem at hand? Essentially, we get these complicated tracks of streamlines that result from T1 diffusion-weighted imaging, and what we would like to do is perform some form of statistical analysis between the overall shape structure of these streamlines, but as you can see, they're quite complicated, and this makes it difficult to do these sort of analyses. So what would be nice is that if you could embed these into some latent space, but in such a way that the latent space preserves important geometrical information. And essentially, a little preview here is that we can encode and then decode it, and you see that decoding is roughly about the same, meaning we are preserving some, at least some properties of the streamlines. But what's perhaps more interesting is that we then perform statistics and average these clusters here. And essentially by reconstructing from these average latent representations, we're able to produce templates that allow you to do population statistics. But of course, if you'd like to learn a little bit more about some of the other details of this paper, please stop by the poster session and I'd be happy to explain more. Thank you.
Hi, I'm Yannick. I'm here to present our work on automatic intracranial vessel labeling based on vessel center lines. We focused on the automatic labeling of intracranial vessels, which allows for the cross-sectional quantification and the cross-sectional quantification and the comparison between subjects if done reliably. This in turn allows to identify possible risk factors that stem possible in cerebrovascular diseases. We did so by extracting the segmentation from MR images and extracting the centerline vessel width and training a pointed plus model. On the bottom left, you can see our performance based on the manual labeling on the very bottom left and our performance propagated to measures, and on the right is the results. I'm sorry, this was a bit bad. Feel free to come by the poster for a better explanation. Thank you.
Hi. My name's Rajat, and I'm presenting our paper on deep structural causal shape models with Imperial College and Microsoft Research. So in medical imaging, we might want to answer questions of the form, given a patient's brainstem mesh, if this person had been 10 years older or younger, how would their brainstem shape have changed? Effectively modeling the prognosis of that patient. And this would be an example of a counterfactual question. So notice that this question is a bit different to the standard kind of age regression problem. So to answer more complex questions of this form, we propose a method using functional causal models. We encode all the assumptions we have about the world in that kind of functional model on the left, and each of those is implemented with an invertible neural network. And then using these functions, we can generate counterfactual meshes, which answer the question that we posed in the previous slide. And here's an example of some of the counterfactual meshes that we generated. So if you go horizontally, you can see what would have happened to the individual if we kind of intervened on their age. And if you go vertically, you can see what happened if you changed the sex of that individual. And here's some examples we generated on hippocampus meshes as well. So there's been a lot of work on like generative modeling of meshes in this field. A lot of work on 2D causal modeling as well. But ours is the first work to do full causal modeling, including counterfactual generation of 3D meshes. So to conclude, like we did kind of the first causally grounded 3D shape model. We answered counterfactual questions. We used a mesh conditional generative model, effectively a VAE. And you can apply this technique to loads of other problems, including like kind of seeing if a particular treatment affects a disease over time and things like that as well. So yeah, come to our poster and have a look. Thanks.
Hi. So hemodynamic quantities can be used for clinical indicators. For example, low and multidirectional wall shear stress has been shown to correlate with plaque development and arterial remodeling. We can obtain these biomarkers non-invasively by some medical imaging modality and computational fluid dynamics. But this takes a lot of time, and sometimes we might want to sacrifice the accuracy this brings for evaluation speed with a surrogate method. So we propose to use graph neural networks that operate directly on a surface mesh, and in particular gauge equivariant mesh convolution. The idea behind that is to represent your signals, you have to pick a local coordinate system at each vertex of the graph. But this choice is ultimately arbitrary. So the different choice of gauge must not change the result of the convolution. And we can enforce this by constraining the weights of the network, so this diagram commutes. So this actually also gives us a second symmetry for free, namely rototranslational equivariance with respect to the input mesh and the output vector field. And this property is actually crucial for the performance of our network because blood flow does not really depend on gravity. So if I rotate my blood vessel, then the quantities of the blood flow should not change, so the magnitude of the quantities. If you want to hear more, feel free to stop by my poster. Thanks.
Hi, I'm Shuprashanna, and this is joint work from Technical University Munich and University of Zurich. So over the last few years, we have made decent progress in segmenting blood vessels from either biological image or clinical image. But that's not the end of the story. The clinicians or the biologists want to do more. Specifically, they want to learn functional properties from these infrastructures and want to kind of correlate those with some biomarkers, different diseases. So for that, they need to kind of simulate blood flow on top of it. And the research question we are asking here is that can we make this flow computation on top of graph end-to-end, kind of avoiding the classical way of solving numerically, and how can we leverage the known framework in the geometric deep learning in order to do so. So happy to chat over the poster session.
Hello, I'm Julius Meyer from Institut Berlin, and together with Daniel Baum, Felix Ambelan, and Christoph von Tikowitz, I did some semi-supervised Alzheimer classification, and as data we used hippocampus shapes and came up with or developed vectorized shape features that also capture extrinsic geometry and put them in a graph-new convolutional network as node features. And the shape features rely on function-wise correspondence on manifolds. So if you're interested in a little shape analysis, see you at the poster.
I think that's it, right? Yeah, okay. Thanks everyone for running this so smoothly. We'll now have the coffee break / poster session. So do drop by at all the posters and have discussions and share your ideas. And be back at 4 because then we have the panel discussion with an amazing lineup, which will be very interactive and lively as well, I think. So enjoy the poster session and see you at 4.
All right, everyone. Welcome back to this last part of our workshop. So we have this panel discussion, and afterwards we have a closing and award ceremony for the Best Paper Award, and drinks, of course. But I would already like to start off with a somewhat informal setting towards this final event. We have this panel discussion, primarily because now we're all together from different disciplines, maybe as a research field. So I'm really happy that I get this vibe of community going on here today with a lot of new applications and new technological applications, and this is really interesting. And today, with this panel discussion, I want to probe a bit whether or not we're on the right track. And to this end, I want to get some input from people from different perspectives. As such, maybe I would already like to call forward the panelists. So maybe Taco Quen. Taco is a prominent geometric deep learning researcher, has done a lot on, please take a seat, on group equivalent deep learning. So I asked him to be on the panel from this perspective, but also because he's working for Qualcomm, a big company that is invested in building new deep learning technologies and technical products. So that's Taco. Thanks for joining. Clarissa Sanchez. Clarissa Sanchez is professor here at the University of Amsterdam in AI for medical image analysis. So she's really at the intersection of deep learning and clinical applications as a researcher. Emma Robinson. So we know about Emma today, so that's great to have you also in the panel. A somewhat similar profile as Clarissa, more in your case, specifically also in geometric deep learning and its applications. I asked Gabriele Cesar, who is a PhD student here at the UVA and at Qualcomm, so a joint position there. I asked Gabriele because he's the developer of an open source code library, ESCNN, that implements group convolutions in all its flavors. And I noticed it with my team, and when I talk to other researchers, that this library is kind of really valuable to the community. So I want to also ask his perspective on this kind of community service aspects. And then finally, we have Jorrit Glaastra. There you are. Jorrit is CTO at Quantip. So Quantip is a medical software company for the analysis of medical image data, so brain, quantifying structures in the brain, but also other anatomies. So I asked him to also get a sense of his perspective on what we as a community are doing and whether or not it makes sense. Angelica, would you like to say something?
Yeah. Well, first of all, after introducing the panelists, we would like to, again, thank you for being part of this panel discussion for GeoMedIA 2022. So the dynamics is as follows. We will encourage all the participants and the public, the public in general, to participate through questions. If you have any question, Eric will be moderating here and ask some particular questions, but this is not only about that questions. If appears that you have, or I have a particular interest in a particular question, just please raise up the hand and we can start a discussion on that.
All right.
Thank you.
Thanks. Actually, I want to start off with taking orders for drinks. I thought maybe that's a nice way to make it a bit more informal. So I'm going to call them. Clarissa, what would you like to drink? Water. Yeah. Okay. Maybe Jelmer, can I give this to you, the phone, and I can start the discussion, and we don't have to wait for this. It should be Boris or Claudia. All right. I would like to start off maybe with a somewhat blunt question, like, still water, yeah, okay. This workshop is obviously, let me speak for myself, much appreciated by me as a researcher, like to see all these efforts in research. And we see a lot of interesting technological advances, like and advanced stuff from differential geometry and also advanced on the deep learning side. Maybe just wondering how, in the end, if we want to make impact, because that's what we want, most of us at least, are we on the right track? So actually I want to start off with a question maybe to you, Jorrit, if I may. Like, we've seen interesting methods like group convolutions and all, but are group convolutions really going to help patients?
Okay, so you promised me beforehand that it didn't matter that I didn't know much about what was going on today. So I tried to study up a bit beforehand. And to me, I think for us, from a company point of view, there is a lot of great research going on in academia, and we try to pull on that. We try to shape it a little bit, but also we just try to listen and follow what's going on. Because I think PhD candidates, master students, they have a lot more bandwidth to experiment also with things that don't work out in the end, or don't in the end impact patients. And the thing is, you can't figure out which things work without trying all the things. So I don't know if maybe it depends a little bit on whether you consider this field mature. And I saw you asking earlier today, like, are we almost done?
Yeah.
It feels to me like you're not almost done yet. It feels to me like there are a lot of avenues still to experiment. One thing that I didn't hear a lot about, which I was curious about, is maybe impact of any uncertainty working through geometric models. So maybe there's an avenue there. To me, most of what I've seen today is a little bit far from application. But the thing is, stuff cannot mature to application without trying a lot of stuff.
I see.
So I would encourage you to keep going. And you can't predict, I can't predict at least, what stuff is going to impact patients and what's going to impact patients most. Yeah, so I think that's my take. It's not a very focused answer maybe, but
no, it's clear.
Yeah, you have to try stuff to find out. And as a company, we don't have time to try everything. So we need other people to try stuff. And I think that's also where academia can excel. Fresh ideas, trying new approaches, and to try to find out what stuff can work in the end.
Yeah, okay, that's a nice summary and a nice answer. I think maybe later I'll get back to you on this topic. Maybe Claire, so would you like to comment on this statement?
Yeah, I agree. There is still things that need to be done, but not only for geometric deep learning. I mean, also for things that we are doing. I mean, in general, machine learning for medical imaging or medical data. There are still things that need to be taken into account. I mean, there is a lot of things going on in trustworthy AI, and that's going to be key if we want to really translate things into clinical practice, because there are things there that we have to take into account, like ethical consequences, legal consequences, but also from the method point of view, is it resilience? Is it, can we trust it? Is it robust enough? So I think, indeed, there are still, we still have, in general, for AI and health, we still have a way to go, but I think I'm going to stay with the last sentence from Michael's keynote, that it was really good saying, like, okay, we have a lack of knowledge that we thought we were going to obtain just from data, but now we can really get these models to give a knowledge about about principles, or we can also get knowledge from biological processes that are also that need to be also incorporated. So I think that we are in the good way, in the good path, but there's still there is way to go. Okay, that's great. I get the sense from both of you that indeed, in order to translate this to clinical practice, it's not about per se about fancy geometric deep learning that basically push well, the tables in your paper to bold numbers, let's say. But it's more an important message that I also hear on your side is the idea of uncertainty and having something that you can rely on. And I think this is also something that in Michael's talk became clear, like at least on geometric priors, that's our, speaking for myself, our view of what you can rely on. No, at least that part is covered. And yeah, maybe if I on this topic, Taco, you have maybe a few.
Yeah, sure. So I'm not an expert on anything medical, but I could still share my thoughts on this in general. I think, you know, geometric deep learning is a very mathematical field, and I love doing mathematics. But what I think is sometimes academic communities, they can keep going just because the ideas or the methods tickle the brain in the right way. Let's say they're interesting to work on for people like me, and I think so. For that reason, it's very important to regularly ask, are we indeed doing something that's going to have an impact? Now, I happen to think, that's why I was working on this for a long time, that actually data efficiency, which is what geometric deep learning gives you, among other things, is very important in medical imaging, more so in some areas than in others, perhaps, but very important nevertheless. But still, I think for every single paper that you write, you should be asking if the trade-off between the additional complexity and the actual value that you get is in the right balance. And another comment is to not specialize too much on purely the math, but also to combine it with good engineering, because I think, on the one hand, you have the very pragmatic deep learning people who just, have this mindset, let's train a bigger network and get more data and just get it to work. And all this math is probably useless. I think there's some grain of truth there, but it's a very limited point of view. And then you have the people on the other end of the spectrum who are very into the mathematics and the beautiful ideas and so on, but maybe they don't have also the practical engineering skills to actually make that impact. So trying to balance those, learn about both the engineering and the math, I think is very, very important. And then I would also agree that beyond just getting this to work well, get high numbers, there's probably a lot of regulatory hurdles and the robustness and other issues that I can't speak to, but are probably very important as well.
Yeah. Okay, I mean, that's a good point for next question. Often used argument in deep learning is skill is all you need, right? And so if we just have more data, then we can just go on and solve our problems. I guess it doesn't apply in medical imaging because data is much harder to acquire. But I would just want to quickly verify: is this still the case? Do you have experience with this, Emma, for example? Acquiring data sets that are large enough, and I know if you have been engaged in maybe translating things to clinical practice to that extent.
Yeah. Well, I mean, we're acquiring increasingly large data sets, so the biobank may acquire up 100,000 images, but that's not really in the same league of numbers as ImageNet still, and yet the complexity is even higher. So you're talking about very high dimensional data sets with many multimodal, with many different features that you can model each imaging. So in that sense, group equivariant methods and trying to build models that work on, rather than just throwing as much data as you can at the problem obviously makes sense. But I think always if you're going to, any application of machine learning, it has to be motivated by the clinical problem. And hopefully I made a reasonable case that for the cortex, there is a problem there that can't be solved with conventional methods. So perhaps there's a solution. But again, as Taco said, there's going to be, we're not even in a phase yet where there's full regulatory permission to be able to use machine learning. So those barriers have to be crossed first, and that's a question of proving that these methods are generalizable. There's a question of bias. Yeah. All these things need to be addressed before they can be deployed in the wild.
All right. Yeah. Actually, maybe I want to ask you another question on this idea of making impact. Let's say, and we see quite often that, well, from deep learning or research in general, some breakthrough happens, and from that flows to these results flow to new applications, things that are now all of a sudden are made possible. And so in that sense, any type of research, curiosity-driven, not always, but can end up making an impact. But you could also maybe reverse this and identify concrete problems, like, hey, because I think you have a lot of experience in, okay, what might be possible with technology, but it needs research to develop and figure out ideas. So I wonder what your approach in this is. It like really purely curiosity driven, or maybe do you focus on concrete, like applications, like more like almost existing applications that are just not there yet? Yeah, I guess not, because okay, sorry, I'm blurring out.
I'm not quite sure. I'm sure you're asking, but when you know when you would develop any, I think whenever we're developing a method that or a geometric deep method, we're always trying to benchmark it against simpler methods. You know, you want to prove that it's not a data agnostic problem. It's not just proving that you can apply a method to the data. You really have to show that it's actually going to lead to some benefit and sort of be robust and honest and transparent, I think, about your validation.
Yeah. Okay. Nice. Yeah. That's. Thanks for that answer. Maybe just out of curiosity, you're doing your PhD, Gabriele, so you're probably still in this mode of let's be creative and do whatever we want. So is this, maybe I'm simplifying it too much, but is this kind of on your mind, like how to make impact, or are you mostly in it for the research, let's say, for the excitement on that half?
That's a good question. I mean, I think that, I don't know, from my point of view, during your PhD, right, you have to balance a bit the two things, right? You start that you're very excited that you want to do biggest impact, right? And then you start searching for new directions. I mean, the search for a direction, right, to follow.
Oh, sorry. No, yeah, sorry, that's also my fault for trying this out. Okay. Yeah, let's try to go on as if nothing happened. Sorry for this. So you were saying, at least you have your beer.
I mean, part of the research is, finding like a direction to, that you can follow and where you can push, right? And there's the part where once you find what you want, what problem you want to solve, right? You can be more creative, and I want to find new fancy solutions. I can find some interesting math there, right? And then, I don't know, my impression is that throughout your PhD, your view changes a bit. At the beginning, you're like, okay, let's find some really interesting problems to solve. And then once, I don't know, as you proceed, you tend to, pick a couple, right? Focus more on those. And then you're more like, okay, what can we bring new in this topic? What is something that has been overlooked before? What is some new insight we can, use to develop new methods? I mean, in general, I would say that, to sum up, it's always a balance between the two. It's very important, the research of what are interesting new problems to solve, right? And also what are new solutions we can bring in existing problems.
Yeah. Okay, that's actually... I have some questions on that, actually. Actually, I want to remind, indeed, if there's questions from the audience, please feel free to tune in, right? And also for you, if you want to grab the microphone from one of you, just do so. Maybe more breaks, but that happens. Maybe regarding, I think Emma, you mentioned something about...
Sorry,
Aaron. Oh, yeah, okay, there you go.
I just want to comment a little bit on this scaling topic before we move on. And since I'm in front of a big room full of brilliant people, I want to pitch this idea. So we were discussing today on Twitter about scaling laws, and basically this is about predicting the performance of a model as the data set size increases. And they found for various kinds of problems like language modeling, but I think also for image models, that these things are very, very predictable. So, you double your data set, you get a fixed increment in performance for a very, very long time, even up to these gigantic data sets. And there are some clues, but not really any conclusive evidence that equivariant models have better scaling curves. So they just... The loss goes down faster. And I think I haven't really seen any rigorous empirical studies of that, but it would be a super interesting project. So,
Okay, yeah, thanks for pitching this, Taka. Actually, I want to, of course, it still connects with the previous topics, like move on still to this idea of making an impact, because that's what we should aim for as a field. And one of the ways to do this is via challenges. And so you have several members in the panel that have experience with this. And I recall that in the past, like there were many challenges that are sort of trying to get, are motivated by getting the technology closer to clinical practice, right? It's like sort of simulating what if we do this in practice. So that's more application-driven, but going back to the curiosity-driven part, and maybe this requires actually some courage and confidence that the idea that you have is going to make it. That requires maybe different type of concepts, and I think maybe your chance, Emma, tries to solve a new type of problem with computational tools that you know are going to be useful in, or pretty certain of are going to be useful in clinical practice because they allow for new quantification techniques. Yeah, so the question is, okay, maybe I want to start with Jorrit first. Like maybe a statement like challenges should be application driven rather than just let's make some toy example data set or yeah.
Now you want me to say so?
Yeah, no, I'm asking because I guess it's your ambition.
So I think what is hard about challenge is that no matter how well you try to organize this thing, in the end there will be some fixed test set.
Yeah.
And what you will get out, what will be the output is some winner will come out on this fixed test set. And I know internally I've been asked, why don't, for a specific sub-problem, why don't we organize a challenge? It's relatively cheap. That's what people think before they have to prep the data, organize the whole thing. People go like, oh, it's not so expensive because all you have needs some prize money and it's done. They think it's a cheap way to find, but I think by working through a problem internally, so engineers working it through, there is a lot that is gained that you would never get out of a challenge. And I think what's really hard to get out, a challenge is always a fixed problem. So you take the clinical problem, you abstract it in some way, so you assume a workflow, you assume a certain human performance or not. You assume, by fixing the test set, you assume only these scanners or only these conditions, only these sick patients, only these healthy patients. So all those things are fixed, they are not, for me as an application developer, they're not real because this is all continuously moving.
Okay, so my naive viewpoint is that I was actually thinking that maybe company interest would be like, okay, let's organize a challenge and then people solve the product for us and then, well, it's easy from there on, but it's not
There's two things. So one, I'm not sure that works. I'm not sure that from seeing this solution that someone else made, there is enough there for you as a company to take that through regulatory clearance even, because you really need to understand what you built. At least I want to understand what I've built to put that in clinical practice. And the other thing is you will solve the problem you defined beforehand, but by the time you get to that solution, your insight into what you wanted to solve may have evolved already. That's my experience, at least. That evolves, yeah.
Yeah, and maybe I want to switch to Clarissa then, because you've also been organizing challenges, right? Yeah. So what's your thoughts on this? Like, what is actually the point of this? Hearing this, for example. Right?
Well, I think there are different things that can benefit by doing a challenge. I mean, I think that you move the field, you bring some of the tasks that are clinically relevant to the community that maybe they don't have or they don't have access to it by maybe because of the feedback from clinicians or because of the data itself is difficult to do it. So you give that to community, and that already triggers ideas and models that can be of interest later to, of course, to bring it up to the company. I'm not going to say that a challenge is going to replace a clinical trial or a prospective validation, of course, no, but it already brings this task to the community to, for example, develop geometric deep learning solutions that are really focusing on this kind of application. And it really steers this kind of community curiosity towards solving these problems. So I will say that that's one of the things that it helps also is because it's kind of a democratic way to share also data. Because then we can make groups that maybe they don't have access to this type of data, they can have access to them and they can develop these models. So that also helps there. And it also gives the clinicians an opportunity to see how these models can solve the problem that they are focused on. For example, now we launch a challenge, it's already finished, and the clinicians were the one that they were interested to know if deep learning can really solve the problem, and what was the performance. So you wanted really to evaluate what is the performance that can be reached in that part. They didn't say like we want to commercialize it, they didn't say, but they wanted to know, is this solvable?
Yeah.
So that also helped them to
Nice,
to
yeah, that's
engage.
interesting to know. Yeah, finally, maybe also your thought on it, Emma. But before I go there, just maybe I want to give you also a task. Like at some point, I'm going to run out of questions. So please think about if you have also things that you want to ask these panelists. Or yeah. So I just want to hear your idea, because you talked about it, these challenges of cortical surface processing. I just get an idea of your thoughts on.
Well, I mean, so developing the challenge, I think, was because all of the challenge and everything we're doing at the moment is because we've identified an area which cannot be solved with classical image processing. And so if you go back to the question of, do I think things should be application driven, then yes, because it's Occam's razor. You should use the simplest method that is available, but if the method that you've got available doesn't work, then you need to do something new. But I think at the same time, you can't take, a data agnostic approach. With brain imaging, for sure, you need to understand the underlying the neuroanatomy and the neurobiological problem, and bring that into what you do. And I think some of what we're trying to do is sort of understand the clinical, work closely with clinicians, understand the process that they're working with, try and replicate that, because ultimately what we're all trying to do is say that there aren't enough clinicians with the high level of expertise to identify very subtle lesions in the brain, for example. So there's clearly a need, but if you do it in a methods-driven way, you might not, and you don't understand the problem well enough, you may not get to the answer as quickly as if you have a clear understanding of the problem and you're driven specifically by the application.
Right. Yeah. Okay. Great answer. Yeah. Actually, maybe on a similar line, I considered organizing challenges and really going to the pain of doing this as a considerable service to the community, right? And maybe to the world, because it's not just our research community, but also clinical end users, industry, perhaps. And to this end, actually, I want to ask you, Gabriele, on this, because you've been developing a library for group convolutional neural networks, which I think is definitely gaining a lot of popularity, because now people don't have to develop this from scratch anymore. And I can also imagine that, you spend a lot of evenings like outside work, making this happen on top of your PhD. And I was just wondering, like, this is a great example of indeed an individual excited about, doing this, but it feels like every now and then, okay, a library makes it to this point where it gains some traction, and it seems a bit odd for us as a community to be dependent on these individuals that do this. So I just want to hear your thought. Maybe let's make this statement. First, your thought on this process, but also want to make the statement like industry or end users should actually fund these efforts. So I'm looking at you, and maybe Darko has some viewpoint from Qualcomm. I don't know. So first, you, Gabriele.
Thanks for the question. Yeah. Well, before that, I like to maybe connect with what you said before, because there's this nice connection on democratizing this process, like with these challenges, right? It's a way to provide data, right, to a community that maybe doesn't know anything about medical AI. I mean, I myself, I worked with medical data in the past without knowing exactly, the whole problems that comes with medical AI. But it's nice that, I can access this data, I can work with it, right? And I know, okay, there's someone that actually can turn this into something that is impactful, right? And I can focus on a much, simpler problem that allows me to get also in the field much more gradually, much more, and that just makes it much more accessible for the whole community, and that's quite cool, I would say. And in a similar way, I think also this contribution in open source libraries has a similar effect, because I mean, by my experience, right, getting into the field of geometric deep learning is quite challenging at the beginning. I mean, if you have never seen any of the math that is there, right, it takes some time to get used to it. It's like some new tool that is not part of, the typical program, computer science program, right? And if you can instead work immediately with these tools because you have a library that implements most of the complicated math and most of the, all the complex things, and you can already get started, that's a very nice way to get slowly into the field, get interested in the field, and understand if that's what you want to do before spending maybe months on that, right? So in these ways,
but that's,
that's
I think
a
that's
kind of similar notion, right?
I think that's a really nice viewpoint, and it also feels kind of altruistic, right? So you do this actually to do good to the world, but I was just wondering, shouldn't you be paid for this on top of this, or do you think maybe this incentivizes maybe others to go to the same effort? I don't know, like, should we somehow try to organize this a bit?
I mean, that would be cool. I mean, I think that the open source community, right, is quite a, I mean, has been existing for a long time, right? And this kind of maybe discussion is not new in this context, right? I mean, I myself am fortunately also part of Qualcomm, right? So, and that gives me also, this, and I get paid for my work, for my PhD, right? And so I can also work on, maintaining this library. That's like something nice. Still, like, I am doing this mostly on my own, right? So that's, of course, like a work in addition to my usual research work. And having, of course, like a bigger community supporting this would be cool also, even for that maybe week or two that I'm also having a deadline. So I'm definitely not going to look at all the issues on GitHub or things like that. That's a bit sad, because maybe when I have a deadline, also many other people that are using the library have a deadline, right? And maybe they need support in that precise moment. So it's cool. It would be cool, of course, if there's like a bigger community that can actually maintain this library such that it's independent from me.
I see,
Because before this happens, I think that the library needs to grow a bit bigger, right? Because right now there's, of course, many people working on it, but all those people are maybe people that just started, right? That got introduced in this field. But maybe, again, this is nice because the library is like a way to attract many people, right? And as they start working with it and they, do their own experiences, learn more about the technical details behind it, then they can actually support that, and the library will have a new life in a bigger community.
I see. I just want to check maybe with George, because you're closer to industry. So I'm not asking for funding this, but I'm just like get a view. Like, is this even feasible? Do you think companies such as Quantip or maybe other bigger companies maybe like would be open to this?
Qualcomm is supportive of you spending time out of your PhD time on building or maintaining this library.
Well, this is kind of part of my PhD. So that's how it kind of works. They don't mind me spending time on open source library. No, they are actually supporting me a lot on this. I mean, one thing that's also good for Qualcomm, right? This is a pretty good advertisement on Qualcomm. Other companies, right, support other big open source projects. Like, I mean, think about PyTorch or TensorFlow, right? That's not something
coming out. So Monai coming out of NVIDIA is another example to me. So we have one open source library at Quantip. So we built our own web-based viewer, and one of our engineers built a WebGL library for visualizing medical images, and part of that we open source. So we try to do these things when we have opportunity. But to me, so one is a matter of scale. So Quantip is about 45 people right now. I don't want to know how many NVIDIA has, or in Google you can look it up, but different scales. Qualcomm the same, different scale. So that's one part to it. But I think it helps once you get out of that first absolutely critical stage, like I could die any minute company stage. It's nice to be able to do something back, so we try to do that. And I think what you see is that a lot of these sort of open source things, but I know also out of Erasmus MC, there are some software libraries that are really just floating on one person's ambition to make a success out of that library. And that is, it can be a really rewarding model, but it's also quite dangerous. If you have, that's not enough. I mean, it's not for nothing that companies that consist of one employee are not very valued, right? There's just too much risk. And I think I see now that, for instance, around the eScience Center, you see a lot of focus on this research, what is it called? Scientific research engineer or research programmer. They have a real role for that. And there's like an actual profile that they're building in the Netherlands now. I see that across different organizations. And that part, the engineering part of it, has been undervalued, but I think it's getting better. And there's also just a group of people that I think really get a lot of reward, a lot of intrinsic reward out of supporting others in building great things. And I think one of the risks of funding things like that can be that the intrinsic, if you make it as something paid, and you lose the intrinsic reward, like I just want to make something great and help people, that can also be a very dangerous switch.
That's interesting, yeah. Yeah, that's actually good that you mentioned this. There are funds indeed for these kind of initiatives, so remember that also when you work on these kind of topics. And maybe then, as a question to you, Clarissa, as a more senior researcher, which has a better overview of what needs to be done, let's say, shouldn't be there more than responsibility with academic, let's say, later stage academic researchers to acquire these kind of fundings and set this up?
I think so, but I think that it doesn't rely on one person or one company or one hospital. Sure. I think it has to be something coming from a group of people, stakeholders, whatever you want to call it, that makes an effort to make this happen. I think we have seen now, for example, with these big models, for example, foundation models, that they are becoming there, or in other fields where this is being done, research that is being given back to the public. I think that we have a responsibility to do so, but also if we really want to move the field forward, I think that, for example, medical hospitals also have a responsibility. They really want to put the eye also on the systems to also support this kind of initiative. So indeed, I think it's also our responsibility, kind of, because we are more senior, maybe we have to do that, to try to move the community towards that goal.
No, I think, I mean, academics have so many different types of responsibilities, and this comes on top of it. So, but no, certainly, I think it's good to think about this, and maybe concretely while we're here, I mean, does anyone have any ideas to, because now we have this geometric deep learning field, and this works specifically on medical image analysis. I feel a bit like there should be maybe some central platform where we can collect interesting papers, interesting code bases, lecture materials, this kind of thing. But yeah, then questions then always, who's going to maintain it? And so yeah, and I think, please think about it. Like, there's plenty of interested researchers here at the moment, and okay, I'm not putting this on you, like, please organize yourselves and do this, but think about it. If you see opportunities, actually, and it could also work out nicely, you know, in your career, for that matter. But yeah. Yeah, I think it works.
Test.
Yeah.
Oh, okay. I have a question to the. How do you think? I mean, we are you guys doing really cool stuff. And in the medical setting, trust is much more important. Let me say, when coming to the entertainment area. And how geometric deep learning can impact in helping increasing the trust in AI methods in general. So at the moment when somehow, let me say, a doctor came from hospital and asking me what happens there and why the computer now, your algorithm says this should be critical, then we can't say that's black box. So at the moment I can't somehow capture the new thing which brings the geometric deep learning. Besides, I can now rotate the image and it gets still recognized. Okay, fine. Yes, there's a group action. I have group invariance. Cool. Yeah, but how we can somehow increase the trust because we want to apply the cool stuff, and in particular in the medical case, we should do something to give them a better image, I would say.
I see. So if I reduce the question, it's that are we communicating our work in a proper way? Is it,
how we can help to enlighten the black box for AI methods?
Okay, cool. I see. Christoph, did you want to respond to that, or did you?
So also responding a bit to the uncertainty quantification question that was in the plenary before, I think in that way also addressing the increase of trust maybe with the hospital. My suggestion would be to use the strength of graph neural networks that they could increase the interaction with the doctor, because you have also in graph the chance to target in a targeted manner, choose nodes or positions and select samples. So I think the discussion point I want to make is maybe graph neural network the right area to do interactive AI in a trustworthy manner? And is it better than with the classical methods, especially in view of uncertainty quantification and trust gain here?
Yeah, I see. If there's any response from the panel, then please feel free to do so.
Yeah, so about the explainability aspect, you hear this a lot, right? That we don't trust the deep network, whether it's equivariant or not, because we don't know what's going on inside. And I think, equivariant networks may address that a little bit. You can understand a little bit more in geometrical terms what it's doing inside. But that's a kind of knowledge that is maybe useful to an expert trying to debug a neural net, not so much to a clinician. And my view is still that the way to get to a point of trust should be through very rigorous empirical validation, not just on one test set, right? But so some out of distribution generalization is what you want to test. And then there's probably a job of convincing the users that should be sufficient. And that maybe I can give two arguments. On the one hand, we tend to think that humans are somehow interpretable, right? If you ask a doctor, why did you think that this lung nodule is malignant, then they can probably give you some story, but it's ultimately just a story. We know this from expert systems in the 80s, when people try to actually distill that knowledge from the experts and then write some rules, and it just doesn't work. Those are maybe useful for teaching, right? To point your attention as a student to certain aspects of the image, so you can learn how to recognize them. They're maybe useful for projecting confidence, right, and putting people at ease. Like, yes, I know what I'm actually an expert on this, and but it's ultimately not the reason why they classified it. They can't tell, because that's what's happening in your visual cortex. And the other part is back to the empirical validation. I think the real problem is that these things are not actually robust, right? So they get high performance on a test set. We're very good at that, and especially with these scaling laws, you can just say, well, we just train more and more and more, and in the end we get to almost zero errors, and then we should trust it. But then you get these silly things like the different scanners put a different logo in the bottom of the image, and in one hospital they had one scanner and the other, and there was a different proportion of positives and negatives, and then it starts to look at this. And no human would do that. And so I think there's really deep questions about how we should approach machine learning in general, very fundamental stuff, and a lot of work on just trying all sorts of tricks to make it more robust and validating those. And once we solve that, I think people will also get more confident, because let's put it this way: they have a point. They have a point to be mistrustful, because maybe it is going to fail catastrophically when something very trivial changes.
Yeah. So maybe it depends on the test that you want to solve indeed. But is trust really a barrier? And maybe Clarissa, you can.
Yeah, I would like to reply first to one comment that Taco said. So I think it's important. Well, first we heard in the pitch that there is no black box, so that's nice to hear that. But the reality is that when we go to a clinician, we want to implement it in the practice. One of the reasons that they come up is, but how do I know that this is working? This is a black box. They are using now as an excuse, but they are using it, and they have valid reasoning there. And I agree, we have to validate these models in a different way, a very rigorous way. Taking into account not only performance, and sometimes we just focus on performance so we can increase 0.01, but also taking into account other kinds of performance, like for example, like if we go to a different domain, how it performs, and or if we have a different data population, how it performs, is it biased or not. Those things we have to take into account. But there is also part of trust that is comes from the interpretability of the results. And we have to understand that these models are going to be set in a group of clinicians, so they need to communicate with clinicians because they have to make a decision together, because they usually make a decision together in a team. So they really need to work in a team. And second, we're also talking here about liability. So, I mean, this is one thing that if we really want to put this in the clinical workflow, if something happened, and if the model cannot explain what was the decision, we don't know who is liable here. So that's also important to take into account here. So yes, we have to validate, but interpretability is also important here. And to the question about what geometric deep learning can bring to impact clinicians, my field is not geometric deep learning, I have to admit this, so I also said to Eric what I'm doing here, but what I have seen, especially with the samples today, with things I have learned, I think I have seen, is that there are things, and also what Michael said in the last sentence, I think it was very important that there is a lack of knowledge there, that this can help, for example, to make, when you go to a different domain, can be more robust. We have seen that example today. Or when you have less data, it can be also important. That it can be, for example, very important for rare diseases that we don't have so much data there. So I think that it has an impact. But I think it's important to put it in the context of use to understand the impact. So it's so important to understand what the doctors need to make real impact there.
Yeah, cool. Great discussion that you started. Did you want to tune in on this?
So what I wanted to ask is, as was previously addressed, right now we only have invariance to things like rotation, but do you think that will expand to things that are more abstract? Like maybe we have invariance to gender, and wouldn't that then address our need for uncertainty if we have these guarantees and interpretability as well?
This feels a bit like a question maybe you, Taco, wants to answer.
So when you say gender, do you mean like you have a gender attribute in your data and you want to be insensitive to this, or more medical imaging and there might be visual differences?
Yeah, for me it's hard to come up with an example in medical imaging, but maybe you have a something that is you want it to be invariant to some attribute that is more abstract than just rotations, but has some like meaning to us humans. Something like gender, like if you're thinking something with societal impact, something like gender, something like race, things that we want to make not part of the
right. So when it comes to sort of sensory data, things like images and stuff like this, I think if you want to make your system work better by whatever metric, right, could be somehow informed by some fairness criteria, or it could just be some raw accuracy measure, you have to be very careful that the ideas that you have about how the system should work are actually accurate. So, you know, early on in computer vision, people really thought deeply about the geometry of the 3D scenes and different materials, and how you know, they had all sorts of very clever ways of recognizing very specific things using human intuition. And the big lesson of deep learning is, of course, that probably a lot of those are slightly wrong, and therefore it's better to get rid of them and just learn everything from data. And the reason I was working on symmetries is because I think there you have some chance that actually it's exactly true. Right? That it's exactly true that the image is still the same if you rotate it, something like that. Now, when you go to these more abstract attributes, probably you're dealing with more approximate symmetries, and then it becomes harder to build that into your network and show some benefit. That's one side of the answer, and then there's this whole field now of fairness in machine learning, and trying to make your your performance be equally distributed, or errors be equally distributed among privileged groups. I'm not an expert on it, but I think it's a valuable thing that we're trying. I do think you have to be very careful with those things, because even when you have provable guarantees about some unbiasedness, I think it always relies on some assumptions that go into your theorem. And in some cases, like let's say automatic screening of CVs, my opinion is even if you have a method that's supposed to be unbiased, maybe just don't do it with an algorithm, because for the same reason as it failing for silly reasons on medical imaging with the logo example, right? It can fail and be biased in obscure ways in those applications as well. And if it's not a problem that absolutely needs to be solved, maybe we just shouldn't do it. Nevertheless, I think this fairness research is very valuable.
All right, thanks. Maybe that's a nice point to close the discussion, actually, because I'm sure you're eager to learn who had the best paper at this workshop. So we'll move to that end. So I think thanks a lot for all the panelists for this discussion. I think for me it opened also some views on where we should be heading with the field and what are some technical challenges, not just technical, I mean, in the literal sense, but also how are we going to validate it? Apparently the fixed test set they construct is not really good at validating where we're at, and so there's all sort of things. Also, when it comes to community, and if you want to build upon each other's research, maybe as a community should put some more collective effort in guiding this. And I think we had some nice ideas here today. And also, you met each other at the poster session. We have drinks in a minute. Remember, like you're all part of this, reach out to each other, reach out to me if you think of certain initiatives, and let's see what we can do about it. And yeah, so thank you all for listening in, and thanks again for the panelists. Thanks. All right. Yeah, so let's clear the stage. Be careful for the glass. I guess there's nothing better than a conference ending up with broken and shattered glass everywhere. No, okay, back to business. Jelmer, you're putting up the slides, I think?
I am.
Because I would like to hand this over to you.
Can I have one of your mics?
Yeah. Oh.
So we have to be a bit careful not to show the winner yet.
So,
yes. Okay, so what we can do is, because we all want to go to the drinks, we'll just do it like this. So you can see this, I can see this. So we want to start with thanking some people. So we've already, of course, thanked our funders, but we also want to thank the program committee. So that's the people who actually reviewed all the papers that you've seen today. We're very grateful for them to be able to review these papers on such short notice, and that we've also been able to collect a group of experts really working at the intersection of geometric deep learning and medical image analysis. So big round of applause. Many of them are here in the room. And they helped us to a large extent to actually find out who is the winner, because we based our decision, of course, on the reviews. And we have a bit of a teaser here. The award actually is a GPU. Oh, that's the 30.
This is what it looks like when it's in the box and not in your PC.
It's the 3090 that we all dream of course. And based on the reviewer ratings, we made a pre-selection. So we have a short list of 3 papers that did very well in the review round. that you've also seen presented here. So the first one was presented in the first oral session online, unfortunately, by Yung Lang, scale effect equivariant UNet for histopatology image segmentation. The second one you've seen on the cortical surface registration, GeoMorph. The third one, of course, you've also seen presented. Excellent presentation on structured knowledge graphs for classifying unseen patterns in radiographs. So without further ado, I've put a lot of empty slides here to prevent any spoilers, but at some point it will appear. The winner of the Best Paper Award is the GeoMorph paper. So
please. Okay.
Please come forward, and if someone can take some nice pictures, maybe.
Yeah. So, no, congrats, Mohammed. So, maybe Taco can. I see with the camera. So, yeah, congratulations, Mohammed. It was. I mean, apart from just receiving good reviews, so the others did as well. And I think in the selection we saw a nice coverage of the entire field of geometric deep learning for medical images, from groups to graphs and whatnot. And in your paper, I think it comes together in many forms, which is really exciting, and it's a new application of a recent technology within our field. And so, yeah, I think props for that, and congratulations. You have anything to add to that, Jan?
Well, I just want to add that I agree. It's really perfectly at what we were hoping for, what was the scope of this workshop. And big congratulations, but also congratulations for all the other authors that submitted their work here, that presented their work here today. For all of you, for being present also here today. So there was a lot of nice interactions also at the posters, which was great. But also big thanks for, because we're really nearing the end here, to Erik and to Angelica Aviles Rivero. So with the three of us, we had many online meetings, so it's great to actually be here physically. Yeah. In preparing this, so it started a year ago, this idea, and it's really great to see it culminate in such a nice in-person meeting with so many people here today. So thank you.
Thank you. And of course, you don't thank yourself, but really, Jelmer had a major role in getting the proceedings published at PMLR, so this is really nice. And of course, your contribution, reviews, but also submitters, we were really happy to see such high-quality contributions. So thank you to Jelmer and to you all for this. So, thanks.
Yeah. Thank you indeed. And I think, do we have anything else?
Drinks?
I think drinks, right?
Yeah.
Yes.
Okay. Sir.
Oh, okay.
Yes.
Yeah. First of all, thanks everyone for coming to the workshop. It was a hard decision, first of all, to decide for the winner. So we are very happy with all the papers, and it was, and we ensure that it was a hard decision to select the winner. And thank you very much for participating. It was an overwhelming reception, and how many submissions. When we started, we thought about, okay, we might have like a small room for that, and now we take a look about it, and this is bigger than we expected. So thanks, everyone.
Thank you. Yeah. Okay, so off to drinks.
The bar is open.